Be talking about building panels for flow cytometry, key steps to success. So I do want to note that I do work for SciTech, but everything that, or just about everything that I cover today should be applicable to all platforms and all panels. So whether or not you're using a conventional cytometer, a full spectrum cytometer, if you're interested in something that's a bit smaller or a larger panel, I'm hoping to cover a whole range of things. But of course, with some panels, you might need targeted strategies. And so as we go through this talk, I am going to go through three scenarios, which are pretty common in panel design. So the first one is a smaller, simple panel. The second one, so the first one is really just the foundational knowledge for everyone out there. And then for those of you who would like to do something that's a little bit larger, a little bit more complex, I will talk about some strategies that are a little bit more specific to that scenario. And then the last scenario is for those of you who have cells with very high autofluorescence, you might want to take some additional strategies to handle that. So we'll talk through those three scenarios and I'll call out those scenarios when we get to that part of the talk. So as an overview, there are four main steps to panel design, and it should be a very informed process. So the first step is where you gather information about the biology. So what is your scientific question? What kind of populations are you interested in looking at? The more information we have about that, the better. The second is where we understand the fluorochromes. What fluorochromes can be run on your specific cytometer? How do they interact with each other? Things like that. So those first two steps really are gathering as much information as possible. Then we can get to the third step where we pair that information together. So we're going to pair markers and fluorochromes together to design our panel. Now, this is not the end of the panel design process. There is one more step that is often forgotten, and that is the optimization. So you shouldn't just stop at the theoretical panel. You should also do an experiment to see if your panel is actually behaving as you expect it to behave. So we're going to go through these steps in a little bit more detail. Starting with the biology, and I'm kind of going to assume that a lot of you have already designed panels, so we're going to speed through this pretty quickly so we can get to the more fun stuff at designing a panel. So first off, we definitely want to understand our scientific question. Very, very important to have that very clear in your mind so you know what markers you need to put in your panel, what populations you're interested in. And you might choose, depending on if you're doing an immunophenotyping panel, you might choose to further categorize those into lineage markers, additional markers, exclusion markers, other markers, like here we're showing some cytokine receptors. Things like that can kind of help you when you're designing your panel. You also want to think about the cell lineage, specifically co-expression is the type of information we're trying to gather. So whether you have it kind of arranged in your mind, something like this on the left, or if you have it on the right in a gating strategy. So some people have some prior data maybe that they're using, so there's actual data to gate, or if you even just kind of roughly do something like this. So you have an estimate of what populations you're going to be gating. That can help you really understand the co-expression. And I would say it is a nice practice to do this, even if you are planning on doing like the algorithmic analysis later on. Just having this for a co-expression context can be helpful. We also want to understand the reagents. So reagent availability is definitely a thing that is very good to understand. For example, if you have one antibody that only comes in 2 fluorochromes, it would be very important to understand that before you get too far into designing your panel. If you have information about clones that work best for your assay, that's very good to know in advance. And also thinking about your experimental conditions. So maybe you have stimulated cells and unstimulated cells, and you know that with stimulation, some of your markers are going to be changing their level of expression. If you have that kind of understanding in advance, that can be very helpful. Of course, that might be your experiment to find out what happens to those markers. But anything you can understand in advance about your markers and panel is going to be really helpful. You can find lots of information on vendor websites or in journals. For example, in the Cytometry Part A journal, let's say you're interested in NK cells, you might find this paper interesting. You might be interested in looking at the OMIPs, or the Optimized Multicolor Immunofluorescence Panels. Those can be a good resource of information. If you're looking at human markers, this paper is helpful. So the dynamic profiling of CD1 through 100 surface expression on human leukocyte and lymphocyte subsets. Okay, once you have your markers, you want to categorize them. So this is something that was defined in this paper, noted at the bottom here. There are 3 categories. So the first one is primary, and these are markers that have a on-off expression. So they either have the marker or they don't have the marker, and there isn't really an in-between. Oftentimes, these are markers that are very highly expressed, for example, CD4, CD3, things like that. The secondary markers have more of a continuous expression. So there's a lot of variability in their expression, or they may have more of an intermediate density. And then the tertiary markers have low density or unknown density. Of course, I would say that if you have markers in your panel that are unknown to you, I would try to do some research, see if you can figure out if anybody out there knows. But if that is the nature of your experiment, you're trying to understand the expression level. If you have a few unknowns, that's perfectly reasonable to put them in the tertiary category. So the reason why it's important to have these categories is because when you get to pairing these markers with fluorochromes, we definitely need to make sure that the tertiary markers are paired with the brightest fluorochromes we have available. If we pair these tertiary markers with a dim fluorochrome, then you're not going to be able to get the resolution that is going to fully capture that low density antigen. Okay, moving on to the fluorochromes. There are 2 components to this. So first you need to understand your own instrument, and then I will note on a few criteria that can be used to select fluorochromes. So for panels, you should really be designing a panel specifically for the cytometer that you're using. And there are 2 things to think about when you are trying to understand the specific cytometer. So the first is what lasers are available to excite the fluorophores. What are those wavelengths of those lasers? And the second component is how are the detectors arranged to collect the fluorescent emission. So this is probably the main difference between a conventional or non-spectral instrument compared to a spectral instrument. So if you have a spectral system, for example, if you have a Cytec Aurora, then really it's very straightforward. You just need to know what lasers are available. So if you have a laser that excites the fluorochrome you're trying to use, then it can be picked up. If you have a non-spectral instrument, then you are going to need to know exactly what detectors are on your instrument and how the wavelengths that those detectors capture correspond to the peak emission of the fluorochromes that you're trying to run in your assay. So I'm not really going to touch too much on those because that's just going to be very specific to whatever cytometer you're using. So please go find that out if you don't know it already. But if you're, for example, using a 5-laser Cytec Aurora, it can be very straightforward because if you have a UV laser, for example, then that will correspond to having a UV detector module, which has a bunch of detectors inside of it. And then all of the detectors on the cytometer are going to be utilized to look at every single fluorophore that you run on the system. So that means when you are using something like a 5-laser Aurora, when you look at an individual fluorochrome, you will capture a signature of it. So we have a spectral plot here with 64 detectors at the bottom. We have intensity on the y-axis here. And so this is what this fluorochrome looks like. We're using all of the detectors to capture that information. Compared to a non-spectral instrument, you would instead really just be picking up information like this. And so that's why it's really important to understand what wavelengths is your detector covering, and so you can match that with the peak emission of that fluorochrome. On to the criteria for selecting fluorochromes. So there are a few characteristics that you can consider. The first is uniqueness. So you do want to make sure that each fluorochrome that you are putting into your assay is going to have a unique spectrum given the cytometer that you are using. And so I will mention a few criteria that we use to assess uniqueness. There's also the brightness, which is typically assessed with the stain index calculation. And so I know Cytec will provide stain index charts. A lot of vendors, I think, will typically do this. So again, that's just understanding the cytometer that you're using. And the last one is spread. It can be helpful to have an understanding of how fluorochromes on your specific cytometer will spread into each other. And I will go through all of these things in a little bit more detail in a moment. So I will say, if you are using a Cytec system, we have all of our tools in Cytec Cloud, specifically the panel builder. And so it combines all of the tools in a singular place, and it gives you a lot of flexibility and where to start, so you can build custom panels from scratch. There are pre-designed panels in there, so a couple of them. So if you want to start with those panels and then modify them, you have that option. Or if you're looking to transfer from a different cytometer and use a Cytec system, then you can certainly enter your current panel and then utilize our tools to see how it might behave on our system. So just a quick overview of the criteria that we have to assess fluorochromes. The first one is called a similarity index, which can be accessed by clicking this tab. We show it to you in a matrix. And so this index is a measure of uniqueness between two dyes. So if the value of the similarity index is 1, that means those dyes are essentially identical, so you should not be putting them in the same panel. The second one is the complexity index, which instead of comparing just 2 fluorochromes, will instead compare the entire panel of fluorochromes that you have entered. And then we also have our panel matrix, which is, I'll show you a few examples in a moment. But this is a nice way to get a quick overview of your panel. We place the fluorochromes in this matrix based on the column and the row. So the column is going to be the primary exciting laser. So if we just focus in on C-Fluor V420, it is going to be in the violet laser column because it's primarily excited by the violet laser. And then it's going to be in this row for 440 because that is the approximate emission maxima. Another thing that is included in the panel matrix when you're looking at Cytec Cloud is the stain index. So you'll notice colored bars below each fluorochrome in this Cytec Cloud. And so you'll see here that C-Fluor R668 is a little bit brighter than C-Fluor B515 because that bar is a little bit larger under C-Fluor R668. And then the last metric we have is stain index reduction, or SIR, which can be used to assess potential areas of spread. So I know I went through those pretty quickly. If you would like to learn a little bit more details about all of those metrics and how you might use them, we do have a really nice video series, and there's a video that goes through everything in more detail. Okay, let's get to our 3 scenarios. So we're going to start here. You'll notice the little tags in the corner here. So we're starting with our scenario of having a smaller, simple panel. And this is really the foundational best practices for everyone. So if you have something small, let's say you have a 5 laser system, and you just want to run 5 colors on it, then the simplest way to do that is to pick fluorochromes that are excited by different lasers and have different emission maxima. So we can see our 5 fluorochromes selected here are on different lasers, and then the emission maxima is that number at the top there, and we can see that they're all different. And just looking at this, these normalized signatures, we can see that they're all very spaced out from each other, and there's not really a lot of overlap between each of these peaks. So that's the easiest place to start. As an example, if you were to instead choose these 5 fluorochromes, which it's the same 5 laser system, but now we have 5 fluorochromes that are excited by the same laser, we can see now there's more overlap between these peaks. Now, this is not to say that this is an impossible panel. You could certainly get reasonable data with this panel. or with both panels. But the thing about the panel on the right here is that because there's more overlap, because they're all excited by the same laser, they're not really fully spaced out. It's just a little bit harder. And my goal is to make your life as easy as possible. And so that's why I would recommend the one on the left. Okay, moving on to our larger complex panel scenario. Now we're going to do 15 colors, and I'm going to show it to you in the panel matrix. So we can see pretty quickly using this panel matrix that all of the selected fluorochromes are spread out throughout the matrix. So they're in different columns, different primary exciting laser, and they're also in different rows. So their approximate emission maxima is a little bit different. You'll also notice that the complexity index for this particular group of fluorochromes is 4.1. So now let's compare that to a different panel, same numbers. So we have 15 colors still, but now you can see that I've clustered everything into basically the center of this matrix. So we have clusters where there's 3 fluorochromes stacked on top of each other here and here. So 2 groups of those on the blue laser. We also have regions where there are multiple fluorochromes that are sharing the same emission maxima. And so because everything is very clustered, we can also see that our complexity index is now 12.96. So the thing with the complexity index, I kind of think of it as a metric of difficulty level or probability of success. So it's just if you have a value that is lower, it just means that your life is going to be a bit easier. You don't have to think so hard. If it's higher, it doesn't necessarily mean that it's a bad panel or it's not going to work. Certainly this panel here is not an impossible panel. It's just a lot harder than the previous panel. Now, you might be wondering, why is it harder? That has to do with something called spread, or actually the official name for that is spillover spreading error. And that has to do with a few factors, one of them being spectral overlap. So we have an example here on the left where we have 2 fluorochromes. We have APC in orange. We have PE Sci5 in blue. And this was run on a 3-laser Aurora, although honestly, for me, I think it's a little bit easier to think about spread when you're talking about a non-spectral system. So you can kind of imagine converting this into a non-spectral system if you want. So we have 3 rectangles here. So you can imagine that those 3 rectangles are 3 detectors on a non-spectral system. So if we focus on this middle one here, we can see that this middle box captures the peak emission of PE Sci5. So if we're on a non-spectral system, this is the detector that we would assign to PE Sci5. And we can see that also the APC in that box is very, very low. So there isn't really a whole lot of signal from APC. But if we look at the red box, we can see that this is where the emission maxima of APC is. So we'd probably assign this detector on a non-spectral system to APC. But we're also seeing quite a bit of signal coming from PE Sci5 in this box as well. So if we put this all together in a sentence, we would say that PE Sci5 spills into APC. So that is before we have done any sort of compensation or unmixing, no calculations have been done. So then once we apply the compensation or unmixing, that is when you'll be able to see the spread in the data set. Kind of applying that math will sort of reveal these patterns. So if we look at this population, the APC positive population, we can see that there's minimal spread. So that population is very compact and round. And so that corresponds to this box here that APC does not spread into PE Sci5. But if we look at the other one, now we can see the spread. So instead of that compact population, now it's a little bit more diffuse, stretched out. And so that is the spread we are talking about. And so PE Sci5 is spreading into the APC channel, which we can correspond to that. So as I said, there are multiple factors that impact spread. One of them is spectral overlap, which we just went through, but there are other factors which contribute to the final intensity of the signal. So one of those factors is the brightness of the impacting floor is going to impact how bright the final intensity is, as well as the antigen density is another thing that will impact that final intensity. So this last plot here shows a nice classical pattern, which some people call the trumpet effect or the umbrella effect, where as that signal gets brighter and brighter, you will see more and more spread. So that tool that I had mentioned earlier, the SIR matrix that is in Cytek Cloud, that is something that can be used to help us identify potential areas for spread. So this is an example SIR matrix, and the darker the red, the more potential there is for spread. So the way to read this is that the dye in the row impacts the dye in the column. So let's look at an example. We have seafloor B515, and let's see how it impacts seafloor V420. So that's a white box. There's no impact in that case. And if we look at corresponding data, we can see here that our population is nice and compact, not a lot of spread issues there. But if we choose a different column, if we compare it against seafloor B532, so now we can see that there is a lot more spread in this case compared to in this case. So when we are designing our panel, we need to consider that seafloor B515 could potentially impact the resolution of whatever marker we have assigned to seafloor B532. And so if we try to think about how we're placing that marker with these fluorochromes, so if you pair your marker with seafloor V420, then we don't really have any concerns about where this double positive population is going to land. If it ends up down here or up here, because we don't have any spread, we should be able to resolve that population no problem. With this other fluorochrome, seafloor B532, because there is spread here, now we're going to have to be aware that if we place it on a marker that is too dim, and it ends up that this double positive population overlaps with the spread of the single positive population, now we will not be able to properly gate these two populations separate from each other. So going back to our two 15-color panels, and I told you that panel A is much easier to design compared to panel B. The reason for that has to do with spread. And just quickly, hopefully you can appreciate that panel B has a lot of red in this SIR matrix compared to panel A, which has a lot more white boxes. So again, both of these are not impossible panels, but if you want panel B to work properly, you're going to have to be very, very strategic about how you assign your markers and how you work around all of this spread that could potentially mess up your resolution. Okay, on to our third scenario for those of you who have cells with very high autofluorescence. Now, the first thing we need to know is, are you in this situation or not? So if you have no idea, and I will say that this scenario is better handled by a spectral system. So I would say if you have a lot of highly autofluorescent samples, then you might want to see if you can find a spectral system to help you out. But if you run your unstained cells prior to any sort of panel design, then you can hopefully, depending on what system you're running, determine if your cells have a lot of autofluorescence or not, and figure out which channels have the most autofluorescence. And I will note that you want to run as many events as possible because you might have, depending on what tissue, some very rare but highly autofluorescent samples. You don't want those to suddenly pop up if you don't run enough events in the beginning. So if you're running on a 5-laser aurora, your red blood cells might look something like this. They're not very autofluorescent at all. Remember, on these spectral plots, the intensity is on the y-axis. Lymphocytes are certainly a little bit higher than red blood cells, but we typically categorize lymphocytes as low autofluorescent cells. And then our alveolar macrophages here have quite a bit of autofluorescence. Remember, these are unstained, so we're getting a lot of signal in pretty much every detector. So it can be really helpful to understand that before you do any sort of panel design. So keep in mind that if you are in the case of having a lot of autofluorescence, there is a chance that depending on where that autofluorescence is, you might have issues with some of these fluorochromes. So this is not a always scenario, but it's just giving you a heads up that some of these fluorochromes, depending on your assay, could have reduced resolution because you have so much autofluorescence in a similar channel. Okay, let's get on to the fun part where we design some example panels. So I'm going to go through designing panels in our 3 different scenarios, and then how you would kind of theoretically QC those panels. So let's go back to our smaller, simple panel situation. And again, these are the same whether you're using a non-spectral system or a spectral system. So starting out, we want to place our tertiary antigens because oftentimes those tertiary antigens are the ones that have limiting reagents. Maybe they only come in 1 or 2 fluorochromes. And we also want to make sure that we have paired them with the brightest fluorochromes available. Then moving on to the secondary antigens, you can just work your way down the list of brightness and pair those secondary antigens with whatever bright fluorochromes you have left, or any mid-bright dyes also work. And then finally, our primary antigens, which are typically available in many colors. You can assign those to whatever you have remaining. Oftentimes, the dim dyes will work well for those, but for smaller panels, you don't necessarily have to think too hard about this. Just making sure that the tertiary antigens are definitely on the brightest fluorochromes. So let's go through an example. We have 7 colors here. Our CD25, we're going to classify that one as tertiary because we don't have a lot of positivity there. In the middle row, we have our secondary antigens. So these are either like an intermediate density or they have more of a variable range of positive and negative cells. And then on the bottom row is our primary antigens, where we have that very clear on-off pattern, and it's very high antigen density. We also have our gating strategy. So we are interested in Tregs as well as some T cell subsets for this particular panel. So now going through the assignment, we're going to start with our tertiary antigens, right? So we're going to pick the brightest fluorochrome we have available, which is PE. And as we go through these, the bar is going to indicate how bright that fluorochrome is on a 5-laser aurora. Our secondary antigens, we're going to assign mid-bright fluors. So we're going to choose BV421, APC, and APC size 7. And then our primary antigens, we're going to go with mid or dim fluorochromes. So we're going to go with BV510, FITSI, and PERCP. And then we do have one last thing to assign, which is our viability dye. Definitely recommend that every panel has a viability dye in there. So we're going to go with ViaDye Red. And then we can also just quickly see that we have pretty well spaced out our fluorochrome. So we have one fluorochrome that's on the yellow-green laser. So I'm looking at the colors here. We have 2 fluorochromes, BV421 and BV510, that are on the violet laser. We have 2 fluorochromes on the red laser, actually viability as well as on the red laser. And then we have 2 that are off of the blue laser. So it's a smaller panel. We don't have to overthink this. It's pretty well spaced out. So we are good with this 7-color panel. So now let's move on to something that's a little bit larger, a little more complex, and see how this is now going to become a little bit more strategic. So the first step is going to be the same, pairing those tertiary antigens with the brightest fluorochrome available. The secondary antigens is now where we're going to have to think a little bit more strategically. So we're going to have to pay more attention to the co-expression. As your panel gets larger, it's going to be more and more important that you think about co-expression, more and more important that you think about spread. So if there is no co-expression between the marker that you're trying to place and markers that you've already placed, then you can just use any bright dye that you have available. But there is co-expression, then you're going to be a little bit more strategic. So you're going to try to assess the spread. Are there any issues with spread and the markers that you have already placed? And if there is spread, is there any way that you can kind of strategically place it? For example, using something that's a little bit dimmer, because remember that the final intensity of that signal is going to impact how much spread that you get. And then lastly, our primary antigens. You do want to be aware of spread for those ones. And so pairing the primary antigens with dim fluorochromes is going to be a lot more critical in these more complex panels. If you pair a primary antigen with a very bright fluorochrome, then you can end up with a huge mess of spread. And so try to avoid that case. So for our example panel, we have a 19 color panel. You can see we've categorized them already. And this is going to be a panel where we're going to have a unstimulated condition as well as a stimulated condition. So you might notice at the bottom here, we have two markers that have two classes. And the reason for that is that in the unstimulated condition, these two markers are primary, but in the stimulated condition, we do know that these markers get internalized, so they become tertiary. So it's important to understand that as we design our panel. Okay, so we're going to do this in the panel matrix, and you can see that we've started out placing our tertiary antigens with bright fluorochromes. Remember, we have that bar underneath each fluorochrome. We pick PE, APC, and APC R700. Then we're also going to pair those, the CD3 and the CD4, because those had both the tertiary and primary. It's important to accommodate the lowest density antigen. So if you have a case where your marker is kind of changing depending on your treatment, then pair it as though it was at the lower density antigen. Then we're going to pair our secondary antigens. And as we go through each one of these, we would be comparing to the SIR matrix. And so if we're placing CCR7, for example, and we look across the row for CCR7 to see how that is impacting any other fluorochromes, we can see that especially our tertiary markers, which are these 5 listed at the top here, we don't have any values. It's a white box. So the placement of CCR7 is not going to have any sort of impact in terms of spread on our tertiary markers, which is a great position to be in. And then finally, we're going to place our primary antigens. So those are going to be on dim fluorochromes. We've picked BUV 615, PerCP, and PerCP Bio 700. And as we place those, so for example, our CD45, we're going to place that, see how, looking across the row, this is hard to do on a trackpad, and see if there's any large values. Now, this is a percentage. So your maximum is 100%. 50 is, not the highest. And so we can keep that in mind. We also can try to logic our way through how it's going to impact the marker in the column, which is receiving the spread. And if you recall, we have categorized TCR-V delta 1 as a primary marker. So it should have really good separation, very clear, distinct positive population that's away from the negative. So primary markers can usually handle a little bit of spread. So the negative population might spread out a little bit, but we should still be able to gate the positive. And so that fact, and also knowing that this is only 50%, which is not the highest value we could get, I think that our risk for not being able to resolve TCR-V delta 1 is going to be pretty low. So we're just going to stick with this and see how it goes when we get to the real data. And then the last thing we want to do is include the viability. So we're in this panel going to go with live dead blue. Now, you might think to look at the SIR matrix for a viability dye and get a little worried that we have quite a few high values, but the reason why we place the viability dye last is because as long as you've titrated your viability dye properly, all of your dead cells, your positive cells, are going to be gated away. So they're not going to be co-expressed with any markers in your panel. Therefore, we can fully ignore the entire row. So anything that is not co-expressed, just ignore that box. So once we have our panel fully designed, then we can do a final assessment. So I like to look at the SIR matrix as my final step. And so we could just look through the matrix for any sort of high values. There's kind of a like no hard cutoff. As I mentioned, there's a lot of factors that go into spread. So it's not the easiest thing to fully be able to predict in a theoretical scenario. But I guess I'd probably kind of go for 70s, maybe down to 60s. So I found 4 high values in this matrix. And then this is where either you try to think a little bit harder about, how much spread could I potentially get? I think I forgot to mention these matrices that we show in SciTech Cloud are made based on CD4. So it's a very highly expressed marker. And that's why it's just a guide, because if you were to actually recreate this SIR matrix with your actual panel, you shouldn't be getting values that are as high as this. So you can either try to see if there's areas where you're thinking you're very concerned and you really don't think you're going to get the resolution you're hoping for, and maybe rearrange things a bit more. Or you can just kind of keep those in your mind as places that are areas of concern. And then when you get to testing out your panel, then you'll know that those are areas where you are expecting potentially some issues with resolution, and you'll see how it goes. Okay, our last scenario for those of you who have very high autofluorescence is you could be a bit strategic. So let's say you're looking at unstained whole blood. So you've got both neutrophils and lymphocytes in your sample. And so we just isolated for you. Here's the signature of neutrophils on the top that's unstained. So a lot of autofluorescence from the neutrophils. The lymphocytes are a lot lower. So if we're trying to pair our BV510 with a marker on this tissue, if we were to pair it with a neutrophil marker, you would notice that we're going to have to make sure that that marker is going to stain at or above 10 to the 4, because we need it to stain above the background autofluorescence. Whereas if we instead paired it with a lymphocyte marker, because that lymphocyte autofluorescence is lower, then it's going to be a little bit easier for us to get a signal that is above background for that cell type. So that's one strategy you could take, is pair markers based on their level of autofluorescence. Or if you want to take it as far, or maybe if you have a non-spectral system and you don't have the luxury of doing autofluorescence extraction, then you might have some specific fluorochromes that you just fully avoid. For example, you can see that BV510 signature is quite similar to autofluorescence. So some people might say, I just have a lot of autofluorescence, so I'm just going to not put BV510 in my panel at all. Another thing you can think about is placement of viability dye. So we have our unstained cells on the left here. This is actually a heterogeneous sample. So there's one very bright signature at the top, and then there's another dimmer signature at the bottom. These plots are like a heat map density plot. So just the highest signature is low density in this sample, or low frequency. So if we overlay it with our viability dyes, we can see that for the top two, these are staining just above the background autofluorescence. And even just the general signature of that viability dye is kind of similar to the general signature of the unstained. Whereas if we instead select this third dye, which is primarily excited by the red laser, there's not a lot of autofluorescence off of the red laser. So it's going to be easier to get a signal that is above background. And this signature is very, very distinct from the autofluorescence. So if you have a lot of highly autofluorescent cells, I would definitely recommend considering a viability dye that is excited off of the red laser. It's going to make your life just a lot easier. Okay, so we got to our final step, which is the optimization. So now we've got everything done theoretically, but how does it actually work on real cells? How do we know if our panel is truly successful? And that is where we determine if our panel can resolve all populations and markers of interest. If it can, then our panel is successful. So you should plan on doing some experiments to make sure that you can determine this. We call this assay optimization. So it's probably going to take you a few experiments. The first one being you need to optimize the individual reagents. So Typically, the name assigned to that is antibody titration, where you're trying to figure out the most optimal antibody concentration. But while you're doing that, you can also think about additional things like, did I assign the appropriate fluorochrome? Is the fluorochrome too bright? Is it too dim? Did I select the best clone? Am I getting an expected pattern? So once you have that optimal single color resolution, then you can combine everything together. And the first thing you want to do is optimize your reference controls or compensation controls, and make sure that you're getting accurate unmixing or compensation, whatever math you are using on your samples. And then once your compensation or unmixing is correct, then we can actually analyze the data and make sure we're getting optimal multicolor staining and make sure that we're resolving all of our populations and markers of interest. So one way we do this is to set up an experiment where we have single stain cells and multicolor stain cells that are exactly the same. The only difference is the number of antibodies in the tube. And so in our first example for CD45RA, we can see that the resolution is the same, so we're happy with this placement. The second one, we can now see that the multicolored, the width of the negative is a little bit wider in the multicolor compared to the single stain. Sometimes it's hard to tell things in a histogram, so if we instead switch over to our dot plot, we can see that we can still resolve our positive population, so we're happy with this. We don't need to change anything. We have that same pattern here with the CD25 where the width of the negative is wider in the multicolor. And so this one we're going to need to investigate a little bit further. And then this last one, instead of the negative, we see that the positive population, the intensity of the positive is dimmer in the multicolor compared to the single stain. So that's more of a staining issue. It's not really a panel design issue. So if you have this outcome, then investigate your staining. So let's investigate this one a little bit further because this could have to do with your panel. So the next steps I would take is gating your population of interest, do your regular gating, see what you find, because it might be that the source of that spread is not co-expressed. But if you cannot resolve your population, then that's the time when you might need to change your panel, and you have data that is telling you that your panel needs to be optimized further. So just quickly with that CD25, our single stain is at the top here. Here's our multicolor. If we were to look at these two fluorochromes, if we hadn't done any gating, we can see that there's quite a bit of spread here, which is causing problems. But if we, instead of just looking at all cells, if we instead gated on our population of interest, which is our CD3 positive, CD4 positive cells, now we can see that there's minimal spread and we can see this nice positive population is clearly resolvable. And we can even tell this from our SIR matrix. If we look down the column of CD25 to see what could impact the resolution of CD25, we can find that our CD16 can. And if we look at the data, we can see, yep, that is where our spread is coming from. It's coming from the CD16, but there's no double positive population. So that's why we ignored that box. And that's why this is a good panel design. And then remember, I had said if you have some areas where you're a little bit concerned based on the SIR matrix, you can just check those specifically. So this 95% might be concerning in your SIR matrix. But if we gate that, now we can see we can definitely resolve that population. So even though there's potential for spread, we have been strategic about how we designed it. So we're happy with that. So just to wrap up, we do have quite a bit of tools available for you. If you want some more beginner stuff, we do have a series called the Spectral Learn Panel Design Series, which is short videos that has a lot of great information, again, applicable to all systems. And then we also, if you are specifically using a Cytec system, you can use Cytec Cloud and Panel Builder. And I will let you guys know that we do have a new tool that is going to be coming out soon. It's not out yet. It's called SpectroPanel, and it's a tool that will be able to accelerate your panel design process. So you'll be able to get to a final result much faster. So that'll be really exciting when that comes out. So keep an ear out for that. And so to summarize, all of these panel design fundamentals apply to all systems, all panel sizes, but depending on your situation, you might want to use some additional strategies specific to, larger panels or high autofluorescence. And then don't forget that panel optimization experiment, very, very important. That's all I have. I don't know if there's time for questions or are we moving on? And so they can hear the question. Thank you.
So our next speaker is Garrett Collette from Bristol Myers Squibb. And we're just going to do a quick switch over.
I just grab this.
You can just grab that. Yeah. And I'm just going to plug this in. So it's
Oh, yeah, sure thing.
okay. So we're just going to switch you on. Okay. Great.
Great, thanks. Hi, everyone. You guys can hear me in the back? Okay, great. Thanks, Laura, and thanks, organizers, for having me here today. This is my first time at the conference, so cool venue and great to be here in front of you all. So like Michelle said, my name is Garrett. I'm a scientist at Bristol Myers Squibb in the Legacy Juno Therapeutics site over on Dexter Ave. And today I'm really excited to be talking to you all about applications for spectral cytometry in the CAR T characterization world. So just a quick agenda for today. I'm going to start by giving a brief background into what cell therapy is. I imagine many of you are pretty familiar with the technology, but in case there are those of you that don't, I'll just do a quick review of the tech. I'll talk about BMS's footprint in cell therapy and spend a little bit of time talking about analytical strategy. But I'll reserve the bulk of the time for today's talk by talking about specific applications for spectral cytometry. So I'll hit a few development highlights. I'll talk really briefly about high-dimensional analysis, and I'll reserve some time at the end to talk about a specific technique developed here at Fred Hutch by Mark Headley and his group called Infinity Flow. And I'll wrap up with some concluding remarks. And I have a lot of content here, so I'm going to hustle through these. So if we could save questions for the end, that would be great. So like I said, just really brief CAR T background. Generally, patients or healthy donors are leukapheresed to get a nice, rich, a fraction that's really rich in leukocytes. After the apheresis is collected, generally the T cells are selected out of the apheresis. Great, thanks. Hi, everyone. You guys can hear me in the back? Okay, great. Thanks, Laura, and thanks, organizers, for having me here today. This is my first time at the conference, so cool venue and great to be here in front of you all. So like Michelle said, my name is Garrett. I'm a scientist at Bristol Myers Squibb in the Legacy Juno Therapeutics site over on Dexter Ave. And today I'm really excited to be talking to you all about applications for spectral cytometry in the CAR T characterization world. So just a quick agenda for today. I'm going to start by giving a brief background into what cell therapy is. I imagine many of you are pretty familiar with the technology, but in case there are those of you that don't, I'll just do a quick review of the tech. I'll talk about BMS's footprint in cell therapy and spend a little bit of time talking about analytical strategy. But I'll reserve the bulk of the time for today's talk by talking about specific applications for spectral cytometry. So I'll hit a few development highlights. I'll talk really briefly about high-dimensional analysis, and I'll reserve some time at the end to talk about a specific technique developed here at Fred Hutch by Mark Headley and his group called Infinity Flow. And I'll wrap up with some concluding remarks. And I have a lot of content here, so I'm going to hustle through these. So if we could save questions for the end, that would be great. So like I said, just really brief CAR T background. Generally, patients or healthy donors are leukapheresed to get a nice, rich, a fraction that's really rich in leukocytes. After the apheresis is collected, generally the T cells are selected out of the apheresis and they're activated using some activation reagent. And following activation, the gene transfer step takes place. Generally, this is using like a viral vector construct or a viral vector to insert the construct into the T cell. And then following gene transfer, the cells are expanded so they can meet dose and then reinfused into the patient. So there's a lot of different process intermediates here and opportunities for us to sample and characterize. Generally, the places where we sample the most tend to be at the starting material step, so the leukapheresis material, after the selection unit operation, so purified T cells, and then towards the end of the process, we call this drug product. And all of these are common opportunities for us to sample. So I'll probably be focusing on most of these as material types to talk about today. Cool. So now that we're all experts on cell therapy, I'll talk really briefly about BMS's footprint in the space. I know there's a lot of folks from BMS here today, so shout out to the team. But BMS is a pretty big player in the industry. We're the only company to have 2 approved cell therapies, Breyanzi for NHL and other subtypes, and Abecma for multiple myeloma. But in addition to these, we have a lot of clinical stage assets as well. We have an asset called NexT that stands for non-expanded T cell product. And we also have CARs for the GPRC5D antigen and then a dual targeting asset that targets both BCMA and the 5D antigen. And in addition to this, we also have a pretty broad pipeline that's investigating kind of novel ways to manufacture CAR-Ts. We're working a lot on developing an allogeneic CAR-T product. And those of you in the cell therapy world know that sourcing viral vector is a huge source of just massive cost in terms of money and efficiency. So we're working on ways to insert the construct without using virus. In addition to this broad approach to new technologies, we're also partnering with other key players in the cell therapy landscape. We're partnering with Century to make an iPSC-derived CAR-NK asset, and Immatics to make an engineered gamma delta TCR asset. We're also working with GentiBio as well for CAR Tregs in the IBD space. So lots of stuff going on at BMS. And I'll take maybe a step back here and talk about like my particular role. Like BMS is a huge company. Cell therapy is like a really large organization. And you can imagine there's lots of sub-teams inside of cell therapy. And my particular department is product and analytical development. And we do everything from, characterize the process to developing assays for sterility and for release and for purity and things like that. But my specific sub-team is called product sciences. And that's a pretty generic name, but most of what we do is trying to identify attributes that are associated with endpoints of interest. So generally, this is like a clinical outcome. How many patients? Are there any attributes that are correlated with progression-free survival or something like that? So clinical outcomes are a big endpoint for us, as well as manufacturing outcomes. Can we identify any attributes that are correlated with manufacturing success? So that's what my team and I focus a lot on. And this is a lot of work to identify what attributes are correlated with outcomes of interest, since CAR-Ts are so complex, orders of magnitude more complex than the biologics class. CAR-Ts can do lots of different things. They can kill directly via perforin or granzyme, or through the Fas ligand axis. But not only can they kill, they can support the inflammatory process by secreting inflammatory cytokine or by recruiting other cellular mediators of inflammation. And not only can they accomplish this, but they can also grow and change. So characterizing the proliferative capacity and the capacity of CAR-Ts to differentiate into different subsets that might have different capabilities is also a big area of interest for us. So each of these bullet points represents an opportunity for us to characterize and us to understand our drug product, and that's no small undertaking, since there are so many different things to measure, so many aspects of CAR-T biology. Like I said, my team is interested in characterizing and correlating these attributes to endpoints of interest, and we think a lot about the best strategies to use for this characterization process. And those strategies have have undergone a lot of optimization and refining as Juno went from a startup to being acquired by BMS. So I'll just walk you through an example characterization strategy for kind of like a first generation characterization strategy. So say you're a scientist in the product sciences group, and you happen to know that a naive like memory phenotype is associated with enhanced CAR-T function. So what you might do is you might become interested in memory as an attribute, and you might start developing a small cytometry panel, maybe on a conventional cytometer, that incorporates a handful of markers that are associated with memory phenotype. And you might collect drug product from your clinical trial and assay, all of these lots of drug product on your memory panel. And at the very end, you can do your best to try to correlate if any of these memory attributes are associated with clinical outcomes. This is a great strategy. There's nothing wrong with this strategy, but we think we can continue to refine and optimize and translate this from like a linear characterization process to something that's a bit more fluid and dynamic. So this is an example of a second generation characterization strategy that my team and I are following now. So instead of focusing on just a single module, we're focusing on several different modules. So in addition to T-cell memory, thinking about T-cell activation and T-cell exhaustion and migratory potential and things like that, that are likely to be associated with CAR-T function. And then we might develop multiple spectral panels, big panels, like 20 to 30 color panels that encompass these above modules. We might then deploy these assays on pre-selected targeted cohorts of interest to save resources and time. And at each stage in the process, trying to think about how we can deepen our product understanding, elucidate method of action, and inform our product and process development. So this is kind of our second gen characterization strategy, and ways we're thinking of continuing to improve this strategy are thinking about the following items. So we think a lot about standardizing our panel design and standardizing our analysis strategy. Since we have so many different assets, we want to go from each asset developing its own assays to having shared common platform assays that are run across the cell therapy portfolio. We're thinking about designing panels with flexible channels, and I'll return to this topic later, but essentially the flex channel strategy allows you to leave empty channels in your panel that you can slot in different markers of interest as needed on the fly. Having multiple spectral panels, these big high dimensional panels really facilitate the use of high dimensional analysis tools like clustering and dimensionality reduction. We can use these to identify novel subsets and to guide our gating strategy. In parallel to phenotyping via spectral flow, we're thinking about deploying functional assays at the same time so that we can try to correlate phenotype with function. What do these phenotypes mean for the drug product's ability to kill? And last but not least, we're always thinking about using new models, new technologies, and new ways of thinking. And one of these is the infinity flow technique that I mentioned briefly earlier. But these paradigms also include thinking about chronic simulation models or scale down manufacturing process models to help interrogate attributes at different drug product states and different timelines. So the goal is, at each of these points and with each of these paradigms that I've described, to learn something new about your drug product. So that's a brief overview of characterization strategy, and I hope I've convinced you that we spend a lot of time thinking about this. Okay, I will move away from kind of theory to the fun part. So I'll talk about what spectral cytometry looks at BMS. This is a non-exhaustive list of all of the spectral panels that we've developed at BMS. Each of these are probably their hour-long talk about development life cycle. But I'm going to focus on these three today. These three panels are called T-Scope, which is kind of a hacky acronym, but we found that ChatGPT is really useful for giving your panels fun names. So T-Scope is a deep T-cell immunophenotyping panel. All PBMC is like a broader panel that's designed to characterize the crude leukapheresis. And as I said, I'll spend some time talking about infinity flow. Okay, So let's talk about what it takes to develop all these panels. We like to think that we've gotten pretty good at developing spectral panels with the breadth of assays that we've developed. So I'm going to hit a few highlights here and talk about titrations and staining optimization. And Laura really teed me up for these, so that's great to have so much overlap. And I'll also talk about reference control strategy and spend some time talking about flex channels. Cool. So I'm going to use the all-PBMC assay as a case study to talk about some of these highlights. And just as a bit of background, the all-PBMC assay was conceived out of a need to more broadly characterize starting material. You can imagine that BMS has a lot of starting material characterization assays. You can imagine that these are really important from the purity perspective. But some of these are the panels are great, but they're small and they're designed to assess purity. They're not necessarily geared towards exhaustively characterizing the crude leukapheresis. So we have this kind of mandate from on high to get more granular phenotyping of starting material. And this is of a special importance for BMS as we move from cancer indications to autoimmune indications. So developing this panel, what does this look like? Again, that's a whole talk on how best to develop your panel, but we leveraged a whole bunch of different resources to build this panel. We leveraged in-house subject matter experts, the existing impurities panels that we've developed, as well as the published OMIPs that Laura mentioned, and a really great paper from Alex Hubik at the Allen Institute for Immunology. I don't know if Alex or anyone from his team is here, but shout out to this paper. I think it's the 4th most read on Cytometry A. So it's a really great resource if you have the time. So what all PBMC looked like in its final state is this kind of amalgamation of one of the panels that Alex and his team developed. This is called the survey panel. It combines that. It combines in-house impurities panels and a few other markers that we anticipate to be quality attributes. And this is what all PBMC looks like. It's a pretty big panel. You can see that we have the ability to interrogate really deeply these major subsets. We can phenotype pretty exhaustively the different T cells, B cells, NK, and myeloid subtypes. And we threw a couple other markers here that the legal team told me that I can't talk about, but they are we anticipate that they reflect product quality attributes. So this is a big panel, right? This is 34 colors total. And in developing it, one of the first roadblocks that we encountered is how the hell do you titrate all of these antibodies? It's a lot of work. So titration is really important. I'm sure I don't need to tell this group that, but in case you need to hear it, titrations are really important. They yield the following benefits. You retain the correct staining profile. You minimize the compensation and spillover spread error, since spillover spread is a function of brightness. If you're doing longitudinal studies, titrating each lot of antibody gives you improved lot control. It's really important if you want to use the same assay over time. And oftentimes you don't need as much antibody as the manufacturer says, so you can use your reagent longer. And I've just given a quick example here of an example titration. As you move from low to high, you can see that the staining profile is really altered if you use too much or too little antibody. So if you need to see an example, this should be very clear that proper titration is important. And there's lots of different metrics to quantify, how good a titration is. I've given a few here, but generally these are things like signal to noise, which is just a simple ratio of percentile of positive over percentile negative, and things like staining index, which also incorporates the spread of the negative population. So lots of things, lots of tools for us to track. So we have a really wonderful young scientist in our group who is not only skilled with the wet lab, but is also really skilled at data science. And so she developed for us an automated titrations analysis tool. My team mostly uses OMIC. If you're not familiar, this is a Dotmatics software, and we found it indispensable for high dimensional cytometry. And we have a beta for FlowJo as well. And the app is version controlled and well documented. And the app is flexible in that, it allows you to set positive and negative thresholds for your antibody of interest, but then all you have to do is take your stats and feed them into this tool, and the tool will generate all these tables for you. And the tool generates these in shareable like doc format. So this is great for sharing with colleagues and putting into master reports and things like that. We found this really helpful. And this is just an example of what, this output looks like. You have, your antibody information on the right-hand side, and you get to see the plots of all the different titration metrics. So this really helps with getting everything in one place, making things a lot faster, and selecting the optimal titration. And this is an example of the tool interface. I wish I could say that this is like an open source tool, but I cannot, unfortunately. I just want to, plug the great work done by this scientist in our team, and show you how kind of easy to use and like slick it is, essentially. So pretty nice tool. It saved us, dozens of hours. So if you are a bioinformatics person, if you know a lot about data science, I would, this is like a great resource for the Flow Lab. It saved us easily dozens of hours of analysis. And if you're a PI in your lab, would strongly encourage, supporting your younger scientists who are interested in data science. Cool. So once we had all of our antibodies properly titrated, we can move into the optimization proper phase. And this is a long and iterative process. And I don't have time to talk about all the optimization experiments that we did, but I'll hit on maybe one really nice, really salient example. This panel has gamma delta TCR in it. Our default strategy to stain is to just stain everything at the same time in a single full stain cocktail. And that gave us the staining profile you see on the left. So you can see that our gamma deltas, you can maybe see them in the corner, but they're really poorly resolved, and we just wanted better resolution. So we read this, we consulted this great paper. This is OMEP 69. It's a great resource for those of you that are developing big panels. And this paper recommended that you use this technique called sequential staining, which is essentially you just add the gamma delta TCR, you give it a head start, you add it before the rest of the antibodies, and you come in and you stain with the rest of your full stain. And that single step alone dramatically improves the gamma delta TCR resolution, as you can see. So if you're interested in phenotyping gamma deltas and you're having trouble, would strongly recommend this strategy. Okay. After we felt happy with the performance of the staining for all of our colors, we moved into the reference control strategy. And I think the default for all of us for conventional cytometry is to use compensation beads like comp plus, ultra comp, things like that. That's just kind of the default, right? So that was our default when we were developing all PBMC as well, to use compensation particles for all of our colors. And you can see right away here, there are a lot of technical artifacts in the data. I'm just showing you an N by N matrix for some of the colors. And as Laura mentioned, some of these may be resolved by gating into your subset of interest, but this was a persistent and widespread enough problem that we thought we might revisit it. So when we switched to using cells for our unmixing controls, you can see that almost all of the issues resolved themselves. So if you're developing big spectral panels and you're having trouble with staining artifacts, would strongly recommend moving to cells as unmixing controls where applicable for your panel. And just an aside, data not shown here, but we had trouble when using cells for our live dead controls. We used to use ethanol fixed cells for our live dead controls, and found some weird artifacts that we were able to fix by moving to heat killed cells. So if you're also having this problem, this could be something for you and your team to think about. Great. So all of our antibodies are titrated at this point. We're happy with the staining, and we're happy with our reference control strategy. So what is next but to deploy this assay and get some real data? But another roadblock appeared, and I'm sure this sentiment will be familiar to many of you. But we spent a lot of time developing this assay, and halfway through development, we're kind of consulting some of the stakeholders that we're working with, and they say, "Oh, do you have marker XYZ on your panel?" And you say, "No, but that would have been great information to know before we started development." And now we're halfway through, and we we have to redevelop, or we have to, think about our strategy differently. So I'm sure this has happened to many of you before, and this is a problem that I hope this next topic will address. So kind of here's the problem statement, right? We now have the technology to make these really big panels like up to 50 colors have been successfully described in the lit. But it's impossible to predict every marker that you might be interested in. And if you're developing assays for other people, you can never predict kind of all the markers that your customers might be interested in. So, I mean, you can add or remove custom markers ad hoc to your panel, but this really weakens your ability to compare to past and future data. So not an ideal strategy. And every time somebody comes to you with a new marker, you could develop a new panel that incorporates that marker, but that's extraordinarily inefficient and very time consuming. So what is the budding cytometrist to do? And I'll say that this is not, we didn't develop this strategy, but I will show you how we have optimized it. The solution that we think to this problem is designate specific channels inside your panel as flex channels, where you can swap out any marker you want on the same fluorophore. So theoretically, this is a great idea, right? Totally solves the problem. But you have to be really careful with the strategy and characterize it really well. And choose colors that have minimal impact to the rest of your panel, and characterize the impact of using different markers and make sure that that doesn't affect the remainder of your reportables. The strategy makes sense? Okay, so I'll walk you through a case study. And the first time we encountered this problem was when we were developing this T-scope panel. So you'll recall that this is the deep T-cell immunophenotyping panel. Just for context, this is what T-scope looks like. We're assessing a lot of different elements of T-cell biology here. We're interested in, not only lineage, but also memory, activation, exhaustion, and trafficking. So again, I apologize at having to hide what some of these markers are. But anyways, this is what T-scope looks like. It's also a big panel, and this was our first case study for evaluating the feasibility of flex channels. So what a successful proof of concept looks like, right, is you try a bunch of different markers on these channels and make sure that they don't impact the remainder of your reportables. So that's more or less what we did here. Our preliminary flex channels that we identified were BV421 and PerCP E4710. So we purchased antigens of varying abundance for each of these colors. We picked like a high, so like CD45, for example, and a low, like PD1, for example, and an antigen somewhere in the middle. And we ordered these, on each of the colors. And so plexing this together, this gives us 9 cocktails. And each of these cocktails has the full panel in it, but it differs in which markers are on BV421 versus PerCP E4710. So we created these 9 cocktails. This is a really complicated cocktail to make, and we assayed them across a bunch of different material types that are of interest to us. And we evaluated the data. And so I'll show you what some of the data look like here. This is a single sample, but it's pretty representative. What we found was that flex marker choice doesn't impact most of the measurements. I'm showing you, arbitrary markers A, B, and C here. And you can see that the staining profile in terms of frequency, intensity, and distribution doesn't really change. Doesn't really matter what your flex cocktail is. So that's great, right? Up until you continue looking through the rest of your panel, and you find that the impact to markers 15, 21, and 27 really depend on what flex markers you're using. So you can see here that the blue and the purple and the gray histograms show a really dramatically different staining profile compared to the other histograms. So these correspond with cocktails 1, 4, and 7. And when you go back to this chart and you take a look, you find that they are all the PerCP E4710 high antigen. So there's some interaction going on here with PerCP, with like the PerCP color and the antigen that we chose. That's making this, this is not suitable for use as a flex marker, right? Because we're seeing such an aberrant staining profile. And I'm not showing the data here, but you'll have to take my word for it that we did not see this problem in BV421. BV421 showed the same staining profile regardless of which cocktail you used. So we were kind of, this is a first pass. We didn't expect perfection, but we were kind of, we really wanted to understand why this was and see if we could predict which markers would make good flex channels, or which colors would make good flex channels and which colors, would not. So after a lot of research and lit searching and thinking about this problem, we came across the stain index reduction, which Laura so nicely walked us through. You'll recall that stain index reduction is a feature between two colors, and it quantifies, as the name suggests, the reduction in stain index when two colors in a panel are used together. And so stain index reduction is a pairwise metric. It's like one color versus one color. But some colors, tend to be spreadier than others, right? So what we did is, or essentially what I did is just summed the stain index reduction for each color and arranged them in a table together to see if this was a useful metric. And sure enough, BV421, which worked fine, right, had a really low sum in stain index reduction in the context of this panel. And per-CP eFloor 710 was one of the biggest offenders in terms of stain index reduction. And you'll recall that stain index reduction is not only a function of the color, but also a function of the other colors in your panel, and also a function of the intensity of that measurement. And so it'll depend on what antigen you have it on, which makes sense, right? That we saw the biggest problem when we used an abundant antigen. And sure enough, this is essentially the same plot that, or a real similar plot to what Laura just showed. I used Cytec Cloud a lot in designing these panels and evaluating them, so can attest that it is a very useful tool. And I'm not sure if the font is maybe too small for you all to read, but the row that I have boxed is per-CP eFloor 710, and the three poorest interactions, the three highest stain index reduction values, correspond with markers 15, 21, and 27, which, if you'll recall, were the biggest problems that I had with the flex channels. So this is all to say that interactions with the highest SIR correspond with like the least suitability for use as flex channels. So we propose that SIR is a useful metric for evaluating whether or not this strategy will work or not. So, armed with this information, we were kind of encountering the same trouble with the all PBMC panel. So we went back to the drawing board, essentially repeated the same experiment with the all PBMC panel, except this time we chose colors intentionally for their lowest contribution to SIR. So here we chose BV480 and APC as our flex channels. And again, we chose, 3 arbitrary markers for BV480 and 3 arbitrary markers for APC, plexed them all together, and then tested them on a bunch of different material types. And the font is really small here, but essentially I'm showing you 2 plots colored by what marker is on each flex channel. And along the, on each column is a description of, the subset that we were trying to measure. So you hopefully the trend is clear that the frequency of subsets that we were attempting to measure doesn't vary across the different flex channels that we designed. And when we compute the coefficient of variance across all 9 of the cocktails for each subset, we find that the CV is really low for all of the major populations. And in fact, for all populations that are greater than 1% of total, the CV is less than 6.7%. So we're pretty satisfied with this. We think that this is a successful proof of concept of the flex channel paradigm, and we're now using these strategies in current implementations of these panels. So I hope I have done an adequate job of talking about flex channels. Here we propose the strategy to essentially leverage the same development effort to evaluate really any marker you want to measure without compromising your ability to make claims to past or future data. And we're continuing to characterize this, and development work is ongoing, but we're hopeful that this will help us find new markers of interest. And our major learning is that your choice of flex channel is the most critical, and colors with high predicted stain index reduction are not really recommended as flex markers. And your choice of marker and your properly titrating these antibodies will only help your flex strategy. Yeah, great. So we're really happy with flex channels. We think they can be pretty useful, and we hope that you consider it for your work as well as applicable. Great, so I'm going to change gears slightly and stop talking about panel design and panel development and touch briefly on high dimensional analysis. So moving from staining and flow to the analysis portion. And I imagine a lot of you are at least somewhat familiar with high dimensional analysis, but I will go through a quick primer for those of you that are not. So the two major camps of high dimensional data analysis are clustering and dimensionality reduction. And in a nutshell, clustering is just a way to group similar cells together for subsequent analysis. And after you have clustered and grouped these similar cells together, you can treat them as a unit for analysis, so analogous to a gate. You can look at, percent parent of cluster 17. Does that differ in my diseased samples from my healthy samples, and so on and so forth. And the big draw of clustering is that since it's unbiased and done by an algorithm, it can account for every parameter in your study in a way that a person just cannot. So very useful. And separate but complementary is dimensionality reduction. And this is a really great figure that I like to use when describing this. So DR is simply it's a visualization only technique. Sometimes people say dimensionality reduction analysis, when really what it is just a visualization strategy. And DR transforms data from the high dimensional space to generally 2D or 3D space, while trying to preserve the main properties of the original data set. So like a simple version of dimensionality reduction is PCA, principal components analysis, which many of you are familiar with. And yeah, you can see that in this mammoth figure, you can see it looks like a mammoth in 3D, but if you use different DR algorithms, you get plots that look like a mammoth or things that don't look like a mammoth at all. So it may be a bit of a segue into a cautionary tale about high dimensional analysis algorithms. Okay, now let's talk about them a little bit in context. So clustering has a variety of utilities. One of the most salient is that it can help you identify new populations of interest, right? Since you can only gate on what you've defined like a priori as important, whereas if you let the algorithm take over, it can find things that maybe you can't. So here I'm showing you two UMaps. The one on the right, I have manually annotated with like populations of interest. And the little like blue on the right, the little blue island is are the gamma delta TCR positive cells. And say I use that single marker to define gamma delta TCRs. I think these are these are all the same. These are all my gamma delta TCR positive cells, and they function in the same way. But if I use a clustering algorithm, the most common of which is flow-SOM, SOM is self-organizing maps, I find that this island is actually two distinct clusters. There's the purple and the kind of fuchsia colored one, and those are clusters 8 and 17, the ones that I've starred. So, if you go into your favorite analysis platform and you interrogate these more deeply and see how these clusters differ from one another, here I've overlaid histograms for cluster 8 and cluster 17 to try to see where they're different. And I found that marker X and marker Y are 45RA and 27 are kind of just putting these into discrete camps. And sure enough, when I go into my scatter plot, I see indeed that these two markers stratify the gamma delta TCR subpopulation well. So now, we're armed with knowledge that there's definitely a phenotypic difference in the gamma delta TCRs, and maybe they have a functional difference as well. And this kind of gives us knowledge for future analysis. So clustering is great, helps us find new populations, but you have to be careful with these algorithms. So your high dimensional analysis can really depend heavily on how you set up the algorithm. So the top plot is the same UMAP that I showed you before. You can see how I've kind of binned it into, this is maybe like 15 or so populations of interest. And depending on how I set up my flow-SOM algorithm, I can adjust these hyperparameters, and I can get extremely different clustering results, right? So if I over partition the data set, I get like almost 70 clusters here, which surely there are not 70 unique populations in my sample of interest. And if I under partition on the far right, it just like buckets them into like major islands, which is also not helpful for me. So I think takeaway from this slide is that high dimensional analysis can be helpful, but my recommendation is use several different algorithms and adjust them and do a lot of reading so you can make sure that what you're doing is appropriate for your data set. Cool. High dimensional analysis can be really its own talk, but I think that's enough of a salient example for today. And I'll end with this analytical technology spotlight on infinity flow, which was developed here at Fred Hutch by Mark Headley and his lab. I don't know if anybody from his lab are here, but they wrote this great paper a few years ago with Mark and Etienne Becht. They wrote this paper called it's about infinity flow. So what is infinity flow? I have to walk you through the bench work before we talk about how to use it. So this is a somewhat complicated concept, so please come talk to me afterwards if this is confusing. But essentially what you do to accomplish infinity flow is you have a tube with your sample in it. You stain your sample with your full stain cocktail, whatever full stain cocktail you think is most applicable. Then you take your bulk sample and you can split it across every well of these special plates that are called massively parallel cytometry plates. And BioLegend has a great one called Legend Screen, and they come as like a set of 4 96 well plates. And in each well is a different lyophilized antibody, but all conjugated to the same fluorophore. This makes sense? So you stain your bulk sample so it all has the same colors. You split it across all these plates so that each well, it's the same sample. It has the same full stain cocktail, but it differs in which infinity marker is in each well. So then you can continue your assay as normal and acquire using your favorite cytometer. After you are done acquiring, you can feed your data into this algorithm. And essentially what the algorithm will try to do is it will try to predict the intensity of each of the infinity markers using the full stain intensities and the scatter profiles as input. So it will try to build this model based on your backbone cocktail. What does the algorithm think is the expression of each infinity antibody? And so it iterates this across every well in your data set, and then it concatenates it all together. And then what you get at the end is, your FCS files, but instead of just, the 20 or 30 full stain colors you now have 400 different parameters that you can look at. And granted, these are predicted values, but the real power here is the ability to look at co-expression data of 2 markers that you have not assayed together, but you can get the predicted co-expression data of the 2 of them. I am sure you all can imagine that this is an incredibly powerful tool. And I'll talk briefly about a couple ways that we've implemented this in practice. So one of the first things that we tried to do is we wanted to profile the CAR-T activation process. And so essentially what I did was a reverse time course and then harvested the material and assayed it on infinity flow and tried to look for which markers or which combinations of markers are most upregulated post-stimulation or downregulated post-stimulation. And we found a bunch of hits. What I'm showing you here is just the CD8 CAR-T fraction, but what I'm plotting is like a percent enrichment or percent depletion in each marker, over time. And I've colored these by whether or not they're canonical markers of activation. Are they associated in the lit with activation or differentiation? And you can see that the red ones are, we all know that PD1, LAG3, CTLA4, and CD69 are associated with T-cell activation or exhaustion in some way. But this reveals a lot of new markers. I personally haven't spent a lot of time thinking about CD120B or GIT receptor or PVR or a lot of these other markers. So we think that this exercise has helped us identify potentially new activation markers of interest. And we need to do follow-up work to see whether or not they're important. But this has been a really informative exercise for us. So we did our best to profile like novel activation markers. which is kind of a generic T-cell CAR T situation. But we've also used it in the context of like program and product knowledge, which for those of you in industry know is really important for your teamwork and the success of the program. So again, this is an area where frustratingly I have to be really vague, so I apologize. But we observed this interesting composition of cells in some samples. I cannot say what the samples are or how they're interesting, but essentially we see this atypical profile that's dotted in purple, and you can contrast it to this typical profile. And we didn't expect this. This isn't necessarily alarming or a safety event, but this is like something we hadn't seen before. And we had done a lot of like prior work to show that these aren't technical artifacts. This isn't a detection antibody problem. It's not an internalization issue. And the cells, these aren't dead or dying cells. So what are they and how are they different and are they important? So we ran infinity flow, on these samples. And from the combinatorial and also singleplex data, we were able to generate, this plot of differential expression. So, we know all of the markers essentially in this 400 marker space, which are enriched and which are depleted in the typical versus atypical samples. So already that's like a super rich data set. We could spend a lot of time analyzing it. But not only can you get this list of differential expression, you can feed this combinatorial data into high-dimensional algorithms like FlowSum and look at the clustering data. So how do these markers co-express with one another? Can you glean anything from the clustering data? And then in addition to clustering, especially in like the sequencing world, there are these pathway and network analysis tools, which I'm not an expert on, but some of you in the room might be. What these types of algorithms look for is like enrichment in pathways, and this helps you put biological context to the data that you generate. So kind of like gold star one is QIAGEN's Ingenuity pathway analysis platform. And by plugging our data into this platform, we can see what pathways, are enriched in this data set, which pathways are depleted. So this, gets us one step further towards product understanding and, intervening or leveraging the data in this way. So Infinity Flow in tandem with these high dimensional, platforms or technologies really allow us to turbocharge our analysis. So I think I'm running up on time, but in conclusion, I didn't mention this specifically, but Infinity Flow is pretty inexpensive to run. You don't need anything more special than a cytometer and to buy these massively parallel plates. It generates a ton of data. The cost performance or like the bang for your buck ratio is pretty good. And we've applied it here to both CAR T and PBMC products. And these specific experiments, have revealed potentially novel activation markers and helped, de-risk or inform, clinical assets. And, this is obvious, but we describe a successful utilization of Infinity Flow, and we intend to keep using it for the future. Cool. So that brings me to the end of the talk for today. Hopefully, I gave you a good overview of the different applications for spectral flow at BMS. And just a few quick parting thoughts. Spectral cytometry is important. It has the potential to really allow us to learn new things about whatever we're studying and offer a lot of insights into product quality, for those concerned about that in industry. And you have a ton of resources at your disposal, right? Like we've got a ton of help from SciTech, a lot of help from BioLegend, a lot of help from Mark Headley and his team. And that's not even taking into account the lit that comes out of the Seattle area. So we have some phenomenal flow cytometry focused labs and like cores here in Seattle. So I would highly encourage you, read your OMIPs, pay attention to what's coming out of these labs, and read Cytometry Part A. And my last piece of advice is, pay attention to some of these novel techniques. You might be able to find something new that's applicable for you and your lab. And that's all that I have. So thanks again to the organizers for having me, and thank you all for listening. And happy to take questions. Yeah, go ahead. We're not using auto gating yet. We still like the ability to set manual gates. It also allows us to discriminate between different, like if we multiplex materials together for our titrations, gives us flexibility to kind of unpack those. So we haven't used AutoGator yet. I was under the impression that AutoGator is still in beta for OMIC. Okay. Cool. Yeah, excited to use it when it comes out. Yeah, that's right. Well, we go in and assign our gates and make any edits if we need to. We have all these templates that are set up to help us save time. And then we're pulling the export stats from OMIC manually, which that could probably be automated as well, and feed it into this tool. Yeah. Oh, no, yeah, I understand what you're saying. Yeah, since SAR is a function of like fluorescent intensity, it'll depend on the marker you use and the color. So when we used the mid and low antigen densities, there was no observable impact. It was only until we crossed that like high threshold that we started to see problems. So that could be another, thing to keep in mind. Per-CP eFluor 710 functions fine as a flex marker as long as the intensity or abundance of that antigen is low, or like medium or low, no brighter than the mid one that we used in this experiment. Yeah. Oh, yeah.
Was that basically solving the mystery behind the differentiation of the two cluster data for that one population?
Okay, I think I see what you're asking. Yeah. I think you're if I understand your question properly, we took the those two clustering data, like those two clusters that split the gamma delta TCRs, and really what I did is I just took the two clusters, overlaid them with one another, and permuted them across all the colors in my panel, and just manually picked out what to me looked like the most different. And then that's kind of how I found these two, that seemed to have like the most difference between the two clusters. And then if you go back to your scatter plot and actually interrogate them via scatter plot, it really nicely partitions them into 4 quadrants. Does that answer your question? Okay. Yes, yeah, there's CD3 in that cocktail. That makes sense, yeah. And in that case, we could probably, if we have alpha, beta, and gamma delta, we could probably omit CD3 and get perfectly good data. That's a good point. Okay, if there are no more questions, if you have questions, come find me afterwards. But thanks for your attention, and thanks to the organizers.
Okay, we're going to get started again. We have our next speaker ready to come up. And after that, after Ryan is done, we have like 10 minutes to grab, or maybe there'll be some time. The next talk is starting after you guys grab your lunches. So the lunches will be, they're getting those set up now. So after Ryan's talk, there's about 20 minutes-ish for you guys to get your lunches, and then Ravi's talk will start at 12:10. Okay, so with no further ado, I'm going to introduce Ryan Brinkman. He's from University of British Columbia and Dotmatics, and he is going to give us a very interesting talk. I'm really excited about this one.
Thanks. Thanks to the organizers for having me here and for the awesome swag, EVE Online. I'm going to be talking about that today. I'm also really excited to be here today. because you're going to see stuff that I just got on Monday for the two things I'm talking about. This is like fresh off the press. I haven't shown this to anybody else, and nobody else. We haven't had a lab meeting about this stuff yet. So it's really fresh. It might not be like this next week. This is a whole work in progress thing that we got going on. It's been going on for about 3 years. So I'm still a professor, so I still have to make a disclosure. I work at Dotmatics, so I have a leadership position. They would love to sell you OMIC and FCS Express. Everything I'm talking about today is already available for free or will be freely available. I'll have the best job in the world because at Dotmatics, they are allowing me to do stuff as open science. And everything I'm talking about today is open and will be open. Except I push a button at one point. You have to pay for to have the button push. So I really have the coolest job in the world because I feel like I'm an action hero. In the really cool movies, the protagonist wakes up and is like, who am I and where am I? And he's got a gun in his hand. And that's how I feel like I wake up every morning. Maybe not with a gun in my hand. Maybe I want to shoot myself in the face. Because the kind of questions I'm answering are in cell populations, we're defining what things are. And to define things means two things. Where am I? And then what am I? The labels you put on these things. And in flow cytometry, the where am I is gating. And gating sucks. It sucks for lots of reasons. And I'm sorry that it does, because you're living that life. It sucks because you spend a lot of time doing it. It's time consuming, it's subjective. You and the person next to you, you'll get the same population, and somebody wrote a paper that says you will get a 30% difference on that same cell population. That's a large difference, and that's not good for science. And then because these panels, like you've heard of, are so complex, 50 dimensional panels, you don't have time to go through all that data to find the cell populations you want to identify. And that's sad. And it's probably not really why you chose to do science. I mean, when you're a kid and you're flicking through TikTok and you saw somebody gating, it's like, I want to do that. No, right? Well, you didn't have TikTok, but it's also probably not why you're doing it. And I'm interested in gating because I'm really interested in saving people's lives. As a cancer survivor, CAR T therapies are a wonderful thing. And I think it's gonna be a while before we get it. Unsupervised analysis is great, don't get me wrong. We've developed some algorithms. They solve a very specific problem. The problem I'm interested in is I have a clinical sample and I want to know that T cell count because I want to develop a new drug. And it's going to be a while before the unsupervised methods do that. And there's been some great ones out there, like FlowSom. But the reasons why these are never going to be as good as manual gating is because they have to make assumptions, right? They have to fit a model to your data. And the size, shape, and distribution of these populations, high dimensional spaces, all very different. And so you just can't get that really good performance when you're trying to get a percentage count that you can get out of something like FlowSom. And the way where we can measure this performance is by using something called the F1 measure. And the way that works is sort of the harmonized, I have to take water, or I'm just going to talk really, really fast. It's the harmonized mean of sensitivity and specificity for getting every single dot in the same group between manual analysis and automated analysis. And we get an F1 score, which is 0 to 1. And you can think of that as a percentage score. And as far as I know, that's FlowSom is a really, really cool tool. And the average performance it gets versus manual analysis is like about 76%. I'd be happy. Okay, my kids could better 76%, but that's not a bad score, right? But sometimes it does bad. Sometimes these algorithms do bad, right? Because they have to fit this model to some populations are hard. And so, sometimes you can, this is again from the Robert Robinson paper, 56%, it's not a great score. Great for doing discovery. Like here's a whole pile of data, what's changed versus treated versus untreated, great tool for that. But when in a clinical setting or in trying to get counts, it's not going to be great. So we came up with an algorithm in my lab called flow density. It's awesome. It's really good. And the reasons why it's really good is because it solves the problem that you're trying to do when you're trying to do manual gating, which is I want to put a box around these dots. And the reason why it works really good is because we tell it to do what you're doing, but in math sense. So for example, here's 5 different samples from the same study. And in the middle, you can see that there's 2 populations trying to gate. And what we tell the algorithm, if there's 2 populations trying to gate, put it in the middle of those 2 populations based on the density. And then, maybe there's only one, on the left-hand side, there's one peak with a smear. So for your gating panel, we're going to look at, see, is there a smear? And then we'll go this way and gate that way. So there's a whole bunch of decision trees that we built into the algorithm for a specific panel to guide it along. As each new FCS file gets delivered, the algorithm goes, oh, what do we got here? Oh, it kind of looks like this. And if it looks like this, I'll do this. And these rules are things that we can articulate to you. And we work with you to parameterize that algorithm. And that's why it works so well, because we tell it to do what you want it to do. That's the downside, because it has to be trained for every specific panel to do what you want to do. Does it work really well? Well, yeah. So we can get straight lines between manual analysis and automated analysis. That's shown here. We get perfectly straight lines. If you have two different people gate the same cell population, and then we have our flow density algorithm down here, somewhere, mouse on the bottom left, will be within the variation of any two humans are from each other, the same way you do as any two humans are going to be as different from each other as flow density algorithm is going to be different from any other human. And that's great. I can publish papers on this because I'm still an academic. But really, what really excites me is the ability to apply this to therapies. And so I don't have to talk about how cool it works because Kite will do it for me. And so these are slides that Kite did out talking about how they used our flow density algorithm for their CAR T therapies. And the nice things that they said is it gates about 1,000 files in about 7 to 8 seconds per file. The really exciting for me, I get shivers every time I say this, the analysts that set up the file, the whole format and the analysis and the gating hierarchy, going through 1,000 files, there's some files that they weren't willing to make the call on. Like we don't really trust how we're gating this sample. And then when they used the flow density algorithm, they looked at the results and they said, all right, let's give the patient the therapy because we understand how it's gating. We see where the gates are and we trust that. So I'm saving lives at scale. And they're happy because they're saving lots of money. But what they had to do in order to, everyone's doing manual gating now, and they wanted to transition to the flow density algorithm. So they have to show how they can bridge between manual analysis and automated analysis, and they couldn't. It didn't work. Like, it's really different. But then when they looked at all the different cases, and there were many, many, many cases where they weren't matching up, in every single case, they trusted the automated analysis. And easy things are easy to gate, but when things get hard, people had a really challenging time. And when things get hard, in every single case, they trusted the automated analysis. So that saving people's lives. So why is it so good? Yeah, I missed my cool James Bond slide. Damn it. I had a really cool James Bond slide. So the whole thing about the James Bond thing is, so the reason why this is different out of unsupervised analysis is different than the supervised analysis that we do in flow density. So all these unsupervised algorithms have to fit your data. And the way you can think about it is that if you buy a suit off the rack, so like, okay, my son got his at Banana Republic, but say you went to Walmart to buy your suit, right? It's not going to fit you very well. But and that's sort of like the unsupervised algorithms, it kind of fits your shape. The reason why this supervised approach works so well is because we stitch fit the algorithm to your data. It's a really good analogy. I'm sorry I missed it. So I wanted to say this going back in time. And it's bespoke fit, like the cloth is cut exactly to fit, and we tailor it really, really well. And that's why we have the performance it gets, and those are some examples of that. Tricky to type very slowly. Okay, and so this is an example of this. So for this one key gate that Kite had in the CAR T therapy, there's 23 different if then else statements that we walk through to try and gate that one sample. And there's lots of information we can get about the density of a population in order to do that. And because of that, that's why they have the performance of get. So, when things are easy on that top left, these are really samples to gate. You can just gate that by eye. You don't have to do anything to get that right. But when these people have failed first, second, and third line therapies, they're dying. It's like blah in their cells, right? But we can still gate those effectively. And that's why it worked. So they're very happy. It's quality by design. We don't have a PhD do all the gating. We just have some little master student going beep, 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 and look at it. That saves them money. They can scale their operation globally across all their centers. They can have a standardized data platform that we might want to sell you called OMIC, and you can just, everyone goes beep, 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 and it all works everywhere the same way. Great for them, sucks to be me, because I want to do lots of action hero movies. But I have to do every action hero movie one at a time. I can only help one client at a time. And there's not a lot of people who can parameterize this algorithm. We have like 3 people in our group who have this expert. So we don't really advertise it, and I'm happy to work with Big Pharma. But I can't solve I can't solve all your problems because it's really, really hard to parameterize this algorithm. But we give it away for free. So if you want to do it, There's stuff online that will tell you how to do it. Google it. There's some tutorials we give. It's like the pizza delivery service. Everyone knows how to make pizza. Nobody wants to make pizza. I make pizza at home, but it's not. But you'd rather just call somebody, right? So how are we going to solve this problem other than deliver you pizza? AI, right? If you've been around anywhere for the last year, couple of years, AI, ML is going to solve all the problems, right? And so so the question I had is how can I use AI, big data, because I've been a big data guy. The only reason I got my PhD is I wrote, everybody in the world says, will you give me your data? You can do that when you're a grad student. I'm still doing it. And they gave me all their data, and I could solve really big problems because I had a lot more data, and then we'd figure out how to diagnose Huntington's disease. And the only reason why I could do that is because I had 100 times more data than anybody else in the world, right? Big data solves really cool problems. That's my hypothesis. So all I had to do was go out there and get to do the gating problem. I just need to go get lots and lots and lots of different gates that you guys all share, right? You guys give out all your gates. No, you don't do that, right? You guys don't share how you gate your data, at least not at scale. Maybe in a certain lab, you can do, there's laboratory developed tests. Like, I don't know why you're talking that way. Here's my panel. And here's how I gate it. And I'm going to do a laboratory developed test. And people publish papers on this all the time, right? Here's my AI algorithm for this particular study on this particular panel. And look, we got really, really good performance. And you're like, good job. But can I use that? Not unless I'm using your panel. Nobody wants to use anybody else's panel. You guys all like your own panels, right? Maybe you'll look at an omic and that's kind of cool, but we'll change it a bit, right? So I don't have the big amount of data to train a gating algorithm. So let's solve that problem. In a video game. So this is Andrea. He's a former president of ISAC, and we put him in a game called EVE Online. So EVE Online is a massively multiplayer online role-playing game where people fly around in spaceships and shoot each other. It's not like the video games that I play where I have like 15-year-olds and shoot me and then teabag me and go meh, meh, meh. If you laughed at that, sick people. So when I went to the EVE Online, they had CCP had the annual conference. The average age of the gamers is about 30 years old. And I was in the conference room and they talked, and we put Python into EVE Online, and the whole crowd went wild. It's really bizarre for a video game conference. And they had a whole panel of NASA scientists who are EVE Online players. These are people who, they're rocket scientists. There's a really cool macroeconomic things going on in EVE Online. They have big giant spaceship battles with thousands of people that burn real money. Like the most expensive space battle ever was like hundreds and hundreds of thousands of dollars were blown up. It's not your typical video game, not your typical audience. I'm just trying to sell you that these aren't 2-year-old kids that are doing flow cytometry gating, but it's still a bit of a sell. So we taught them a bit about flow cytometry, but we can't teach them the awesome knowledge that you and the person next to you has about biology. We can't teach them how to go through gating hierarchies, and this is what to gate, and this is how to gate them, because we're looking at, I don't know, some disease, and this is how you should gate that disease, right? We can't teach them that. But the hypothesis I had is, if we just break down the problem to its really simple component, here's a plot, gate this plot. And this is how we do discovery in my lab and at Dotmatics, where you have something called flow type. It just basically give us all the bivariate plots. We'll go down all the different pathways and put them all together and see treated versus untreated. And this is your magic population. It's sort of our way to do discovery without doing the unsupervised analysis. So let's break down the problem as simple as it is. And I thought, if we just give the simple problem to these people, maybe they can gate. And this all started at the beginning of COVID. EVE Online wanted to do something to save the world, and somebody gave me a call, said, is there anything you can do with COVID and flow cytometry? And this is right when Italy was having the whole outbreak, and there was no flow cytometry data, but like, it's going to happen one day, so sure, I want to see your data. And so within about 6 weeks, they built I was going to say FlowJo, I can't say that. They want a FlowJo, like, of course I can say it, a FlowJo-like interface within EVE Online. So when they're not going pew, 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 they can tab out and gate flow cytometry data. And it looks like this. And people do this, right? For fun. And these and they can draw boxes around the dots. And these aren't this actually, it's not, these aren't the cells you're looking for. Those aren't the cells they actually gated. We gamified it and we gave them like, you're doing really good, pat them on the head, because we have some examples where we know what the results are and what they should get. And so we check them every once in a while so that they don't have like bots doing things or they're paying attention, just like click, click, click. Turns out people really like the gate. You know, when I gave this talk in Reykjavik, they're like, I find this soothing. I don't know if you guys get that. She's like, oh, I'm having so much fun gating. It was like this heartwarming. This father had his little kid on YouTube, and he was sitting on his lap, and this kid's gating flow cytometry data. This is what we're using to train our AI algorithm. Not really. I'll get to that in a second. So this has been running since the start of COVID. This is a couple days old now. I did this slide on Friday. So 1,355 days, 24-hour days. People around right now, people are drawing gates for me. So far, I'll just round that up. 800,000 people have gated flow cytometry data. 463 million bivariate plots have been gated. 87 different markers, 1,200 different marker pairs. I got all the data, and I was really smart once in my life. It's like because I don't have my own wet lab, and I thought I don't have any data, and I'm trying to do bioinformatics. Let's build a repository where I can trick people to give me all the data. Thanks. And so I got all the data from Flow Repository, I got all the data from Import, I got all the data from Cytobank, and I threw that against the gamers. And we're going to give all these results away so anybody can do whatever they want with them. We're in the process of putting together the publication. We're just trying to figure out how to do all this stuff. It's a work in progress. But we were doing this for about 6 months, and I realized this isn't really working because I got all this data and I'm throwing it at the gamers, but showing them plots of like CD3 versus, I don't know, I'm not very good at biology, CD3 versus CD4, you get these two plots, two populations. These are really easy to gate, and we're just throwing all the data. That's not really good to train an AI algorithm, right? There's lots of variation that we want to train people on. So we just can't give them all the data. We want to show them heterogeneous, really lots of syllables in that word, heterogeneous data. So we developed an algorithm called FlowSim that we published last year that give us all the data in the world, find us all the different bivariate plots of that. And that's really more useful to train an AI algorithm because heterogeneity is important, right? If you train your ChatGPT model on a bunch of TikTokers looking at Kim Kardashian, it's not going to be very good for solving any other problem except Kim Kardashian's problems. I don't know what your problems are. My problems is gating, right? So I need a lot of variety. So that was a cool thing. So we're training on lots of heterogeneous data. Black screen. Everybody wants to do AI and ML because it's fun and exciting. 80% of it is data wrangling, right? There was a lot of shit data, and we spent most of the effort for us to develop the AI algorithm was out of those 463 million results we get through is how to get rid of all that shit, right? Because there are some in-game, spoiler alert, it's not really spoiler alert because I already told you the story. There's some in-game rewards that people get to in EVE Online to incentivize them to do this thing. Some people are bad people. Some people enjoy gating, want to do the good job. So how do we figure out, we spend a lot of time trying to figure out who's giving us the really good results that we can use to train the AI machine learning algorithm. And that was a really interesting problem. Did I go too far? Did I skip a slide? Yes, I did. This is what we consider good results. You nodded, thanks. Everyone nodded, right? Yay, right? We automatically filter out good results. And the way we do it is, and this took us a long time to figure out, what a good result is. So people can do 1, 2, 3, 4, 5, 6, 500 gates. Some people just stop around one bivariate plot. What's correct? It turns out what I believe is good to train a machine learning model is we give each plot to about 500 accounts. Lots of people look at it. And what the question we say is, okay, look at how many polygons, how many people have drawn around that from 1 to n. And then look how well the correlation is between gamers who've done the same amount of polygons. And we have, we go to the highest number of, over here, there's lots of polygons. Maybe it's 1, 2, 3, 4, 5, 6, 7, 8. And at some number, we have at least 5 people agreeing between each other. That's that result. And if you go, so this is 8 polygons. And if we go to 9, people just aren't agreeing with a very good F1 score. We go like 0.85. So some way we get agreement between lots of different people at a very high polygon count. And that's what we're using to train the AI algorithm. And then what we do is we take all those people who have the same number of polygon count. And then we don't know what's the best model for training the AI. And this is what we're working on right now. Is do we just want that one result, which is a consensus of those 5 people? Because you can see that guy on the left, or girl or person or thing, cats, it's on the internet, everybody's a dog. They've drawn the gates kind of going long to the left, and they still get, does it really matter? Because, and this is a question maybe we can talk about over, is there beers here? Yeah. Yay! This is maybe a thing we can talk over beers later, is it doesn't matter to you that the gates are going long, because I believe that all you care is how many events are inside that gate. So it really doesn't matter how big the gates are. We're gonna have, this is one we're talking about internally. Do we want bigger gates or larger gates? Are you gonna be sad if the gates are a bit larger, or do you want to shrunk around your data? And so open question, but what we do is we just make a consensus out of all the best results. And we're either gonna train the AI algorithm on that one consensus, or just throw those 4 or 5 different examples. And we're not sure, different AI approaches, some want warrant data, maybe we just want the consensus. Open problem, we're working on it. We'll get the right answer very soon. So how well does it work? So how well does it work? So a couple of years ago, there was a Cyto conference. Really good. I'm going to plug Cyto. I'm a huge fan of ISAC. Everything awesome in my life has come about because of ISAC and by extension Cyto, at least academically. Other good things in life besides science. Most of them are science. And so what we did is we went to Cyto and we put EVE Online in front of scientists, people who know flow cytometry. There's Sophie Van Gessen who developed the flow cytometry algorithm playing EVE Online at Cyto. And then we compared all the Cyto people against EVE Online people. And I wouldn't be showing this if it wasn't a really cool slide, because spoiler alert, the gamers did better. And not just all the gamers, it's the expert gamers, where we pick the best gamers who are doing the best thing. And the truth that we did is our flow density algorithm, because flow density, and this is on data that we published on. We know flow density does a really good job. You drank that Kool-Aid. You all want to use flow density now and not do manual gating. And so the blue is the people at Cyto, and the red is the gamers, and it's statistically better. Statistically. Now this is a bit of a game. It's a game within a game within a game. It's like, what's that movie, Inception? We're like, because at Cyto, we had to make it a game where how many gates can you do in a minute and a half? Because otherwise, you guys love gating. You'd be like this. And then you'd be in front of the computers all day. And I wanted lots of results, so I'd have it. And I wanted us to do better. And the gamers have as long as they want, but still, right? They were doing a good job. But it's a bit of a joke, and it got some laughs, and that's what's important. But in the end, we have data like this that we're training the machine learning algorithm on, where there's at least 5 gamers with an F1 score greater than 0.5 at the highest number of gates. Those look pretty good. So we have lots of that data. Now we've done this twice. We have 2 different implementations going on. I live in 2 different worlds, academia and industry. And so we have 2 different implementations going on, completely different methods. The kind of results look like this. Now, is this a good result? No, it's one of our worst performing results. Why is this a bad result? Because you guys can't make up your mind on what truth is. Right? Truth, I don't know, this is probably some quote from a movie. Truth is whatever. That's probably a good thing. Truth is whatever. And where it's whatever is in smears are hard, right? So because we don't know what to do with them. Maybe you do in a biological context, but in the bottom, we have 2 different things. There's 2 different things going on this slide with smears. On the bottom in that smear, truth is, and this is according to the expert, why am I doing this with my hands? The expert has decided in that smear, and red is showing what should have happened, and the black is showing what did happen. Is that the right? Or is it the other way? No, right? On the right hand, your right is what the ML gating, and on the left is what the gold standard is. And what I've done is shown you what the difference is. So in the gold standard on the left, the scientist, like the human, we're all human. The human has decided that in that smear, half of it goes to the left, half of it goes to the right. On the left-hand column, in that smear, the human decided we want that smear in its own little population. The problem is there's no information in the data that says how to make that choice. And at least how we train the gamers. The gamers have learned that at the highest end, that we want to split off all these things into individual segments. You all nodded when you saw that plot of what the gamers are doing. All these features are in its own little box. So you can see here on the bottom, that stuff in the middle, they put that in its own little box. And they never split, or usually don't split that in half. That's because we're going to the highest number of polygon counts. Because we're going to the highest number of polygon counts. And this is by my own design of what I wanted. Because we'll get that to in a second, is if we don't learn how to split these things off, then it never will work, right? We'll never be able to split them off. So we have to train the algorithm to be able to be splitting. We can always join things together if they're split off. But if we don't know that they can be splittable, we can't split them in the future. So we have all these things split off. And so we'll get that in a second, the secret sauce about splitting. So that's the problem. Truth changes. Biology changes our perception of the data. Now, interestingly enough, this population was also the one that we had a lot of trouble with when we developed our flow density algorithm and published a paper on it. We compared 2 humans, which are the circles, with the flow density algorithm, and that the humans couldn't agree on how to gate this. Even though they're in the same lab, in the same study, trying to gate the same population, you're doing the same protocol, they're not agreeing with each other. So we don't have a hope in hell of having an automated analysis match that, because truth is all over the place. Now, we can be really, really good at gating. We just can't be, like I've told this story before, we can do really good job at gating. It's not necessarily going to match what one single human does. So that's a problem. So how can we solve that problem? The idea is the same way that ChatGPT and all these other algorithms work, is it gives you an answer, and then you give it a bit more information to drive that to the truth that you want to hear. My God, I'm gonna get so much shit saying this right now. My daughter is addicted. My daughter is addicted to ChatGPT, and it's reinforcing her ideas that we were bad parents. My parents are really bad. What should I do? We're not bad. You're giving it information that is guiding your perception of reality. So well, the whole idea is you give us one template that is your view of the way the world works, and then we take truth and then we push it towards your reality. So you give us a few templates on how you want to gate, and we're going to layer that on top of a general purpose model. Now, what's important here is how different the data that you're giving it is from your idea of truth. So if how you've gated it and that file that you gated on looks a lot different than that file that's coming up into the machine, it's going to do a really poor job on that. So the template target distance, we can measure distance with the flow SOM algorithm. If the distance is really really high between those two, so at the top the distance is short between the train template and the one you're trying to gate, versus, so we're gonna have a better chance of getting the right answer. So it looks pretty good. Things go to shit at the bottom because the template that you've gated on doesn't match the target that you're trying to gate. So even though we have the general purpose model, we layer on your idea of truth, fucked it up because it looks different. So probably one template isn't going to be enough. And one reason why we haven't released the algorithm yet is we're trying to figure out how many templates you need under what kind of conditions. We're trying to put around some parameters around that. It's 1 can do so good, 10's better, but nobody wants to do 100 because I'll just do it all by hand. We're trying to understand that. And so what we're working on right now, this is the slide. So this is like fresh meat. What we're working on right now is trying to understand in a really deep way on where things are failing. And so when things are easy, we do great. And so I took all the data that we had in the world, is how can we categorize a bivariate plot in its hardness? And we came up with some metrics that actually correlated in the end really well with our performance. So that's a good win. And what we found is things like if there's a lot of events and there's not a lot of variability in your samples and how smeary they are, it's going to be a pretty good idea. Just a general purpose model, that's what we got. When things are easy, anything's going to do a good job. Moderate things to gate is when there's maybe a high variability sample to sample and how things look, or if there's a fewer number of events and it all goes to crap on plasma cells, right? There is no truth that little blue dot below that black line is inner. Like, why did you decide that that's truth? And the problem with the F1 score is we count on how many events we get right inside the box. And when there's only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 dots inside the box, and then you put 11 in, you're like 10% off. And if you miss 2 dots, you're 20% off, right? There's no way we're ever going to get a good score on this kind of stuff. It's no way you're going to get a good score. So I don't think it's fair to evaluate our algorithm on stuff like that. Maybe. I don't know, but that's where I'm going. It's not our fault that we suck. It's a hard problem, right? Now, how bad do we suck? Not very bad. So on easy stuff, we do great. So this is using the general purpose model is on the left-hand side. And if you give us 10 random templates, just get us 10 files. And we can probably tell you what 10 files to gate. If you've collected a whole bunch of data, don't just peace out, just wait. Collect a whole bunch of data, and then we'll give it to our algorithm. And we can say, here's some files that look very different, because we have the flow sim algorithm that'll tell you these files look very different. Gate these 10, and then we'll use those 10 templates to gate everything else that you collected. So so you can see that we get about a 10% performance boost from having a few templates. And this is on thousands of bivariates. So we have 2 axes here, the number of samples. And on the bottom is the mean number of cell counts. So these are tens of thousands of events that we're trying to gate. And these are specific bivariates that we're trying to gate on. Then we go to the hard case. We're still doing pretty good. The F1 scores, we still get about a 70 to 87. It's like 20%, 90% boost in our F1 scores by having a few randomly chosen templates. We're doing pretty good. And then we get to the hard stuff. Hard's hard, right? You saw what we're trying to gate. This is stuff like look at the cell, there is no cells there. Well, there's maybe a couple, right? Very, very few number of cells. We can still get not bad, and it's probably going to be okay. And you can still adjust things how you want, and maybe you don't have to gate anything anymore, but we can do most of the work for you, and we can point you where things get really hard, because we can say, we can do things to help you. Say, we can look at the distribution of gates, and you're really happy. Maybe look at these samples down here. Maybe you just want to regate those first. Maybe you don't have to gate all your samples. Maybe only the ones where things are really different from the distribution of everything else. Those might be biologically motivated. Those may be technically motivated, but they're different. Difference is interesting to us because we're scientists. And then we can get as low as just the algorithm is called Flow Magic, the academic version. Just one random template. We still do pretty good on easy, medium, or hard. These are a few examples. This is, I got these slides yesterday. 99%, 88%, 90% F1 scores. It's pretty freaking good. I'm really, 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 really happy with that. One reason why I'm too far. One reason why I'm really good is because we beat everything else that's out there. Not flow density. Remember, flow density is the best. It's really good if you want to get the best score. So what's the best? I spoiler alerted way at the beginning of my talk. Flow SOM, people like it. You have a good F1 score. So on the red here is flow SOM results, and the blue is our flow magic results. And in we're getting, flow SOM's getting 56% on average, which is kind of what they got in the Weber-Robertson paper, between 56 and 75%, which is kind of matching what we're getting on our results here. You can see flow SOM got 70% on the moderate. I don't know why they did so bad on the easy ones. We didn't cherry pick this stuff. My graduate student's not that smart. So we're getting about 30 or more % better than anything else out there. And we're getting scores like 98%, 96%, that's pretty awesome. And again, the hard stuff, nobody's going to do really good on, but we're still doing quite well, 75% F1 scores. I'm pretty happy with that when there's no events. Not as good as flow density, but probably going to be good enough for the real world. And so all that matters to you is you get a button somewhere. There you go. You get a button, you push it, you get gates. And this is an omic. But this doesn't solve anybody's problem, right? Because we're scientists, right? Putting boxes around dots doesn't solve problems because we're trying to solve, why is this person dying? The other problem it doesn't solve is we're getting labels out of this, like 8PU4 population and BWFR. That sucks. Second problem we're trying to solve. the names of things. Names are important. Names are how we talk about things. If we didn't have words, we can't talk. So snows, I didn't have this here before, just now. Snow is really important to Eskimos. How many words do they have for snow? It's like some very large number, right? Because that's really important. Cell population is really important to us. We have lots of different names to different cell populations. You can't freaking agree on what these things are. You don't. It's not standardized. We use words like dim, low, double positive, bright in different contexts and different situations. Me, what I want to do is, I'm the important one in the audience. I'm not in the audience. I'm the speaker. Never mind. The important thing for me is I want to do big data analysis. I can't take all your data and combine it because you've used different words to describe what you found. And if I'm trying to help Pfizer and they have this whole pot of data, even within Pfizer BMS, I'm sure you guys do it different. You guys are awesome. Pfizer's not here. They're not. I don't know. Even within different silos, right? How you label things are very different. So we can't integrate all your data. And it's not why you chose science. You didn't see somebody labeling a I'm not a labeling person. I'm not a gating person. I'm a labeling. No, it's not why you got into science, right? You want to solve problems. This doesn't solve anybody's problems. I want to solve that problem. The problem is there are many different, and I don't have the good analogy yet. It's like, how stupid is this? It's like if you work with me here for a second. It's like if you have an X-ray of the body and we have the bones and people can't agree that this is the tibia, right? It's that literally stupid. So let's change that. I want to change that for everybody so that I can do this cool science I want to do. And so here's some examples. I just Googled, I don't know what this is, I forget, CD16, CD56. And you see they're gating it all differently. We'll solve that problem. And then they're calling it different things. So how do I solve that problem? Well, my hypothesis, the only hypothesis I have that I need to have work in order to solve this problem is every time I look at a bivariate plot, it's scaled to the interesting data range. We don't draw, these aren't, and it's always kind of like a square. You guys don't use triangles, you don't use ovals in your plots. It's not some squiggly round circle, it's always kind of a square, or at least we can zoom in to the square. Interesting data range is the only thing that has to work. If that's true, now we're gonna do some big data science. So everything I talked about before this was citizen science. I have a bunch of NASA scientists gating data for me, great, that's working. Now, how can I do scientist science? So I want to leverage the knowledge because you guys are smart. You do have some good things going on. You have ideas about how to name things and you have data where you've put labels on this. And there's not many pathways that people use. I don't know how many it is. Is it 100 maybe? This is the T cells, this is the NK cells, this is to, I don't know, whatever cell. There's not a lot of pathways. Can we just agree that these are all the bones in the human body? At least most of the bones. I don't want to do everything, but at least all the important ones. And so what I'm doing right now as we speak, when I'm co-op students are doing this for me, get all the data from Flow repository, and then let's look at all the OMIPs and things like that, and figure out what are the interesting pathways that people go down. And let's what we did for the general purpose gating model is just give us all the data. Now and a lot of it is at the top level of the gating hierarchy. Let's go down specific pathways and gate just those pathways and get the data on those pathways. And then layer and give that to all the gamers and have them gate all that. And the whole idea I have is if we have a population we're trying to gate, we can put it on a 64 by 64 grid and look at where the pixels are and then label that population with ones where that pixels are. We can give this population a computational label that identifies it in the same way that you're using today. Like you go, it's this would be like a CD19 high, CD24 high DNB cell, right? When a lot of times we name these, we have this whole string of positive negatives that effectively give us the gating hierarchy. What I'm trying to change that is into the absolute position of where it is in the plot. And the only way it's going to work if I make this position absolute for everybody. And I'll talk to you in a second how I'm going to do that. And so the label, the competition label is going to be this idea of where these populations are. Then we can assign that computational label a name that we all like foo cells or B cells. But what matters in the computational sense is this computational ID, which doesn't really change. Now, one hypothesis I have is if this is all, when this all works, we can also use this interchange language to do something like translate between FlowSOM or TISNI or UMAP because we can't, they don't have the same words. But if we have, and we do now, drink the Kool-Aid, a way that we can go through all the different bivariate plots in an automated way and get everything really well. If we go down all these different pathways, we can probably find some events at some really good level of sensitivity that match something you got with UMAP. Then we can translate between all these different methods through a bivariate gating, which is kind of the weird way to think about it, but I'm pretty sure it's going to work. So we can have these computational labels. We can call them what we want. And then what we're going to do is, I'm going to tell you how we're going to build the truth map on the right in a second. I'll give you a hint about that already. And then you come along, you have the stuff on the left that you've gated. Now you can see this person is gated. We're trying to put the labels. Imagine the labels are red, blue, orange for these populations on top of what they've gated. And then it doesn't really match up. So this person on the left has split that green population into two. That's okay. Maybe they discovered new biology, and that top part of that population is something that the atlases can change, right? It's going to be a living thing that, hey, I've discovered something new. We'll modify the atlas. And we can also use for this education that they split off this orange and blue. They just do that as one gate and then go, wah, wah. Somebody at ISAC said those are two different things. You might want to split that off and maybe use our words for these two different things because you're wrong. Somebody has to decide what truth is. I hope that we can get there, and that's going to be hard. But how I want to get there, I'll tell you about how I'm going to get there in a second, how we can agree on truth. The atlas is going to grow based on knowledge, right? So we might have the idea of, this is like the Christopher Columbus, excuse my privileged Western world thing. He sailed and found the new world, right? It was always there. We just didn't know it existed, right? That's biology. So if we have the original map, if we find something new, it's not like all those populations are gone. It's not like they've changed. We drop in new things on that map. It's not an infinite map, right? But I want to get that full knowledge to build the truth map. So how I'm going to do this? I'm going to get all the data in the world. So I'm going to start with two plots. Here's two plots of, what is this? I got this result yesterday. This is CD4, CD8. So what we're going to do is here's two different X-rays, and we're trying to find all the bones. And once we have all the map of bones, we can say this is truth. So I want to get all the CD4, CD8 plots in the world and to combine them all. So here's just two. So I'm going to zoom into this data range, and what we're doing as of, well, in the shower this morning, came up with a different idea. We cut off 0.2% of the data to zoom it in. That's probably not the best result because we can lose a little bit of populations that are right around the edge. It's probably have to be a scaled thing to look across all the plots. It's going to work. We're really close. Zoom into just data range. Then we pixelize it. We zoom in the instance data range. We make it to a 64 by 64 bin. And then anytime there's we're going to cut off the density. Anything above 0.2 density from a standardized scale from 0 to 1 gets pushed to the highest level. And that sort of lifts up the rare populations so they glow a bit brighter so we don't lose them. And then there's some math that we do of sort of matrix alignment to align these. And then what we do is here's those two plots in the beginning and we align them. And this is the aligned result between the two we start with at the top left, and we get that. And now we're going to do this at scale. And so here's 4 different plots, 5 different plots at the top. We zoom them all in. We cut them off. Here's the results if we don't do the alignment. And then we see how far we have to shift each of these plots from 1 to 4 versus all the other plots. We get rid of ones that have a big shift because something goes wrong. And then we use the averaged amount, how we shift these, the number of pixels, left, right, up, and down. We're sliding these plots on the 64 by 64 grid. And the math tells us how far that optimal alignment is. And then take the average shift and we line all those plots. And this is what we get down here, which looks to me pretty good. This is the example. We did it at scale yesterday with a whole big pile of plots. This is a CD8, CD4 versus CD8. If we didn't align them, this is what it looked like. If we align them, this is what it looks like. And then I went to Google and Googled CD4, CD8. Most of the stuff looks like this. Thanks for nodding. Most of the stuff looks like this. Now we have a truth map. So can we all agree? So we don't have to agree. Data tells us, when I'm done, this is what CD4 to CD8 can look like under any circumstance. This is the parts that get filled. And then all we have to do is now I feed this to the gamer and say, gate this. And there's going to be 4 or 5 different things. They'll probably put one here, down here, down here, down here. And then maybe let's split this off. Now we're gating that. And then I show this to somebody who nods a lot. I said, can you put names on this? Now that's truth. These are the names for these things. And this is all the options that you have. I think that'll work. Every we're gonna have an atlas. Every page in the atlas is gonna be it's not a book because nobody reads books anymore. We're all on screens. It's a computational atlas. People still read books. Sorry, that was stupid. It's a computational atlas. Every page in the atlas is a bivariate plot. Every chapter is a gating hierarchy. We put it all online. We tie this to ontologies, and that's really cool because now we don't just have labels. By tying this to ontologies, we have knowledge about what that thing is. And so now you can do really cool science, but you can combine flow cytometry data with other kinds of data types because you discovered like, this is T cell biology. Oh, this is also T cell biology. And you're looking at the same thing. You can, discovery is going to be awesome. We can put this out as a resource. I've talked IUIS and ESCA into this. ISAC we're trying to get them on board with this too. We're going to do this anyway. And then this is going to work because you guys are lazy. And it's going to work because I believe that, actually, I didn't have this idea to this now. If you had a button that you could push that said, label my cell populations, put your hand up if you would push that. Yes. Oh, wait. I need somebody's camera. Quick. You got a camera? I take a picture. This is my truth. All right. If you had a button, you all have name tags, so I'm gonna come chase you. If you had a button that you could push, and it's like label my cell population, put up your hand if you would push that button. Fuck yeah, who didn't? So if you wouldn't have that button, I want to know why. Right? Why wouldn't you push that button? If somebody said that's the truth, and we all agreed on that, I don't know, but I'm happy that most of you did, and that's how we're gonna get adoption, right? Because now people will be using it. This is all going to be free. I love my job. OMIC is really nice. Not OMIC, I work for Dotmatics. Dotmatics is really nice. Is that yes, we can make this proprietary thing, but let's just make it open and everybody has that same button and you can use it in OMIC and Cytobank or FlowJo and everything else. I developed a standard called Gating ML, so it all can be interchanged. It's just gonna work. And that's the story. And once we have all this done, this is where it gets really exciting. Now you guys can do science. Now we have entirely interconnected automated workflows from benchtop to desktop to cloud to science, where all this stuff now exists. This is not a fairy tale. All this stuff I'm about to tell you is true and exists. Some of it's in R&D and some of it's graduate student, but I'm on the hook for ML for this year. I want to have the annotation done this year. It's working. You push it today, you can push it. You don't have to push a button. Your instrument is connected to the internet. It sucks up all your data and puts it into the cloud. There's software that'll do this for you. It's now in the cloud. It gates for you. You don't have to do anything. It's gated for you. We've labeled it all for you. Yes. Statistics isn't hard, right? These are some plots we did with pharma. The kinds of things that you're looking for is, what we did in this study is from baseline all my data, all my patients to day 0, and look at the trends over time. And I'm interested in patients that are different, because I'm doing a drug study, and if one patient goes off, that's danger Will Robinson. I want to look at those patients. And so we do all those plots, and then we can pull out those plots for you that are interesting, because that's just stats. And Prism is a great stats tool. We have that. That's not hard. We can automate that entirely workflow. So you wake up in the morning, you go to work, you don't have to gate, you don't have to label, you don't have to do any plotting. You just say, and then it says, "Hey, look at this." It's that voice again. Look at this plot. It's really interesting because it's different. That's science. That's where you come in. You understand that. And we've done all the work, including saying this is the interesting plot, because look, this patient is much higher than all the others. You're doing science, not data processing. And imagine that we can scale this across all your data, right? All these tools are automated. So we can go back to all the data that's ever been collected. We can regate everything for you. We can relabel, you being big pharmaceutical companies, because that's the problem I'm trying to solve. We label everything for you. And then we can put that into a whole data platform. All the data is living in one silo. And then somebody somewhere says, "I found DNB cells and they're really interesting and they do something." And you go like, "That's great. It cured cancer, but does it cure toe fungus? We have a drug for toe fungus. Does it light up that same, maybe it's the same thing." We can ask those kinds of questions with big data because everything is then standardized and harmonized. You can put that all data together. And that's going to be really awesome for me because I give it all for free and I give it all at once. None of this is my work. I tell them to make the database blue. I have to thank, I didn't change this slide. There was a lot of, thousands of gamers. Thank you all for uploading your data. It's a huge thing for sharing your data. This is what's enabled that for you. A whole bunch of students in my lab. I like to thank my collaborators at McGill. MMOS Science, the company who does nothing but help gaming companies do science. Thank Dotmatics. Thank all the people at CCP who put together this in a couple of 6 weeks. Thank the funders, and thank you for paying attention. Don't know we have time for questions. I've talked too long. We started late. You guys were all in the hall. Thank you. Wonderful talk. He said it's a wonderful talk for those of you in the back. Thank you. Oh, okay. No. No. Because that's hard. Let's talk over beers on how you would do that. It's not my problem. So there's a whole cell ontology that's already been done. Experts have sat down and developed something called the cell ontology, and they've done a whole bunch of flow. It's really, really cool. I have no clue how they did that, but they put a bunch of people in the room and locked them up and said, "This is truth." Okay. It is. It is, right? Yeah. That was my contribution. I was like, I went to ChatGPT. I want to do this thing. I'm interested. Yay! And I went in Python and then I gave it to my student. Look, I've solved it for you. Yeah, it's exactly image registration.
Is someone how do you think about self-state? I do a lot of characterizing material, which is like might look a lot different. We have this problem where we define Treg a set of markers, and that means Treg in T, but that same combination.
Yeah. So the question is, I think it's tissue dependency, right? I think one of the things I didn't, it was in my slide. I didn't mention it. I think I have it. Per Bavaria per hockey is probably also per. I had somewhere in there per tissue. We probably want to have normal human peripheral blood is its own thing. So it's per. I have this idea that per tissue, and per species per tissue, per gating hierarchy, per Bavaria. Yeah.
So
Over time, yeah.
So do they look? Is the atlas
right? So babies are probably, but I think for babies versus adults, we just smush it all together. I don't know if you can smush tissues together. Maybe you can. Smushing.
It's a hard problem. So.
Yay! We have time for one more question.
Yeah, excellent talk.
Thank you.
I'm really intrigued because you're going down these gating hierarchies. How does that match up with the expanding size of panels, where they might extend beyond?
Yeah.
Hey, this is working now. Perfect timing. Extend beyond kind of our classic gating strategy.
Yeah. So how is this going to work when we go down gating hierarchies, when we get bigger panels? So one of the first things we did is grabbed all the omics. And so here's a 50 color omic, and let's go down all the pathways in that. Even with large panels, doesn't necessarily mean the pathways you're looking at are probably similar. We just need one panel to do that. Right? There's only so many ways you can get the T cells. Doesn't matter if you throw an extra 500 markers. there's only a few ways you can get to NK cells. So I'm just trying to find the pathways, and pathways go, what these are, I just want to know pathways through markers. It doesn't really matter what panel you have. It's the the pairs of markers that you're going down that I care about. So it's panel independent.
And if you're gonna do like activated T cells, or there's lots of different ways that you can mark activation, is there a point where it turns over to the user again to go and manage?
Yeah, so I'm at the, let's get 99%. Yay, okay, good. Right, there's gonna be some way, if I can get 90% agreement on 99% of the data, I'm done. Fringe cases, you have to do yourself. The buttons aren't ever gonna do everything. You still have a job. Thank you.
There we go. Okay. Hey, how's everybody enjoying the program so far? Thumbs up? Good. And I hope everyone's gotten their lunch and got what they ordered or thought they ordered and sounded good today. Everyone's happy. So really thankful for all of the sponsors that we have for this event. That's how it's free, is because the sponsors generously donated. And one of our sponsors, BD, were the first in line to bring you guys lunch. So thank you very much, BD Biosciences. And now Ravi, who is from BD Biosciences, is going to do his talk.
Thank you, Michelle. Right. So you guys already have got an idea about all the spectral, the panel design, and maybe have heard a lot of things about how the flow cytometry actually works. But today, I just, for the fact that you already have this preamble of all these spectral talks already taken place, we're going to be focusing a little bit about comparing the traditional flow and the spectral flow. And for today, I'm just going to be talking about what spectral flow is. Just give us a short introduction. You've probably heard about this, go through those slides pretty quickly. And what are the similarities and differences between conventional flow with the spectral flow? And talk about the fundamentals of panel design. Laura did a good talk about the panel design and what you should be keeping in mind. But essentially, we'll be talking about what you should keep in mind additionally for the spectral flow panel design. And then what are the tools to simplify those panel design and consideration at the top for the design of these spectral panels. So let's get right into it. So just an overview on what flow is actually doing. So let's ask a lot of questions. What is it that we want, right? In the end, a successful experiment is based on the fact that you get the resolution of the markers that you're interested in, right? And there are a lot of all these background things that are working that we don't really think about. It is, how are these formats being detected? What are the optics? What are the detectors? We really do not worry about that too much, right? But in a sense, the flow cytometry is a process of signal transformation. So what it's doing is it's collecting all this information of the formats and the antibodies on the surface, and then compiling them all together, doing the signal, or kind of formatting those signals into digital signals, and then giving you the output so that you have certain, the expression of the markers that are on your surface of the cells. So if you have a cell type A with 3 markers, which is in green, in the end, you expect 3 markers to come up. So all of this is signal digitalization and then giving you the output of those things. So it is kind of a mixture of all of these things that you will see at the end, which deconvolution in a way is doing that, right? So what are the traditional and the spectral trying to do? They're trying to solve the same problem. The aim is to determine and find out what is the contribution of each fluorochrome, and then give you the output, right, the resolution of that. In the end, the two are trying to do the same, right? The only difference, or one of the major differences, that the spectral has more detectors, right? In the conventional flow, you have these discrete bandpass filters that you use to detect. In the spectral, it's like a picket fence. Everything is there, right? So one of the major differences between spectral is this, that spectral uses more detectors for the fluorochromes. So you, in the end, have more options that you can use for the spectral. And the second one is that the spectral uses unmixing. So this is, again, a software related issue. So there's the hardware, which has more amount of detectors, and the software. So having said that, these are the two differences between the conventional and the spectral, right? So this is what your conventional traditional flow looks like. This is a 28 color instrument, the A5, and you can see that there are channels which are discrete, right? In the spectrum, you have all of these taken care of. So it's like each detector will have multiple detectors. So each laser will have different detectors that are like a picket fence. And in the end, the signal in all of these is detected for each fluorochrome and then stitched back together to give you the output that you would see on the spectrum. So you have additional detectors, which gives you more information, right? In the traditional flow, you're trying to look at one detector and you're looking at only that signal. In the compensation, you're trying to remove the signal from the others. But in the spectrum, you're looking at all these detectors on all the lasers. So you have more information that is being processed, right? So these are the similarities and the differences between the conventional flow. What remains the same is how you're going to be doing your experiment. There's no difference, right? What you're going to be doing in the experiment workflow, you're going to do the same type of sample preparation. You're going to do the same type of sample staining. You're going to have the same type of sample acquisition. The controls you're going to be using is going to be the same. The panel design approach is also essentially similar, right? So remember what we talked about earlier on about the spectral and the spread and the spill. These are physical characteristics of the dyes. That is not going to change. What is changing is how you are detecting this and how you're unmixing it and giving you that, right? So the panel design approach remains the same. You're going to be knowing your value, keeping the biology right in the center. You're going to be looking at the fluorochromes and what can be detected in which detector. And you're going to have kind of, whichever, regardless of which instrument you're going to be using, you can use multiple fluorochromes on that instrument, right? So you're going to be essentially managing the spread in a similar manner that you would be doing it on the traditional flow. The resolution in the end is going to be similar as well. And this is some examples of the resolution. And the top, it's a 12 color panel. It was done on the A5, and then the lower panel is done on the A5 SE, but these are two different instruments. So even if you have two different instruments, are the same instruments where we've done the traditional and the spectral on the same instrument. This is the A5 SE. You can do both of them on the same. For you, data in terms of resolution looks similar, right? I wouldn't say identical because sometimes you have better resolution in the spectral, sometimes you have better resolution in the conventional. But in the end, your resolution is similar, right? So it's the same sample. So what are the advantages then when you have the spectral flow? The advantages is that you can do background extraction, right? Laura talked about a little bit about the autofluorescent extraction. That is something that is possible on your spectral flow. And it depends on the autofluorescence of the cell types, right? Whether they are cell lines, the larger cells, more activated cells, and they help in reducing the background so that your resolution is better, right? You have an option of now using more fluorochromes, right? More detectors, more fluorochromes, right? And it is, of course, now limited to the fluorochrome ability as long as these fluorochromes do not overlap on each other, right? So it becomes, in a way, filter independent because you're looking at the signature all across all these different laser lines and then trying to match and see how different it is from the other fluorochrome that you're going to be. And in that case, we can do up to 40, and now we have gone up to 50 on the instruments that we have. So it's small sample and a high amount of output on that. So you can do a lot of things with spectral that unlike traditional, you would struggle to do that. So this is an example of fluorochrome that restricted you if you had this type of instrument in the traditional flow where you have just the PE, CF194 and PERCP. If you wanted to run PE size 7, in that case, you had to swap the filters, right? That becomes moot now because now, with in the conventional flow, you had to shift the filter sets, but now you have a picket fence of all these detectors that regardless of which format you're going to be, you can use either of them, right? So you're collecting the full spectrum. As long as the fluorochrome is excited by the suitable laser, you can detect them. And this also gives you greater flexibility because of multiplexing different fluorochromes, including those which are quite similar in nature, right? So in traditional flows, you would never be able to do this, right? You had one detector, and if you were to use BB515 or FITCI, it would be impossible to do that on that if you had two different markers, right? So they have very similar excitation and emission profiles, and they come up in the same detector, right? But there are differences. Like if you look at the whole spectrum of the FITCI as well as the BB515, there are differences. And those differences become more enhanced when you're looking at the spectra across all the detectors on all the laser lines, right? So you have these differences that are there in the same blue laser, but you have also there those ones which are marked with the star on other lasers, right? So the violet laser also sometimes excites. So all these additional information that the spectra is collecting is being put together to give you a unique spectral signature. And that spectral signature then gives you the ability to resolve very close emission profiles, right? So these are highly similar profiles, and these are so if you put it on non-co-express markers, of course, it's always better, but you can do it on co-express markers. You wouldn't want to, just because you can do it doesn't mean you have to, but there are examples that you will need to do this. So these are the things that you need to keep in mind. How similar are these fluorochromes in terms of the emission profile, right? So let's talk about the panel design. We know always that the panel design, the thing to keep in mind is the spread that can impact the biology in all your experiments for flow cytometry, right? So if you have two dyes which are spectrally distinct, those are the best ones. You want to choose them first, right? If as much as possible, right? If you have it overlapping in the spectral, you can use them, but again, you can see that there's a lot of spread, right? In the double stain, you can see that almost from 10⁻³ - 3 to 10⁻³, that's 6 log decades of spread. That's huge. So your resolution of your positives from the negatives becomes difficult, especially for the dim markers, right? If you're going to be looking at the dim markers, right? And that spread is a function of the spillover, right? So the more spillover, the more spread. But when you think about spillover, think about, you have 20% spillover, but what does that mean? 20% of spillover for 1,000 receptors of 1,000 fluorochromes is going to be very different from 100,000 receptors. The 20 is going to remain the same. The percentage is going to remain the same, but that's a huge amount of spread when you have high antigen expression or a brighter marker. So then it is also dependent on the MFI, which is dependent, of course, on the antigen density and the brightness of the format that you're using. So your spread becomes dependent on the antigen density, receptors, biology, your fluorochrome brightness, which is what you're going to be using, your reagent, as well as the detectors that is that is available for you. So it can impact resolution in both conventional and spectral flow. So it's almost similar, right? Of course, in certain things, because you're taking more data for the spectral, you can analyze them a little bit better. In some instances, you can see in the middle panel at the bottom that the spectral has done a better job. But in the conventional, if you look at the top right, that seems to be having less spread than this. But the bottom line is that there will be spread because the fluorochromes have not changed, your biology has not changed, your cell samples have not changed. The only thing that has changed is the instrument and the hardware and the software, right? So the fundamentals of panel design in this case to minimize spread still remains the same. The challenges remain the same, right? So for that, you need to know the instrument, you need to know the biology, right? Especially the co-expression, antigen density, classification that Laura talked about in the morning, and also know the brightness and match the correct antigen to the correct format, right? Dim antigens, bright formats, vice versa, right? And keep the co-expression in mind. Choose fluorochrome combination for co-express markers with minimal, which show minimal spread into each other, right? So these challenges are going to be the same that you need to handle in the spectral as well, right? That you already did. And then the tools that are there available also are similar. You need to understand what is there in the literature, where is this antigen expressed, what are the things that go up and go down, what are the brightness of those formats, what are the antigen classifications, the primary, secondary, tertiary, or is it the, we know an actual number, which is the antigen database that we have, or the BD spectrum viewer, you can go and figure out what is the spectrum spillover for a particular dye, and then design your panels, right? So to summarize, the conventional and the spectral use two different systems, right? But the end goal is the same. The conventional collection is in a discrete emission profile. So it's almost one-to-one fluorochrome. The spectrum is the entire spectrum of all the lasers, right? The collection of the entire spectrum makes it advantageous because now you can, have higher resolution, you have more data, you can abstract the autofluorescence and make a better resolution of autofluorescent cells, monocytes, granulocytes, cell lines, to give you a few. And you can have more fluorochromes, right, per panel that you can do. So development of larger panels becomes easier in the spectrum, right? But like I said before, just because you have the option doesn't mean you have to use it. There has to be a strategy, right, in terms of understanding the co-expression, where the spread is taking over and what you're going to be doing to mitigate those spreads. Mitigation of spread is among the key panel design consideration that you have to keep in mind, right? We are not telling, and no one is going to tell you that do not use this fluorochrome with the others. That's not the case. You want to make choices, informed choices of how you're going to be using those fluorochromes, right? So the spectral flow, it does not eliminate the multicolor challenges, multicolor flow challenges, but so you still have the spread, and that is based on the biology and the panel design principles. The same tools that you can do. So let's talk a bit about the tools, right? And that are unique to the flow cytometry. So this is just an example of the picket line fence that you have of the A5 SE versus the A5 with the X20. You can see there's more detectors in the spectral instrument, which is at the bottom, compared to what you had at the top, right? When you're looking at fluorochromes, we talked a bit about the similarity index, right? It is a quantitative measurement to assess the fluorochromes, right? And what is the essentially what is the overlap of these fluorochromes? A low similarity means that they are very distinct. A high similarity of close to 1 means we are very similar, right? Similarity should be used. You can use this to identify and exclude fluorochrome combinations with identical or overlapping fluorochromes as much as possible, right? Not always the case, but you can at least start off with the premise that you're going to be using non-overlapping fluorochromes as much as possible first. So these are examples which are in the best case scenario you would want to use. The non-overlapping fluorochromes, right? For all this, you would get best resolution. This is comparing with the compensation, which is the conventional flow, with the spectral flow, right? But we know in reality, we have to work with the overlapping dyes, right? That are excited by multiple lasers that show up in different detectors. That's just what we have to deal with. With those, with compensation and with unmixing in the spectral, you would see that you can use both of them, right? You can use spectral as well as things. It becomes a little bit more trickier when you have highly overlapping fluorochromes, right? We talked about how it is discrete detectors in the conventional flow, but in the spectral, you still can use this, right? It is not always optimum. You have to be very strategic about it, use it on non-co-express markers, CD4, CD8, for example. If you know that they are going to be spilling over, you can use it in those markers and make some sense, or use it in a high antigen density markers like the CD3 and the CD4, which are far apart. So even if there's spread in the negatives, you will still be able to manage it, right? So this is only possible in the spectral. You would not be able to do that in the thing. Additionally, if it is identical, right, you can't do it in the regular or in the spectral because, so the point of this slide is to know how similar your fluorochrome is to the others. So, you have different vendors selling you different fluorochromes, different formats. We will call it something, someone else might call it something else. Make sure that these are similar or not similar, right? How much are they similar, right? Right. So with that said, we are trying to mitigate this aspect of fluorochrome by trying to generate dyes that are spectrally distinct in a sense that these are dyes that are excited by only one laser, right? Or possibly only one laser with minimal excitation from the other lasers, right? Using AI, using dyes specifically for those type of excitation emission. At this point, we are targeting the blue laser and the yellow-green laser. We've come up to 7 different distinct spectrally enabled markers, right? So we are going to be looking at something that is excited only by the 488 laser or only by the 561 laser. We anticipate 7 in the blue channel and 7 in the yellow-green channel to come by soon. We have something that is equivalent of FITC, the BB515 already, but we have something between the FITC and the PE channel that can be run only the spectral, which is the RB545. So this is in between channel. I'll be talking a little bit more about that, or if you can come by if you want some more. We also have the RB613, which is the PE size CF594, or the Texas Red on the blue laser. So excited only by the blue laser, seen only by the blue laser. The PE CF594. Unlike the PE size CF594, you would see it in the blue and the yellow-green laser. This is only by the blue laser. And then you have the RB705, which is again in the PERCP detector. And then you have RB745 and RB780. And for the yellow-green laser, you have the PE equivalent, the RY786, and the PE CF594 on the yellow-green laser. So I'll be talking a little bit about this. So let's talk about the PE. Why do we have to do this? Traditionally, we had the PE, and that was a gold standard. We tried to fit it in the lasers that was available. At that time, the blue laser was the first one which was available. So you put the FITC, we put the PE tandems all on that. Now we know that the yellow-green laser is a better laser to excite the PE, and we have a distinct laser, which is separate. But now what happens? If you're going to be using both of these lasers, they're going to be exciting the PE molecule. You may have better excitation by the yellow-green laser, but it's going to spill into the blue laser as well. So what happens is it's going to be a lot of spillover, and you're going to not be able to use some of these markers. So this RY586 is a PE equivalent, is as bright. It's a small molecule, so you can use it for intracellular staining as well. And it's restricted to the yellow-green laser, as you can see from the right plots over there. This is the PECF594 equivalent on the yellow-green laser, right? Again, compared to the PECF594, specific for the yellow-green laser, right? Just a few examples to give you a sense of what this is going to be, right? And then we have the RB613. Again, this is the PECF594. We have a lot more dyes coming out. I'm not going to show all of these data right over here, but I'm going to give you a little bit flavor of what is now possible, given that you will be able to, you can use these distinct ones. The RB780 is a PE size 7 equivalent, specific to the blue laser. So you can use it on the blue laser. With careful strategy, you can use it along with PE size 7 on the yellow-green laser. Again, co-expression, spread, antigen density, all of that becomes the key. And then it boils down to knowing what you have in your lab and what you can buy, right? So knowing the vendors, knowing what the excitation emission of that format is, getting that information becomes the key. Here are some few examples where you can see that similarity of those dyes is very, very similar, right? So essentially, even if you use it on a spectral, it will be very difficult, right? It will be huge amount of spread, huge amount of spill. So the suggestion would be not to use these type of fluorochromes together, right? Alexa 405, BB421, super bright, they're very similar in the excitation emission profile. So it becomes difficult to handle all of those things. For the 488, you can see that there are multiple distinct fluorochromes. Again, not to say that you cannot use it. These are very similar. All I'm trying to emphasize is that there are similarities that you need to keep in mind and use it with caution, right? Only when you really, really want to use it, right? So these identical fluorochromes cannot be used together or should not be used in a panel, and they should not be used unless absolutely necessary. So now you have a scenario where you do not have these formats in any other, but BB515 and FITSI or Alexa 488. Then you may want to think about using it. What are the controls? What are the FMO controls? What are you going to do to identify if there's going to be spill and spread, right? There might be limited availability of these reagents. So are these reagents that are available compatible with each other? Are those markers going to be expressed on the same type of cell? Co-expression becomes the key. But as I said, keep that in mind before you start doing that, right? This is an example of very similar dye, which is the FITSI and the 515, right? So we show that this can be used. Again, not that it's, we saw the similarity was almost close to 1. It can be used, but it can be used on non-co-express markers like CD4 and CD8, or it can be used on high expression markers like CD4 and CD3. So there's a lot of spread that you can see in the CD4 negatives, which are CD3 positive. So there's a lot of spread, but you still make sense and identify the CD4s, right? It's not that you want to do that with all the markers. It will be very difficult if it was not CD4, because that's a bright marker. Right? What you can do is look at alternatives. This is the RB545, which is very close to the BB515, but not identical. It's more right shifted. So this is one that I was talking about that comes between the PE and the FITSI channel on the blue laser. Right? By making a switch, which is a little bit different, you can have more resolution than you would have with the BB515 and the FITSI. Again, if you can use it on the non-co-express marker, that would be better. But even with co-express, you can do that. You can see this, right? So better fluorochrome combination will give you higher resolution and more flexibility, right? You can use it, but be cautious, right? These are the fluorochrome combinations that just a flavor of what we have found seem to work best, right? Or at least you should not be the ones which are in the same row. I would suggest being careful about using them together. So you have BITSI, BB515, BB515 and Alexa 488. You have the Kira Blue 520, BioBright, FITSI. Those ones are very, very, very similar. So even with the spectral, it becomes a little bit challenging to use them, right? For the 405, you have the BB421 and the 450 that can be used together, or the Alexa 405 and the Pacific Blue that can be used together. So we avoid using fluorochromes from the same row at the top, and instead use one which is in the row 1 and the second one from the bottom, right? In the 561, you can use the PE size 7 and the PE Fire 810, or the PEYO 770 and the PE Fire 810, right? For the blue laser, sorry, for the red laser, you can use the size 7 and the APC Fire 810. They're very, very close, but still distinct enough. But you cannot use the APC size 7 and the APC Fire 750. It becomes more of a challenge if you're doing that. So using 3 fluorochromes from each group Challenging, but can be done. Be careful and cautious about that, right? So to conclude, spectral flow defined is defined by acquisition of the entire spectrum of the fluorochrome, like we talked about. It is looking at all the laser lines and stitching all the information in the different detectors in that laser line and giving you an output that is distinct from the others. It is all based on having more detectors than fluorochromes, so which makes it now open to higher panel design. Spectral, it offers advantages, of course, right? It is more advantageous because you can do remove the autofluorescence from your samples. You can have choices now. You have more choices now. You have more fluorochromes that you can use, and you can increase your panel size. So on a small sample, pediatric blood, small little stem cells, you can now do higher panels and make sure that you get the resolution that you intended to. It is not immune to challenges. So you will see spread still in your panel, right? But you can manage that with careful strategy. The same tools that you use for your conventional flow, use the same ones for the spectral as well. And for that, again, literature, you have Spectrum Viewer. We have the new BDRC, which is on our FlowJo portal. You can go there. There's a panel design builder on that, and that can give you an opportunity to look at the spectra as well as how they spill into each other. You can go ahead and design the panels, share it across different institutions. You can have your own account, share it. It'll be in the cloud. You can then, bring it back again. All the OMIPs are there in that module. So you can use that as your building blocks, and it'll tell you if there's anything that seems to be suspicious in terms of spillover and spread, and give you caution when you're using those. Again, you're going to be in control. You're going to be doing the choosing of those formats. It'll just tell you what may go wrong if it is co-expressed, right? So fluorochrome choice still matters. And the consideration for spectral is to minimize the challenges which is introduced by highly similar fluorochromes. So if you want to look at it, start by building panels with spectral signatures which are absolutely distinct, then work your way towards non-co-express markers that are overlapping or similar in nature. Okay. I'm going to end over here and open it up for questions. And we are here still. If there's any questions, you need help with designing panels, 30, 40 colors, we'll be glad to help you out with those as well. And if you have questions about the new dyes, feel free to come by and talk to Michelle. She has some few samples as well, if you want to try them out.
That I first went to one of these Northwest Regional Flow Cytometry meetings, and since it's kind of fallen off, and I'm super happy that Michelle and the board has decided to revive it. Today, I am going to be sharing some practical insights for using analytical tools so that you can get the most out of your cytometry data. I manage the Human Immunophenotyping Core at Benaroya Research Institute, and you'll note I put the disclaimer that I am not a bioinformatician, but I have for over the past decade and more dealt with high-dimensional cytometry data. And so what I'm going to be sharing with you today is kind of my struggles and successes in incorporating high-dimensional analysis. And so the talk today is really a culmination of all the people and conversations I've had about data analysis over the past decade. Plus, this includes people at Benaroya, particularly those in the Human Immunophenotyping Core and Alice Long's lab, as well as those in the cytometry community, and some vendors of analysis software that have been very kind to have discussions with. I want to acknowledge them up front. So has this ever happened to you? So I'm going to have some interactive points here. If you're a shy person, you can see they're coming. I have a little raised hand and the question I'm going to be asking. So who here has ever generated cytometry data or analyzed cytometry data? Yeah, good, great. I'm looking the right audience. And keep your hand up if you've done that with a panel with at least 10 parameters. 15, 20, 25. Yeah, there's some people with some high dimensional data coming right at them. And this is just a glimpse of the future. Back when I started, we were doing 4-color panels on a caliber, conventional flow cytometry. But with the advent of new technologies such as mass cytometry and now full spectrum or spectral flow cytometry, the dimensionality of our cytometry data just continues to rise. And so how do we extract the maximum insights from this high dimensional cytometry data? I would suggest you turn to computational analysis tools. And when I was just starting out in this space, Cytoph was coming online, and that's really where I joined this high dimensional field. They were churning out tools monthly, and I thought, hey, if I just wait, the magic tool is going to come out that tells me how to analyze my data so I can produce nature-worthy papers. So if people also experience that they found this magic tool, yeah, no hands. And the reality is that if you don't match your tool with your type of data, you're really not going to get the answers you need, and you end up wasting a lot of effort. There are a lot of tools available. So a year ago, there was this post on LinkedIn by Mike Leopold. So he runs Cytoforum, which is a Cytoph community, but they are very involved in high dimensional. I suggest you check it out if you haven't already. And he had this post where he kept track of some metrics, but the one that interested me was that there was 262 algorithms published used to analyze mass cytometry data. And so I had asked him if there was an update to that, and today there are over 500 published algorithms. And so you might argue, hey, more tools is better, right? As the dimensionality of our data increases, we're getting all these new tools to be able to analyze our data. So I wanted to ask you, yeah, how many of these tools have you used? Have you done some clustering, some dimensionality reduction? Who's used at least one computational analysis tool? More than one? Yeah, not a whole lot of people. So what's the problem here? And so a group of us, including Anna Belkina, you'll hear from later. Oh, she walked right in. What a great entrance, Anna. You'll be hearing from later today. A group of us asked, why aren't people using these tools? There's a lot of high dimensional cytometry analysis sets out there, and people are still manual gating. And so when we polled over 200 about what the impediments were for them adopting or at least trying some analysis tools, one of the main categories of reasons why they wouldn't is selecting a tool. And specifically within that, they were unsure whether the tool provided the appropriate analysis for their questions. So this insight has really informed a lot of the remainder of my talk. So how do you even start? There's so much data, so many tools. So what I'm going to go through as a 3-part talk today is 1, reiterate what analytical tools can do for you. Secondly, I'll give you 3 practical considerations for finding an analytical tool to meet your needs. These are components, parameters that I've found were most relevant, and then give you some examples of tools that I've actually applied to some of our data. And really what I want you to take home is that you can do it. Just simply try
any analytical tool. So what are the advantages? I think it's probably obvious. People have talked about these things ad nauseum. Efficiency, right? You have less time, analyzing manual gating, less effort, and then it's scalable, right? So, it takes 7 or 8 seconds to process. That's great. Also the consistency. So because a lot of these are user independent and objective, then you don't run into the problem of, your 30% error on ground truth, which is manual gating across individuals. And then also because it's unbiased, you can uncover populations potentially that are novel, that you weren't explicitly looking for. And this, of course, requires some investment, such as a learning curve to get, operating on the tool if it's not customized to press a button. And then the time and effort to evaluate which tool is going to be best for your needs. And so that's what I'm hoping to share today, is to cut down on what the investment would be need for using these tools. And so the 3 factors for selecting a best tool that I found really rely on the analysis goals. Can we answer the questions that we want to answer? The data quality that you have, both size and consistency, and then tool accessibility, just whether we can operate the tool, if we can find it, and if there's some costs associated. So the first of these, analysis goals. So what I've found is that the tools really come to answer 3 different types of questions. So you can ask a question where you want to assess known populations, such as are regulatory T cells depleted by drug in this case. And so the gold standard would be manual gating, right? And you can send your grad student or technician to gate all these populations. But the computational approach would be some sort of automated gating. The thing here is typically we require an a priori subset definition. So like when you say Treg, what do you mean by Treg? So benefits of this question is that you have known subsets, but if using computational analysis approaches, it may be variable in the results you get or not comprehensive. Another option is to explore data-defined subsets. And so this might be like, how do Tregs change after treatment with drug? And so this would let the data determine your different subsets rather than having a priori expectations about what would come out. Important points here are that it matters which markers are used. And then nicely, you get a comprehensive set of annotations. So each cell is going to be defined in a cluster, and it's unbiased. And outside of picking those markers, then the algorithm is the one, placing the hard edges. Problems is that it may not align with the established subsets, published subsets, or previous iterations of using this clustering. And it's difficult to add a sample onto this. And so some other, I think Garrett covered what is a cluster, and I'm going to kind of reiterate this point. So a cluster is, there's a couple definitions, but layperson is a group of similar things positioned or occurring closely together. And bioinformatician type answer is clustering is an unsupervised machine learning technique which finds certain patterns or structures in the unlabeled data to segregate them into different groups according to their properties. But we can sort of intuitively understand clustering with this nice simple example here, where if you look at 2 parameters, X and Y, against each other, there's just kind of groups of things occurring closely together. But it's a bit subjective. So hands up, what is the natural grouping among these objects? Yeah, so if you were to separate these Simpsons characters, what groups would they go into? Well, you could, like on the left here, separate Simpsons family from school employees, but you might also separate males and females. So it really does depend on what parameters you're addressing. And sometimes that can behave in surprising ways. And the example I gave before was how many feet do they have on the ground? So, so we have the clustering. So you get to explore data-defined subsets, but it may not always match up with what the literature subsets might be. And then the last goal for analysis, which is really a follow-on to the other two, is to discover differences. So an example would be, what feature of Tregs distinguishes better outcome to drug treatment? So if you have responders and non-responders, what differs between them? And so really it just incorporates statistics. The thing that's great is that it's focused analysis or subsetting. So you're not worried about all the things that aren't different. You're just focused on the true biology that's interesting. But in that process, because it's not overseen, it could amplify artifacts or miss broader trends that maybe aren't statistically significant, but still of interest biologically. And oh yeah, and then you may iterate through multiple goals using an appropriate tool at each stage. So a second important factor for selecting a best tool I've found is data quality. And by data quality, I mean data size and then whether there is batch effects. So really the data quality in these two components determines whether the data can be combined across samples before being analyzed. So for instance, some clustering algorithms will take all your data, all data points from every single file you put in and mush them together. And so that can create a very large amount of objects or cells. For instance, 100,000 cells from each of 100 samples. Now you're at 10 million cells. You could see the same being for 500,000 cells and 20 subjects, et cetera. And you'll see this is a publication where they showed the runtime necessary to run the clustering for each of those. Sorry, I don't have a pointer. Run the clustering for each of those with a couple different algorithms. And you can see lots of them end up into the greater than 1 day computation memory exceeded by the time you get to 10 million cells. And then the other reason they might not be able to be combined across samples is simply because there's batch effects. So for each dimension, there might be slight variation day to day, even sample to sample, that causes it to be systematically different when you combine them all together. And so here's an example where we had 2 different studies. I combined all the data from those 2 studies together, clustered them on the same parameters, but because they were completely different intensities, they pull out very distinctly here. And so that can lead to inaccurate clustering. And then that issue only grows with greater dimensionality. And I will note that in my experience, the data type in terms of conventional versus spectral versus mass cytometry is actually not as big of a bugaboo in this whole scenario. So long as the compensation is good, compensation or unmixing is good, and there's appropriate scaling or transformation. And so the last of the factors that I found to be really important for selecting a best tool is just the accessibility. And by accessibility, I mean you can find it. There's an option that meets your needs. It's easy to use and that it's within your budget. And so I think of tools to be mostly on a spectrum. And I will say, not all are code and not all cloud-based platforms before I say this whole thing. But generally, on the one end, we might have R or code-based algorithms. I have some listed under there. You may have heard before, FlowSum and Phenograph, for instance. And these are great because there's a ton of options. They're almost always free. Issues are, you can't guarantee they're supported, either that there's supporting manuals or that they're supported ongoing. And then they typically lack a nice user interface. You do have to look at some lines of code. On the other end, there are solutions to this where they've made cloud-based platforms so they can take one or more of these algorithms and really build a user interface and experience around it that's useful to the user. I've defined these in 2 different categories, either aggregators such as Cytobank, Omic, and Tursin, which would like host a bunch of algorithms that then you can determine how to put together, or there are other single algorithm type cloud-based platforms such as Astrolabe or Ozette, which have really built upon one algorithm, but used it in multiple ways to give you full service across the analysis. The downside is that this has the fewest options. They do cost money typically, but you get the benefit of, you know, much greater support and this easier user interface. In the middle, I found a nice medium where there's software plugins. So FCS Express, Flojo have these options to pull in plugins, and these borrow from some of the best features of the R scripts so that you can have it without having to interact with R. And I will say that learning just to type a few lines of code in R has been the most freeing thing for me. So it opens up a world of opportunity. If you at least try a plugin and see what that can do for your data, I think you'll be hooked on the high dimensional computational analysis train. So really this sets us up for this kind of rubric for identifying a best tool. Each of these boxes representing the type of question we have, either assessing known populations, exploring data-defined subsets or clusters, and then discovering differences. And for each of these, tool usability and then data quality on the y-axis can modify our need for a tool or another. And so I'm just started populating this with just a few algorithms tools that either I'm familiar with or that represent their particular goal well. So if we all relate this to manual analysis, yes, it can handle big size and technical variation because a person can come in and change the gate at each step. It's not that hard to do, but it does take a lot of man hours and effort. So I put it a little lower on tool usability. Then there's automated gating, which for the most part, I've seen be a little more sensitive to things like technical variation, and then size of your data. And then there are some programs that are very specific, Astrolabe and MaxPar PathSetter, for instance, that seem to have good tool usability. And because they analyze samples on an individual basis, really can take in different sizes and account for the technical variation well. Among exploring data-defined subsets, I put clustering here. Tool usability can vary, but a lot of these combine all cells together before doing their clustering, and so lower for the data quality. And then I put 2 here, Faust, which underlies Ozette's platform, and Discover, which we've developed at BRI, are both much better against technical variation, again, because they analyze on a per sample basis. I will go a little more into Discover later. And then discovering differences. So Citrus has been around a long time. Tool usability is dependent. It's been rolled into Cytobank, so that makes it much easier. But again, it condenses everything down before it starts it concatenates everything down before it starts clustering. T-Rex similarly concatenates everything before doing some analysis. And the third one I have there is Impact, also a Benaroya program, which I'll go into a little detail on. So that's the rubric for picking a tool and just populating it with some of the tools that I've thought about. You may have also noticed you can do some tricks to move from one side of the axis to the other. For instance, if you assess known populations, that does not mean that you can't discover differences. If you apply statistics, you'll get that. Conversely, if you discover differences and there's like a cluster and it's just got some weird definition for it, you can come back and apply some different algorithms in order to give an annotation to that. Similarly, for the y-axis, technical variation, if you want to limit technical variation, you can do things like in the wet lab, such as alignment beads, barcoding. For Cytoph, we can do frozen panel, which keeps consistency really strong. On the analytical end, there's batch correction algorithms and downsampling, which can help. And then for tool usability, so anything that helps the user interface, such as, or helps the user, such as step-by-step tutorials, adding the statistics onto the analysis, adding visualizations, generating plugins, or just putting it on a platform that has a lot of support can help with tool usability. And so now I just want to go into 3 examples of where I've used these 3 features in a rubric to kind of help me select a tool to use for analysis. So in this first example, we wanted to compare drug effects in rheumatoid arthritis, and I'm purposely not giving much information here, I'm sorry, but the blue below tells us our requirements. So we were interested in particular subsets that changed in other trials. So really assessing known populations was key. For our data quality, there was a lot of samples and a lot of batches. And in terms of tool accessibility, we were new to computational analysis. And we didn't have a dedicated bioinformatician, but we did have some money. So what worked out well in this case was to take on Astrolabe for our analysis. So Astrolabe is a cloud-based platform. It's kind of full service. So you input just the full FCS file. You don't have to do any gating on it. It does the pre-processing. It does cell subset labeling, which underlying this is a hierarchy that you don't see kind of outlined here is an example of one branch of that hierarchy where you start at the root, go to T cells, CD4 T cells, T conventional, effector memory, etc. It builds in follow-up analysis, so differential abundance of those terminal nodes or differential expression. And then, of course, it's cloud-based, so it has that nice interface. And so what you get out is these annotated FCS files. So each of your cells gets a label. And then you also get the population frequencies and MFIs. And so this is just an example of overlaying annotation on top of 2 of the samples. So you can see it's very robust against technical issues such as a variation in batch effects, where this CD4 versus CD8. And you can see it differs a lot, yet it still pulls out the CD4s and CD8s. And then we can take all this information, such as frequency information, and see if there's a difference in drug response on our end. Another example is the phenotyping of autoreactive T cells in type 1 diabetes. So here we didn't want to explicitly look for biology defined subsets. We were going to let data define the subsets. And so we wanted unbiased CD8 T cell populations. We wanted to assess a rare self-reactive subset. The data quality, there was, it was basically light on data, but there was 4 batches and then 2 different Cytometry instruments that we used. So the potential for variation. And then in terms of tool access, by this point, I had nominal comfort in R in that I could write out a couple lines if somebody told me what they were. And we had support from a bioinformatician. And so together with our bioinformatics group, we developed this tool called Discover, which you input exported FCS files of gated subsets, a parent population in this case of CD8 T cells. You tell it the markers, and the antigen-specific subset thereof, which is on the right. You tell it what markers you want to use to cluster. It clusters it individually, so it's robust against batch effects. Then it aligns those clusters across your whole sample set, and then you overlay the antigen-specific population. And so it currently exists as an R package that you can get there. Minimally, it can be run in just like 5 lines of code that are well written out in the instructions on GitHub. And what you get out is these cluster heat maps, so you can see the features of each of your cluster. You also get the cluster frequencies of both the parent and child subsets. So in this case, total and islet or specific or autoreactive CD8 T cells. And so this is a publication where you can see that islet specific CD8 T cells tend to have increased frequencies of each of these 3 clusters. And moreover, rapid and slow progression was enriched for different phenotypes. And then the last question or project that I'm going to share today is looking at immune differences in Down syndrome compared to healthy controls. And so here we did indeed want to discover differences, but specifically the interest was in identifying differences in gateable populations. So the PI in this case was fed up with these kind of like clusters that were amorphous and didn't really relate back to reported biological subsets. In terms of data quality, there was quite a few samples of a high number of cells. There were 2 panels, but it did have barcoding. So a mix of potential technical variation with a lot of adjusting so that was not the case. And then tool access. In this case, we had full support from a bioinformatician. And so what the PI wanted did not already exist. So with this bioinformatician, he developed what's called impact. And so here, what the input is a subset that you want to have parsed, essentially. And so here the example is naive CD8 T cells. The other input is gate coordinates. And so it can be positive, negative gate, like the first example there. It could be quad gate there, or it could be, some sort of free form gate, as in the third example. And what it does is it does exhaustive permutation analysis of like combinations of 3 of these markers within that subset. So you get this huge matrix of frequencies that you can kind of put together in different ways. So in this case, there was, over 100,000 different permutations of 3 markers or gates. The nice thing is there's no downsampling. There's a bunch of statistics laid on top of it with the ultimate end goal of understanding differences. Right now, this impact requires proficiency in R and statistical analysis. But with a bioinformatician, what can come out is essentially differentially abundant subsets. So this is really nice in Down syndrome, which is in red and is in the right part of this heat map, compared to healthy controls or type 1 diabetics, which was in the left. There's several B cell populations that are different. And then one was within the naive B cell, and specifically what they labeled as activated, the CXCR5 negative, CD11C positive subset. And so this is the data in the plot that came out of impact in terms of numbers. And then they could go back to the actual data, apply the gates that would have generated those permutations, and gate that exact population. So in summary, getting the most out of your cytometry data, I want you to take home that you can do it. So cytometry analytical tools can help to efficiently and robustly extract insight from your data. There are many options for computational analysis of cytometry data. I hope I've given you some tools in order to narrow this down for your specific needs. Specifically, does it answer your goal or question? Does the data and quality or data quality and size of your data set, is that compatible with the tool? And do you have access or proficiency with the tool? And then more broadly, just want to indicate that having a clear picture of your biological or scientific goals helps to streamline the assay design and your analysis strategy. Stepping into your discomfort zone to test out an R package or a FlowJo plugin opens up a world of opportunity for you. You will be hooked, I guarantee you. And just to remind you that no tools are completely hands-off, so all require some biological consideration and intelligent use and interpretation. So also a reminder, you are not alone. Reach out if you want any more help. That QR code sends you to the Human Immunophenotyping Core website. And with that, I'm back to the acknowledgements slide. Anybody has any questions, I'd have to take them. Thank you. Thank you very much.
Yeah, so up next, I enabled the chat for people online, so I don't know if anyone has tried it to ask questions, but we have the roundtable discussion coming up. If there are questions online, you can in the chat say whether or not your mic is functional. Hopefully it is if you want to ask questions. So next, what's that? What? You need to go to the Teams. Or I can read them off. Okay, so we have a panel of people from various different professions within flow cytometry that are going to come up and do a discussion, roundtable discussion. This is supposed to be interactive. We encourage your participation. The panel is going to be assembled here, go through whatever Thane put together, which I believe is going over kind of where people came from, how they got into the position that they're in, and then open it up as, you know, different types of questions, whether you're just starting in your career or you're trying to move into a different place in your career, you're just curious. We'll let them get set up, and just don't forget, we have 2 more talks. We have an hour of the poster sessions where the presenters of the posters will be available next to their poster, and the vendors will all still be at their tables. And then at 3 o'clock, I believe, we are going to be back in here. Sorry, it's 3:30. So it's 2:30 to 3:30 is the break. And then 3:30 to 4:15, we have Catherine, who is going to be doing a wonderful talk about standardization and validation. And then Anna Belkina, who's going to be doing a talk about bad data, which you should not miss. And then we have the reception. So go ahead and let you guys get all organized. And I'm just going to take this down because I need to do something. So.
Alrighty, well, I think we should get started now since I have the team is assembled for everybody. So thank you for coming to this talk on careers in flow cytometry. As Michelle stated, I want this to be kind of an open conversation. So this is to kind of answer any questions that you guys may have. So if you don't have any questions, then it's going to be a very short talk. First start by having everybody introduce themselves, give a short introduction and kind of how long you've been in flow and what areas you've worked in flow. So I guess we'll start with Lynette over there.
Space, so I get contracted basically to do the flow cytometry work. So if you guys don't want to do it in-house, you call up someone and say, hey, you know, can you do our flow cytometry testing? And I actually saw how important that it was to the drug development process that I created my own company. So in 2009, I started out and created Flow Contract Site Laboratory, which is dedicated to the technology of flow cytometry and having clients contract that technology with us. And so during that, I played many hats, careers in flow cytometry. You name them: business development, processing samples, analyzing samples, scientific review, writing a report, data interpretation, you name it. I have done that career for the contract research organization space. And so I'm happy to answer any questions that you have on being not only a business owner, but also on the flow cytometry aspect of it.
And then Daniela.
Yeah, everyone. Daniela Andrade. I'm the flow cytometry specialist with Miltani, based here out of Seattle. I've been with Miltani for 3 years. So my first role was an application scientist. And I've been doing flow for 10 years now. During my PhD, that's when I really started doing flow for my studies with dengue virus infection. And after that, I did a relatively short postdoc at Seattle Children's. And that's when I started diving into the B cell sorts and those antigen-specific B cell sorts, which are a little bit difficult sometimes for downstream sequencing. I love that part, but I decided to become an application scientist. But now I moved up to AKA your sales rep. So yeah, I interested in sharing that point of view as well with you.
Hi, I'm John Louis. I'm at Sonoma Biotherapeutics. It's a CAR T company startup in Seattle. I learned flow here at the Hutch more than 20 years ago on a fax caliber. But I learned from the HIV Vaccine Trials Network, the HVTN, here at the Hutch. Also went to work at a BRI, so had experience on the academic side, and then moved over to the industry side, where I've worked at various different companies for the last about 12 years. I've worked in many different facets, and on the research side, on the discovery side, to the more process development side, so similar to what Garrett was talking about at BMS, and now working on the translational sciences side, so on the clinical side, so looking at more post-infusion monitoring, and so lots of industry on both academic and industry.
Thank you. Good afternoon, everybody. My name is Mark Reese. I've been in flow probably for 30 years or more. I started out in the 80s back at Scripps Clinic in La Jolla, working underneath Dr. Larry Sklar, who famously told me years later, "You're an example of somebody I can take off the street and give him a job." I'll never forget that. I had a master's degree, but he didn't think much about it. Anyway, he taught me flow cytometry, and I worked in the core lab there with 3-4 other people. It's where I learned flow cytometry in the beginning, self-sorting. We had a FactStar Plus, if any of you remember that. Fact scans, all kinds of old instruments. From there, I went to Genentech, actually, and worked in the core lab there, doing self-sorting and analysis, and spent a number of years there. Then had an opportunity to move up here to the Northwest and have my own lab over at a company called CellPro, which was in the east side in Bothell, where a lot of currently manufacturing facilities are for Lyell, for BMS, used to be Juno, so forth. Used to be Amgen out there. And that was a great time, but we were sued, we were closed, and we were all out the door. That was my first experience with the commercial side. Now, well, the opportunity came up, though, to make a transition from a research position into something in between commercial and research, and that was as a field application specialist. And I enjoyed that. I was a scientist, right? But now I got to travel. I got to go all over the world and work with customers who were using what was called laser scanning cytometry, which was mostly used in pathology, for tissue sections and staining. And I eventually did well enough. I was a director of operations for Europe for a company called CompuSite based out of Boston. And I ran that, lived with my family there. They moved me there to Europe. I lived there 4 years. 9/11 hit, and I had to move back to the United States for various reasons. But I picked up in a more commercial role, another commercial role with Beckman Coulter. Here, I was the Western Regional Sales Manager for Beckman Coulter for Cytometry. For a number of years, I was there. So you'd think, wow, that's enough for a whole career right there, right? Well, believe it or not, that was 25 years ago. And I'm still doing stuff. I actually retired and had an opportunity to sit at home for about a year thinking about, geez, what a waste of time. There's a lot more stuff I could do. I'm still worth something, right? So I went out and found a small company called Accelix, which is based in San Jose, a startup, perfect, startup flow cytometry company, trying to make an impact into the CAR T market in flow cytometry. And that's where I'm at today. I have a poster in the other room if you want to see. Drop by, I can talk to you about it. We don't have a booth here, but I'm happy to talk about the company. So that's where I am today.
Thank you, Mark. And my name is Thane Mittelstead, and I've been in the flow cytometry field for, say, 14 years, I think. First started at a CRO up in Everett, actually the same one that Lynette worked at initially, called SMBL USA, now Alta Sciences. Then from there, I moved over to the UW core facility under Michelle Black, where I worked there for about 5 years as well. Then from the core facility, moved on to become a field service engineer for BD, servicing all the flow cytometers in the Seattle area. And then I am now currently back at the UW as director of the flow core facility there. So as you see through all the panel, there's a wide gamut of experience, and collectively probably over 120 years of experience in flow. So please ask away for any questions that you have, because there's a wealth of experience and knowledge here to help you guys succeed in the field of flow cytometry. And if you have any questions, please come up, can line up at the mics, or if you don't want to do that, just raise your hand and we'll find you in the crowd. So either one. So. Oh.
There it is. Thanks. This is kind of just like a general question, I guess, based on a conversation I had at Cyto last year. Do you find that there is difficulty
I could take a stab at it. I'll give my rendition of it. Actually, there are not too many flow cytometry courses, right, available, and at the college level. But there is one, I believe, here at Shoreline. There is a course taught. So if you look at Shoreline, look up flow cytometry, you'll see it. I forget the fellow who runs it, but that's the only one I know of here. Now, I think one of the reasons is flow cytometers are expensive, right? So a lot of universities can't afford to buy it. They need to maintain it, need to buy antibodies and support it, right? So it can be expensive. That's probably one of the drawbacks to it. So I'd say anything you can do to get out there and volunteer to learn it. When I started, I just picked up a book and started reading. So that when I got into have a chance to talk to somebody, I could mention a few words. It made sense, and they said, well, maybe you'd like to work here. So that worked. Um,
Yeah, I have experience with that too. So, running a company, a lot of times, we have a hard time trying to find really experienced flow cytometrists to come in and, hire for this type of position. So what we have found that works really great is actually to hire just out of school, graduated from the universities. They have good, science backgrounds. They've done, a bunch of scientific stuff within their college work, and we bring them in as what we call them QC specialists. And so what we do, we train them how to start up the instruments. We train them how to run the QC, ultra beads and, all the instruments, they help with that. Sometimes they help with making buffer. But the one thing that they do routinely is they QC all the paper that is generated from the laboratory, what the technicians are actually producing in the lab. So not only they get an idea of all the antibody panels that are being created, they get an idea of the processes that are being done. They're starting to understand how to set up instruments. They're getting and reading all of our SOPs. And then as the time progresses, we have somebody that's, moving on, to something else. And these people, they move right up in our facility, go into a research assistant. Or we have two paths that they can take. They can go into being a research assistant, to go into a laboratory, start actually, hands-on flow cytometry, or they go into a study coordinator position, which actually gets to look at the data more, does the table generations. They actually talk to clients more, look at specimen reconciliations, and they do a lot more of that route, talking to clients and doing more data versus going into the lab and actually running instruments.
How did you guys first hear about it? Because for me, I just kind of fell into it. Working at the CRO, I started in the husbandry area in the small animal, and I didn't even know what flow cytometry was until I moved over into the lab and first got introduced to it, running with calibers and cantos, so a bunch of older equipment. So yeah, how did you guys first hear about it? Was it in industry? Was it in academia?
So at that time, flow cytometry was like this uber difficult and exclusive technique. But I was lucky enough to be in a well-funded lab, so we actually had our own BD instrumentation. That's when I got my first exposure to that and to working with an application scientist. But I mean, looking back, I was a newbie. I really didn't know much at all. And I think that's the thing with flow, you'll be continuously learning. Yeah.
Oh yeah, definitely.
Yeah, sorry, my voice doesn't project. I fell into it as well, like you did. My first job out of college was at the HVTN, and where flow was the main readout. So I was forced to learn, and I just fell in love with it. Worked with flow cores here, and got familiar with the staff, the technology, and that just grew from there.
So I just wanted to add one more thing for jobs, looking for jobs in flow cytometry. I mean, now we rely on social networks and so forth, LinkedIn to find positions and so forth. It used to be we'd look at Science Magazine. Job I got at Genentech was a half-page ad in Science Magazine for a flow cytometry operator. Wow, that's changed, right?
Yeah, that doesn't exist anymore.
1989 or something like that. But they're around. The thing is, it's a very specialized skill, and it's not very common that you find people. It's very hard to find good people that know flow cytometry really well. So if you have those skill sets, you don't have to have everything. You have to be an expert in sorting. But if you know the concepts of flow cytometry and analysis, CD markers and so forth, you'll get a job and it'll pay well. Even at the basic research level, which typically doesn't pay that much. But from there, you can go on. You can go into the commercial side. If you have a tendency to like the mechanics, the hardware, and so forth, there's plenty of jobs. If you like the software, the interpretation, there's plenty of jobs, both in research or in the private companies out there.
Yeah, I too didn't, I had no idea what flow cytometry was when I graduated from college. And I actually, and the one side that we really haven't mentioned is more the clinical, the diagnostic world. Is that I got a job at the Puget Sound Breast Center, it was called at the time, now it's Bloodworks Northwest, doing cross matching for organ donation. And so we just had our standard routine, you know, kind of diagnostic tests that we had to do, and one of them was cross matching with flow cytometry. But, you know, we had somebody special that was running, that set up all the assays and did the compensation and did everything. So I didn't even know what any of those terms were. But we were coming in and learning, and we just, you know, stick samples on. And I was like, you know, this is a really interesting technology, and I really want to go into research. And so that's when I moved up to SMBL, and we had the calibers, and you know, you can't get away with you not doing compensation. So that's when I really learned, you know, what compensation was, what it meant. And then we ended up getting at Cantos and you know, I was doing all the validations of all their non-human primate work at that time. And again, like I said, I just saw how important that flow cytometry was. You know, I took it on myself too to learn what it was and the technology. I didn't just let people, you know, just one of those people that went into the lab and just ran my assay and went home for the day. You know, I really wanted to understand it and know what its usefulness was. And so, you know, that's what I ended up, you know, starting a business. When you start a business, you know, what do? You know, I had all these like ideas. And my husband always told me, do what you love. And so I started a flow cytometry lab. Sorry. That's just me, I'm a muscle person, but anyways, so, you know, I knew how important it was, and, you know, started Jennifer, and I started, you know, our own company. And, you know, we actually use interns too, going back to the other question is, there was interns at community college in Tacoma. At first, you know, we were bringing them in and helping them, and they actually helped us make a poster. So they helped us put a poster together for one of the conferences that we went to starting out. So, and, you know, every once in a while we'd have them like come back in and, you know, kind of just help us out. And, you know, they just got the, you know, more familiar with the technology. And, you know, I didn't mention this, but we did sell our company like last month. So we actually are not flow contract site anymore. We're a translational drug development. So you'll see our booth in there kind of has both of the logos. So it's a big transition right now. But, you know, it's been fun. I love flow cytometry, and I hope to continue to do it for the next few until I retire. Yeah.
Yes.
Okay, I don't know if you can hear me. Basically, do you guys have any advice for somebody who is a little bit further on in their career, maybe a postdoc or finished with a PhD, and then kind of have a background in academia, but somebody who's looking to transition more into an industry role?
I think a good way to make that switch would be to look for a company that does the type of research that you do, where you can really bring your knowledge to that company. Companies are always evolving, where their research is changing. Indications are changing very quickly. That's in my experience in the industry, where you can be working on T cells for 2 years, and then within a couple weeks notice, you're all of a sudden now supposed to be an NK professional expert. And so you know, there's and no one at the company knows anything about NKs. So there's that happens. Things like that happens all the time. And so I think reaching out, going to conferences, and networking with industry and people in industry. There's a lot of there organizations. I think Washington State has a biotech organization that has postings, has a lot of different companies part of it. So going to those functions and learning about the companies.
When you talk to people about your PhD dissertation, think about it like at a high level. Don't get into the nitty gritty of the bazillion molecule pathways and all those things. Just share your story and then try to find that link to how that would translate well in an industry setting. So from a resume perspective, keep it simple, to the point, highlight your technical skills, and all those transferable skills are super relevant because one of them is being able to be flexible and adaptable. And that goes beyond the bench experience you may have.
I want to second that. I think when candidates give their talk at their interviews, I think having the component of how I can fit in your research or how I can fit in your type of work is very helpful.
One comment that I do have, like from the contract research side, organization side, is when we hire, PhD postdocs, we kind of caution them with going into a contract research organization because we're doing other people's research. We're not doing our own. And sometimes there's a misconception when people come in to our group that they're going to have their own projects. They're going to be able to go into the lab anytime they want and kind of set things up. And that's not really the case at a contract research organization. So I just caution people, if a contract, you have to have a special person to come into contract research organizations because you're going to be working for somebody else. And a lot of times we don't even have any idea what they're doing. They send in samples, blinded, we go into the lab, we process them, and we give them the results. And if we see something weird and we're like, I wonder what's going on. And we don't get any of the information. We don't know what type of science people are doing. It's like talk today, you were taking out all the markers and taking out all the information. It's like, that's what we get on a constant basis. So, I mean, if you're really looking for an industry like type work, going into maybe a pharmaceutical company and then understanding, getting to know the actual like science of it might be a better fit than maybe like a contract research organization.
Yeah, and I would add that networking with local specialists really help. I think most people would be happy to spend 30 minutes just laying out everything that is true about the role. And you may find, okay, you know what, that doesn't really vibe with me. And, you don't waste your time going down that path.
Yeah, I'd like to add, get to know your like reagent reps and if you are at a core facility or if you run into like your service engineers, because they are traveling all over in the area. So they know pretty much everybody in the industry. So they're good resources to ask if you are looking for a change. Like, hey, looking for something, is there anything out there? And you can be specific or whatnot. So I think, as it's been reiterated several times, like networking, knowing who is in the industry really helps with trying to make that leap. It's turning off. There might be
This one's.
I was going to ask for people who've been in different roles within flow cytometry in the industry, sort of what made you switch and go into a different role? Like what was happening in your current role that made you want to branch out or change directions?
Well, for me, initially, I was laid off at the CRO, so kind of made the decision for me there. Then after working at the core facility, it's an interesting job. It's not very dynamic, and there's a lot of downtime. So, and there's not a lot of upward mobility. So after getting a good experience there, I transitioned into the field service side of things where I continued on. So it was more of a impetus of wanting to learn more and get into just do something different. And then returning back to as a director was a different skill and a different something to learn as well. So that was kind of my impetus for moving into different jobs.
You know, I just comment that the move from academic into private industry is a big move for profit all of a sudden. And your job is sometimes, you think of it in terms of 6-month periods of time. Like we've all been through that. But if you're a people person, if you like to talk to people, if you like to talk science, I think the commercial area and doing sales or support, application support is a really good way to go. Perfect. You get to travel. Maybe not everybody wants to travel. Some people do. You get to travel all over the world, over the United States. So there are opportunities there. But the academic environment offers also long-term career opportunities in flow cytometry. You can be a core manager in an academic facility for many years. You do a good job. It's as fast-paced as can be a commercial role.
I don't think it's working. Does it help any? I don't think
That might help. Sorry.
So there's a lot of ways to go. I'd say just know yourself. Do you like to talk with people? Do you like to communicate? Do you like to work with people all the time? Or do you like working in more of a laboratory environment, okay, which is a little bit insular? You have to know yourself. Then you'll do well. These things. I can hand you this, it doesn't work.
I think hello. For me, I was on the research side for a very long time and wanted a different setting, basically. And a lot of companies will allow for you to move within different functions. And luckily, especially at a startup, that's very where you have to wear many hats. That's very easy to do, to switch to a different group. And that's what I did. I switched to the translational sciences group because I wanted to be closer to the clinic side and just learn more new things, new regulations, and what is needed from the FDA, and more so being closer to the clinic so that you can actually see the patient impact, being closer to that.
Thank you. For me, when I was doing my postdoc, I already knew I didn't want to continue on that path. But then I had the option to go into a scientist at a biotech or go more into the commercial side. And at that time, I had an amazing experience working with the 10X FAS. And I was like, wow, she's showing me how fun this application scientist job can be. Very interactive, but it's still, very technical and scientific. And so then that's when the opportunity came up with the flow cytometry team with Miltani. And then I was on that role in that role for 2 years. And there is a tremendous amount of travel that goes with it. It's fun in the beginning, but then personal circumstances, and I'm like, okay, I'm done with this. And then the sales role opened up, and I decided, okay, well, that gives me more flexibility, less travel. I thought that's not necessarily the case, but you learn other skills as well. So I think I had in my mind that preconception or a little bit of prejudice against like a sales rep, but it's really quite a role if you are creative, if you want to really expand on your business skills, scientific skills as well. So I'm actually surprised at how fun it can be.
Yeah, so working at the Puget Sound Blood Center, I did the same thing every day when I came in. I would either do flow, I would do PBMC prep, I would do PCR, or I would do a cytotoxicity assay. And honestly, it got very boring. I was so bored that I just said, I can't do this anymore. I need to go into research. And then I changed, went to SMBL, and ever since then I have not been bored.
There's a question up there.
Yeah, I just had a quick question. It's kind of for you, but it's one thing to be able to operate a flow cytometer, design a panel, run it, analyze data, but I feel like that's a very different skill set than being a service engineer, right? Being able to operate one of these instruments with the cover open. So I'm just kind of curious, like how you made that transition and learned that skill set.
Oh, it was relatively, it was not difficult. It was challenging, I should say, because I did have some hands-on like troubleshooting already. Actually, I learned from Michelle. She was quite the teacher. I learned most of my flow experience from Michelle here. The biggest thing for me was all the mechanical side, like voltmeter, pressure gauge, height gauge, oscilloscope, things like that. That was difficult for me. I didn't come from an engineering background, which most field service engineers have. Actually, most are like ex-Navy since they are heavily into the engineering side of things. What really helped me was the science side of things, like the software, especially with the BD instruments. So learning Diva was a huge plus for me. The initial training I went through was not fun, to say the least. It was a pretty arduous 2 weeks, and if I did not have the software experience, I don't think I would have made it, because it was just kind of, here you go and have fun type of thing. So you had to learn the software as well as the internal workings of the instruments to kind of make it as the service engineer. And it also helps with the biology side of things, because it's not always the instrument that is the problem. I've had to point that out to a few people. Good question. Thank you. Yes. Oh. Yeah. Sorry. I'll come back to you.
Okay, I'm a little scared of speaking in front of people, so if my voice shivers, I'm sorry. So kind of jumping back to the initial, like the first question, I don't really have a background in science. I majored in accounting and realized it wasn't my thing. I'm now a veterinary technician, and with all this flow thing, it's just really interesting, and it's something that I may want to kind of pursue in the future. So in the case of someone that doesn't have like a BA or BS in biology, chemistry, that kind of sciencey field, what's the best way or best route on top of the connections and all of the things that you guys already had mentioned?
Good question.
That's a tough one. I'm trying to think. Veterinary science is one area that could benefit from flow cytometry. There's plenty of animal antibodies out there. So, but it's an expensive process. But I don't know how many of you paid to have your dog's teeth cleaned. It's also expensive. And so it's changing. So I would say that veterinary sciences certainly could benefit from that. But from your question, I'd say you have the academic background at a certain degree, right? Accounting and so forth. But you're going to have to know some biology. You have to know some biology.
You could do the 2-year BA program. That's fine.
Ah,
That
yes.
was 2
Yeah.
years, and that would get
Great
you all the
suggestion.
background courses that you do. And they offer a flow cytometry.
Yeah, we have a communication major that went through that course and 2 years. And so he's actually a senior research associate. So he's been in the flow cytometry for over 20 years now, but way back when, yeah, he did that. So he graduated in something not science related and took that program at Shoreline.
Yeah, I was going to say the same thing. I also know of a couple of people who went through the Shoreline program. I was also going to mention internships at companies that would use flow. So any cell therapy or biologics company.
Yeah, I think internships for sure, but also if you also like some self-learning, there are incredible YouTube videos and flow cytometry bloggers. The flow director at the University of Alberta, for example, Aja, her videos are very, very clear and easy to follow. Miltani has a bunch on YouTube as well. So if you want to just get a head start and do some self-learning, that's a route I would recommend.
Is this working? Yeah.
Yes.
So this question is more for Thane. I feel like I'm following your exact footsteps where I also was at Alta Sciences, and now I'm at the core working under Michelle, and she teaching me literally everything. But I'm still trying to figure out what to do after this. And so for you, what was your you can't get rid of me yet. Yeah.
You mean way down the line?
Yeah, way down the line. Because there's so much more to learn in the core under Michelle. But once I reached that threshold, how did you make that decision to join BD or decide what branches were after working at a core?
My decision was I learned the experimental side of things and I found it interesting, but I wanted to learn more on the instrument side of things. So that's what kind of drove me to be on the field service side. And I figured I could leverage my experimental knowledge into the engineering side of things, because as I said before, it's not always the instrument that is the problem. So having that background really benefits both of those things. And then I think you could take that anywhere afterwards. So if you have like all that information, all that knowledge, you could be an application specialist, you could go into sales, you could pretty much go wherever afterward with that type of background. And like working at a core lab is a great way of kind of getting that knowledge. Either if you don't know it, if you're coming fresh, don't know anything, yeah, core lab is a great place to learn. So, thank you.
One, Eric's not allowed to leave yet. He has to stay. But I also want to say that as a core director, and I've had a lot of people work under me. And one thing I always encourage is people to continue to learn and to continue to grow. And if you can do that within the space that you're in, then you stay there. But eventually you may exhaust what that position can offer you. So anyone who's ever worked for me, I will always encourage them to continue to grow. And if that means leaving, they may leave. One thing too is that the fact is that especially for people who work in core labs or just getting started, getting in a core lab, you are a hot commodity. If you have good skills and experience, the world really is your oyster. There are positions all over the world that you can apply for. So don't forget that, this is an opportunity to just have an ability to live overseas or do other things. But yeah, for right now, don't go anywhere.
Thank you, Michelle. I was going to say something. Can't quite remember. I forgot. I was going to add something to that, but I completely forgot. Sorry. If I remember it, I'll come find you later. Yes.
I would just add that also if you love flow cytometry, but you still want to work remotely, there are technical support positions that you could, take an advantage of. You're still going to be a scientist, but behind a screen or over the phone. So yeah, you can live in Spain and work remotely.
Yeah, I remember. So it depends on the environment that you're working in, but if you have a good boss or a good manager, they should want to support you, like Michelle said, into wherever you want to go. And if it's not where you are currently working, they should be okay with helping you find that position to where you are going to excel or where you want to be in your career. That's what I've told my tech, and I didn't quite I wasn't comfortable with it initially when I first started working, starting to look for a position when I was working for Michelle. But it was later where I asked her about it and I told her this is what I'm looking for. And she was very supportive of me doing that and was helpful in having me get that position. So look for those people that will help you. Lost my screen. So we have a few more minutes. Is there any last minute questions? Or we can start. Oh, there's one. It is. Or I don't.
I'm curious as research sort of becomes increasingly interdisciplinary, sometimes it feels, to what extent you see it being valuable to be a really deep expert in flow cytometry versus having proficiency with a number of tools that you're able to integrate. Although I'm sure that's also a very research context specific question. Pass this off to.
Yeah, I would say both. There's some there's plenty of scientists who have no flow background and learn it at wherever they're at, whereas there's another person who is the flow expert for the entire company. So there's absolutely need for both. So it's not a definite answer.
I think that's a great question because it's true. It's become more interdisciplinary. One thing I've seen now recently is, as a flow cytometry expert, you're also expected to know some genomics because intracellular staining and markers is becoming a bigger demand with gene therapy. So we know the CD markers, great. Guess what? Now you can start on the intracellular markers. And you can learn those, right? So that molecular biology is also a good skill to have. PCR, how that's going to help you, right? Identify what's the next sequence. If you take something that you've sorted out in flow cytometry, cell population, you've sorted out, now you need to study the genomics. You need to know molecular biology and genomic sciences as well. So that's a good, trio of disciplines to focus on.
Yeah, definitely being up to date on the hottest trends. Like spatial biology right now is a big topic, so you have to be aware of what are the expectations and where you can take the field with that technology. But it's interesting that flow, it is still a critical part of any workflow. But if you know your downstream single cell or single nuclei sequencing, for example, that certainly helps with troubleshooting your upstream cell sorting process, for example. So yeah, flow is really pretty in that sense. I think people will always need your skills.
I agree that the need for flow cytometry is still there. I even see that in the contract world, the people are always coming to us. They have projects that they need flow cytometry on, and I don't see it slowing down. And we try to keep up with the technology, a little bit harder for a research organization. A lot of the validations and the FDA regulations and everything that we have to go through. But I still think the technology is, it's growing and we're not slowing down. So.
Yes. So that's the end of this section. So thank you so much for attending, and thank you for all your excellent questions. And again, a round of applause for the panel here. And now we are on break.
Thank you, everybody, for sticking around through the afternoon. My name is Catherine Schwedhelm. I'm a laboratory research manager at the HIV Vaccine Trials Network Endpoints Laboratory here at Fred Hutch. And today I'm going to be talking about our approach to flow cytometer standardization and characterization, as well as how we have validated an assay for vaccine clinical trial testing. So just a brief summary of the topics that I'm going to cover today. First, just an overview of what the HIV Vaccine Trials Network and COVID-19 prevention networks are, and the intracellular cytokine staining assay that we use. our approach to flow cytometer characterization and standardization, how we validated intercellular cytokine staining assay for clinical trials, and then just finish up with a summary. So the HIV Vaccine Trials Network was established in 2000 by the NIH, and it's a large network of domestic and international clinical sites and laboratories. There are several immune monitoring sites that are part of the HVTN. They were initially established here at the headquarters of the HVTN at Fred Hutch in Seattle, and then at Duke University. And then in 2012, the HVTN added sites in Cape Town and Johannesburg, South Africa, to support clinical trials that were being run in sub-Saharan Africa. The Seattle and Cape Town labs use the intracellular cytokine staining assay to monitor T-cell responses to vaccine candidates, and reliable and high-quality data to support these vaccine candidates require high-quality samples and rigorously quality-controlled instruments. The COVID-19 Prevention Network was founded by the NIH in 2020, and it took advantage of existing expertise in federally funded vaccine and prevention networks, including the HVTN. So the immune monitoring labs require cryopreserved PBMC samples for testing. And early on, the HVTN realized that high quality samples are required for robust and reproducible results. And they put a lot of effort into optimizing and standardizing processing procedures, including establishing a network of processing labs near the clinical sites. And in the figure, I'm showing the ability of the HVTN processing sites to obtain high quality samples. So different sites of the HVTN are on the x-axis, and then viability after thaw is on the y-axis. And overall, the HVTN does get very good high quality samples throughout the sites, although there are some that do perform not as good as the others. We also work with collaborators, and sometimes, and then when we work with these collaborators, we have no control over PBMC quality. And sometimes we do experience poor quality samples, and that really hampers our ability to give our collaborators high quality data. And sample quality impacts the cytokine responses in the ICS assay. And here I'm showing the impact of sample quality on cytokine responses to CMV stimulation. So we took a random sampling of 12 vials from a leukapheresis and thawed them and used them in our ICS assay. On the right axis of both plots is viability in the assay. And then on the y-axis on the left side of the plot is the cytokine response for interferon gamma IL-2. And then the left plot is CD4 T cells and the right side is CD8 T cells. The sample quality from this leukapheresis was inconsistent. We had viabilities ranging from 80% down to 40%. And the response to stimulation directly correlated with sample quality, with lower responses seen from the low viable samples. So I've talked about the ICS assay. What is it? We use it to monitor cellular immune responses to candidate vaccines using flow cytometry as the readout. In our lab, it takes place over 3 days. On the first day, we thaw cryopreserved PBMC, and then we rest them overnight to recover their function. Then on the second day, we stimulate the cells with a peptide pool or other antigen of interest in the presence of Golgi stop so that the cytokines are not excreted from the cell. We stimulate them for 6 hours or longer, depending on the assay that we're performing. And then on the third day, we surface stain the cells, then we lyse, fix, and permeabilize them to allow access to the inside of the cell. And then we perform intracellular cytokine staining for the cytokines, as well as other markers of interest. Then we acquire the samples on the flow cytometer and analyze the data. So moving on to how we characterize and standardize our instruments. I do want to note that the HVTN uses BD instruments. Currently, we have 4 Symphony A5s and 1 spectral instrument. And the procedure that we use for the quality assurance program was developed by our colleagues at the Vaccine Research Center. So all of our instruments are rigorously quality controlled. Prior to implementing any instrument in the lab, it undergoes a characterization and optimization process, which includes background testing to measure electronic and optical noise, a voltage scan to determine the optimal voltage ranges for each detector, and then we address any issues that we do find. Then finally, we move on to standardization, where we match fluorescence intensity of stained cells across detectors for all of our instruments, and we establish QC bead targets. And then prior to each assay run, we perform a QC procedure. We track robust CVs and voltage changes over time, and then periodically we assess standardization and then rematch targets if there has been any drift. So background noise comes from two sources, electronic and optical. Increased background results in decreased sensitivity, which may lead to decreased ability to measure low intensity markers. And the measurement of this background noise is done with a device called the QuantiFlash that produces consistent, precise, and uniform light pulses. And we are able to measure electronic noise with this device with the lasers off. And then when we turn the lasers on, we're able to get a measurement of both the electronic noise and the optical noise. So here I'm showing the results of background testing on one of our instruments. The different detectors of the instrument are listed on the x-axis, and then the background is on the y-axis. The red dots are the background, the measured background with the lasers off, so just the electronic noise. And overall, we have very low electronic noise in all of the detectors. And then the blue dots represent the electronic plus optical noise with the lasers on. And again, overall, very low background except for two detectors. The first one is the G660 detector in the green box, and this is expected as this is the wavelength for Raman scattering. And then the other one, the V450 detector, which we tested with the 450/50 bandpass filter, had unexpectedly high background, and this was investigated further. After the background testing, we perform a voltage scan. So for each detector, we stain cells with CD4 conjugated to the fluorochrome of interest, and then we acquire them in voltage increments. Then we calculate the stain index at each voltage increment for each detector. And we use, to calculate the stain index, we're taking into account the median of the positive population, the median of the negative population, as well as the spread of the negative population. Then we plot this stain index versus the voltage for each detector. And then we obtain a filtration curve, which I'm showing in plots on the bottom right. Then the optimal voltage is the one where the stain index is maximal or close to maximal, but where the median and the robust standard deviation of the negative population remains low. The results of the filtration can reveal additional issues that may not be apparent from the background testing. And here I've listed three potential issues, how to identify them, and means for addressing them. So if you have a lower stain index than expected or compared to another instrument, or you saw high background in optical testing, this could be a result of a faulty filter set, either a bad long pass mirror or a band pass filter. You can test this by iteratively moving each filter set down the array, performing a cascade test. You can swap filter sets with another instrument or a similar detector. And then in the right, I'm showing the filtration curve for the V450 detector, where the brilliant violet 421 fluorochrome is measured. So the red curve represents the filtration curve with the old mirror, and this has a much lower stain index than you would expect for a very bright fluorochrome. So we performed some testing and found that the issue was actually a long pass mirror further up the cascade. We replaced that mirror and then redid the filtration and ended up with this new filtration curve in green, where the stain index more than tripled and became more like what you would expect from that fluorochrome. If you have an abnormal filtration curve, or where the optimal voltage is very high and it's not due to any filter issues, this is most likely a poorly performing PMT, and then you can call in your engineer and have them change the PMT, and you may have to go through several iterations of PMTs to find the best one for that detector. And then on the right in the plot, I'm showing a filtration curve in red for the R660 detector of the old PMT. So this curve is shifted far to the right. It has a saturating stain index at a very high voltage, 750 volts. When that PMT was replaced, that curve shifted to the left. It's the green one now. The stain index stayed about the same, but the maximal stain index occurred at a much more reasonable voltage of about 600 volts. If you see for all detectors for a given laser, much lower stain index than expected, this could be due to diminished laser power. And then you can have your engineer measure the laser power at the flow cell and compare it to the laser output power. And we've had this happen to one of our instruments in Cape Town. After any issues have been addressed, then the optimal voltage and the optimal voltage is chosen, then the target medians for the QC particles are determined. This allows for daily standardization of the cytometer to its optimal settings. So what we do is we run QC beads at the optimal settings. Our choice of beads are a mixture of 6-peak rainbow beads and super beads. The super beads are a little bit brighter than the 6-peak bead. We run these beads at the optimal voltage for each detector, and we record the MFI for both bead type, and then we choose the optimal bead for each detector. And we want to choose the brightest bead for the detector, but where it's staying within the linear range of the detector. And then that associated bead MFI becomes the daily target moving forward. So in the table on the bottom left, I'm showing the 3 detectors off the red laser. I'm showing the bead MFIs for both the 6 and Supra, and then the red boxes are circling the bead that's most appropriate for that detector. And then the bead target that we establish, I just round up to the nearest 1,000. And then in the bottom right, I'm just showing a representative example of what our QC template looks like on Diva. So the targets are at the bottom. I have 3 plots, 1 for each detector. And then we put those, we run those beads on the instrument, and then we individually adjust the voltage of each detector so that the MFI of the bead today matches their established MFI. We can also standardize across multiple instruments, provided that they've all been characterized as previously described. So we select one instrument as the reference, and then we QC that instrument to its optimal setting using the 6 and Supra beads. Then we run single stain CD4 cells at the QC voltage for each detector and record the median of the positive population. Then we take those cell medians and cells and move to the instrument to be standardized, and one by one, match the median of the positive population for each detector. Then we can go back to the filtration curve for that newly standardized instrument and just make sure that it's within the optimal range for that instrument. And then once we're happy with that, then we, on the newly standardized instrument, we run the QC beads to determine the targets. And I want to point out that the 6 and Super targets will be unique to each instrument. You can't standardize the instrument with these hard dyed beads. You have to do cells. And I've also done this with stained beads, and I found that they didn't give quite the same results as stained cells. So our preference is to always use stained cells. And then if you have additional instruments, this can be repeated. So in the table at the top, I'm showing 4 standardized instruments, reference instrument, and 3 additional ones. In the rightmost column, I'm showing the CD4 MFI, and they agree very, very closely to each other. But then in the column just to the left of that, in the red boxes, I'm showing the MFI of the Supra bead, and they're different between all the cytometers. So this is just illustrating that even though the cytometers are standardized to stain cells in your population, your cell population will be in the same place for each instrument, your bead MFI will be different for each instrument. And then on the bottom, I'm just showing the result of standardization between 5 of our instruments. We have 3 symphonies and 2 LSR2s. We now have 4 symphonies and 1 spectral. So this is a little bit old, but this is what it looks like when you have standardized instruments. So then to verify the results of the characterization and standardization process, we perform an additional assessment using a validated ICS assay using samples for which we have extensive historical data. So we bulk stimulate and stain 3 samples, and then we split these samples into replicates and a set of plates for each instrument. And we bulk stain and stimulate because we want to make sure that any difference that we see is due to instruments and not difference in assay technique. Then we compare the mean primary cytokine readout to the historical data that we've accumulated, as well as the mean cytokine response between instruments. And we're wanting our mean cytokine response to fall within plus or minus 3 standard deviations of the historical data. And then also we want the CVs of the mean cytokine response between all cytometers to be less than 30%. And if we fulfill both those requirements, then we say that we've validated our instrument. And the end result of this standardization is that we can use instruments interchangeably, and we can use the same gates in our analysis templates, and we've even done this successfully across labs. So just to summarize our quality assurance procedure, first we perform background testing and a voltage scan, and then we address any issues that are found. Then if we're standardizing multiple instruments, we select a reference one. If we're just like standardizing one instrument to its optimal settings, we just pick the optimal voltage for each detector. Then we match MFIs across all instruments. If we are standardizing multiple of them, we ensure that the voltage is within the optimal range for the PMT for all the instruments. And then finally, we run the 6-peak and super beads to establish the QC targets for the optimal settings. Then we cross-validate the cytometers using our ICS assay to verify the standardization. And then prior to any assay that we run on the instruments, we perform daily QC to these targets, and we monitor the performance over time. There are many benefits to standardizing instruments. We can use all of our instruments interchangeably, even those at different laboratories. So we can run the same assay here in Seattle in our instruments or in Cape Town, and we can be sure that they're going to look the same in both sites. We establish gating templates at the start of clinical testing, and they're not changed. This reduces potential bias. Populations will typically always fall in the same place because the MFI of expression is similar between participants. There are some exceptions like perforin and granzyme, and for those we do adjust on a per sample basis, but within strict criteria. On occasion, we may need to do slight adjustments to parent gates, such as for defining our lymphocytes, but our functional gates are rarely adjusted. Standardization can assist in troubleshooting when unexpected results are obtained. We know that nothing is wrong with our instrument, so we can go straight to looking into potential technical issues. And this also allows us to pick up any potential issues with instruments well before they may become a problem in our assay. And we've also been able to establish procedures for efficient data pipelines. So we have instrument templates that we can use across each instrument. We make one, we can use it across all of them, and they have standardized keywords that only requires minimal data entry. And then changing subjects, I'm now going to talk about how we validated our ICS assay for a vaccine clinical trial. So at the start of the COVID-19 pandemic, we developed a 27-color panel for the sensitive detection of CD4, Th1, and Th2 cytokines, as well as CD8 T cell responses using our expertise in developing high-parameter panels for HIV research. The goal of this panel was to maintain the sensitive detection of CD8 T cells and Th1 cytokines, but also optimize the detection of the low-frequency Th2 cytokines, which were of particular interest in this context due to the concerns of Th2 skewing. For the Th2 cytokines, we used the brilliant blue fluors to improve sensitivity. So we were pairing low frequency dim cytokines with bright markers. And then we also included IL-5 and IL-13 on the same detector to maximize detection. And then we also included other markers, such as those for Treg identification, activation, cytotoxic potential, Fc gamma receptors, and Th classification. The primary readout of the assay developed for COVID-19 vaccine research is interferon gamma and/or IL-2 for CD4 Th1 responses and CD8 T cell responses, and IL-4 or IL-5 or IL-13 cytokine responses to spike-1 and spike-2 peptide stimulation. And this assay allows for the determination of Th1 to Th2 bias. And here I'm showing some representative staining plots of the primary cytokines of interest. The left plot is the Th1 readout, interferon gamma IL-2, and then the middle plot and the right plot are the Th2 with IL-4 and IL-5 or IL-13. And as we would hope, the response to Th1 is robust and frequent in relation to the response to the Th2 cytokines. And this is just an overview of the final markers that went into this 27 color panel. The key markers that were about the key critical markers are in bold. So these are the T cell markers and the cytokines. And these are the markers that we validated. And then the other markers were either verified or standardized. And then we've also, based on this panel, developed a next generation one that substituted in CXCR5, PD1, and IL-21 to be able to detect circulating T follicular helper cells. So we have a very methodic approach to assay qualification and validation. We perform extensive optimization and standardization, and this includes both of our cytometers, which I previously talked about, as well as optimization of our assay. And we do many iterations of the panel to make sure that we have resolution for the markers that we're interested in. We perform fluorescence -1 controls, and then we only move towards validating that assay when we're really happy with it. We base the qualification and validation on 2 sets of guidelines: the FDA guidance for industry bioanalytical method validation and the International Conference on Harmonization Validation of Analytical Procedures. And we performed actually 2 validations on this panel. The first one was in 2020 using PBMC from HIV vaccine recipients because we didn't have any SARS-CoV-2 suitable samples. When we submitted the data to the FDA, we received feedback from them that basically required us to repeat the validation using spike-specific responses. And they said that responses induced by HIV vaccines were not a suitable surrogate to SARS-CoV-2 specific infection or vaccine-induced responses. And this goes back to the objective of a validation of an analytical procedure, which is to demonstrate that it is suitable for its intended purpose. So we had to adopt some of the guidance documents to be able to validate this assay. And there were some challenges for that because this is a functional flow cytometric assay and not a bioanalytical one. Each sample contains its own negative control. There's no gold standards, there's no calibration or reference standards. The results of the assay are not based on a curve or compared to any knowns. In the ICS assay, the analyte of interest is an antigen-specific T cell and one that expresses a specific cytokine or specific cytokines. We compared the new version of the assay to a previously validated version that serves as the reference, and we use the concordance correlation coefficient as it assesses both correlation and slope. For these high parameter flow cytometry panels, the assay measures many markers and not all of them can be validated. So we need to identify key functional markers to validate, either individually or in combination, such as interferon gamma and IL-2. Then we have secondary markers that we consider for qualification, and then all others are considered standardized. And there's some other important considerations to take into account. One is that you need to establish before the validation your acceptance criteria, and then you need a large number of samples with known responses. To perform this validation, we use peptide pools. These were sequential overlapping peptides that covered the entire SARS-CoV-2 spike amino acid sequence. They were 15 amino acids in length, overlapping by 11 amino acids, and then we divided them into 2 pools to cover the spike 1 and spike 2 subunits. We used 3 validated, optimized, cross-standardized BD-FACS Symphony A5 conventional instruments. And before each assay, we performed daily pre-acquisition QC to ensure that the performance was up to standard and to account for any variability. And this ensured consistent high-quality data over the entire course of the validation, which took multiple months to complete. The negative control for this assay is DMSO, which is the peptide diluent. And we determine positivity by assessing the statistical difference between antigen stimulation and the negative control. And the method that we use at the HVTN is the adjusted Fisher's exact test. So there were 5 parameters assessed in the validation: specificity, precision, sensitivity, linearity, and accuracy. I'm going to talk about each one of them. So specificity assesses the ability of the assay to return a negative result for a known negative. In the initial validation that we performed, we used HIV specific samples, and this was successfully done. However, in the context of SARS-CoV-2, we can't actually directly assess specificity due to cross-reactive responses from endemic beta coronaviruses. But we did perform the testing nevertheless. It required a large number of samples that are presumed to be negative, which were 40 PBMC samples that were collected pre-pandemic. And then we also included 8 PBMC samples from SARS-CoV-2 vaccinated or convalescent individuals for comparison. And then we had the false positive rate acceptance criteria of less than 10%. We did actually see some positive responses. We had 3 that were CD4-TH1-S2 responses that were likely true cross-reactive responses. And then we had 1 positive CD4-S1-TH2 low-level response that was likely a true false positive. But even with these positive responses, we were still below the false positive rate of 10%. And the precision assesses the degree of agreement of the results obtained from repeated testing of the same sample. So we performed 6 independent runs by multiple technicians over different days in triplicate for each sample. And we chose 9 samples that were pre-selected based on their responses to S1 and or S2, so that the range of responses in these samples would cover the range of expected vaccine-induced responses. And then we calculated both the intrasample and intersample intrasample and interassay CVs using the 18 data points that we collected for each sample. And we also used non-background subtracted data to avoid negatives. And the acceptance criteria for this may seem a little bit strange. It's CVs across all runs of less than 35% for 80% of the values. But we chose this because this was based on prior data that indicated that this was the expected behavior of the assay. And the FDA was in agreement with this. So this table shows the precision results for CD4 T cells expressing interferon gamma and or IL-2 for combined S1 and S2 responses on the top, and then S1 and S2 separately below that. And they're ordered in decreasing magnitude of response. So we're looking for the lowest level of response for which the CVs remain less than 35% and where we're still getting positive responses, which is 0.054%. And if you'll notice in the interassay CV column, we actually never exceeded the 35% threshold. So in further experiments that we perform, we actually used lower level responses to try and reach that 35% threshold. We also perform this for CD8 T cells and CD4-TH2 responses. So for CD8 T cells, we maintain CVs less than 35% for responses down to about 0.013%. And then for CD4-TH2 responses, there was lack of suitable responses to SARS-CoV-2, so we had to determine this with HIV peptides and samples. And we maintained our CV requirement for responses down to about 0.05%. Then for the sensitivity validation, this is comprised of 2 components: the lower limit of quantitation, which defines the lowest T cell response for which the CV is less than 30%, and limit of detection defines the lowest response that can be consistently detected. So for LLOQ, we use serial diluted SARS-CoV-2 convalescent samples stimulated with S1, S2, and SEB. And then we use samples from specificity to assess LOD. So the LOD has 2 definitions. There's the analytical definition, which is based on the distribution of the data for the blank, which in this case is the response in the negative condition for each cytokine of interest. And these are the values listed in the green box. So there's actually a value for each cytokine for each sample being assayed. So then you have a lot of different values. But since this is a functional assay and not an analytical one, we also developed a functional definition for LOD, which is the lowest magnitude of response that is determined to be positive by our statistical tests, and these are the values that are in the red box. The magnitude of the functional LOD is typically around 0.05%, which is actually consistent with what we get for our LLOQ and precision results. And we prefer the functional definition because we realize that the background varies by sample, and there's no single value for LOD that can be used across the entire assay. And I do want to point out that the LOD involving CD154 is quite high in comparison. When we were first doing the validation of this assay, we had a way to gate CD154, and then we changed the way that we gated that marker, making the gate crooked to improve the specificity without reducing the sensitivity. And then once we did that, the LOD value for that marker actually fell back in line with what we were seeing for the other cytokines. And here I'm showing the LLOQ for CD4 T cells. And this table is showing the average S1 and S2 responses from the various samples and dilutions, and they're sorted in decreasing order from highest to lowest response. The LLOQ is then determined by looking for the average response for which the inter-assay CV is less than 30%, which again is 0.054%. We also perform this for CD8 T cells and CD4 Th2 responses. We couldn't determine CD8 LLOQ because of a lack of detectable response. And then for CD4 Th2 LLOQ, we had to use HIV seropositive samples and peptides again because of lack of response to SARS-CoV-2 stimulation. So linearity assesses the range of reliable responses, and this was determined for both Th1 and Th2 cytokines. For Th1 cytokines, we were able to use SARS-CoV-2 specific samples, but for Th2, we had to use a mitogen for the stimulation. So all of the stimulated PBMC were serially diluted into unstimulated PBMC, and all the dilution levels contain 3 replicates, and then only data points above LLOQ with CVs less than 30% between the 3 data points were used to determine the linear fit. And here I'm showing the linearity results for CD4 and CD8 T cells in response to S1 and S2 stimulation. So the top plots are CD4s, the bottom plots are CD8s. On the x-axis is the expected response, on the y-axis is the measured response. Response to S1 are the left plots, and the response to S2 are the right plots. And for S1 and S2, CD4 and CD8 T cells, these data points were all above LLOQ, and the linearity was maintained to responses of about 0.01%. And here I'm showing the linearity results for CD4 T cells expressing Th2 cytokines in response to PMA and ionomycin stimulation. Again, like the previous plot, I have IL-4 and CD154. So we're including CD154 here because it increased specificity and sensitivity for this cytokine. And then on the bottom, IL-5 and IL-13, and then 3 individual participants. And again, expected values on the x-axis and measured values on the y-axis. So for IL-4, we were maintaining linearity down to about responses of about 0.05%. And then for IL-5 or IL-13, it was a little bit higher of about 0.1%. And then in the plots, the gray dots are dilution levels with CVs greater than 30%. And these ones were not included in the linear fit, but they're just left in here for illustrative purposes. And then the accuracy validation is performed by comparing the positive responses obtained in the new assay to responses obtained using a previously validated assay with the goal of establishing equivalence between the two. And we establish equivalence if the 95% lower bound of the concordance correlation coefficient is greater than 0.7, and we only use responses that are determined to be positive based on the Fisher's exact test. The primary comparison in this accuracy validation is preferentially used using the positive responses to the stimulation of interest, but we can use results from all samples and or results from SEB in the absence of sufficient data points. And then for this validation, we used interferon gamma and or IL-2 responses for HIV GIG, POLNEF, or CMV. And here I'm showing the results of the accuracy validation. So we combined the interferon gamma and or IL-2 responses for all the peptides. And the responses obtained from our validated 17-color ICS assay are on the x-axis. And then the responses obtained from our new 27-color assay were on the y-axis. And the results show excellent concordance for both CD4 and CD8 T cells, with the concordance correlation coefficient exceeding the 0.7 threshold. So the results of the validation, except for specificity that couldn't be directly assessed, we validated our ICS assay, a biological and complex assay for precision, sensitivity, linearity. And throughout this process, we gained key insights from our interactions with the FDA. We have to have samples that have responses to the pathogen of interest, and the gold standard are samples from the specific context of vaccination. And I also want to add that we are able to apply what we learned from the FDA for this validation for SARS-CoV-2 to other vaccine realms such as malaria, TB, or HIV. The batch controls must have a vaccine or pathogen-specific response. And for our purposes, we actually had to wait until we were able to obtain leukapheresis from donors who had the responses that we needed. If we're wanting to use this assay for phase 3 trials, we're going to have to perform another partial validation using PBMC from participants vaccinated with EUA or licensed vaccines. Other important things to consider was that sample quality is critical. As I mentioned previously, if you have poor quality samples, those poor quality samples are going to impact your results and thus would impact the validation parameters. And we couldn't have done this without our standardized and well-maintained cytometers that produced consistent data over time. Just to finish up, cytometer optimization, characterization, standardization is really critical in the clinical testing realm, but also in the research realm. It's really important to realize that brand new instruments are not immune to deficiencies and manufacturing issues, and it's important to check them out and make sure they're performing as expected before you fold them into your daily use. If you have standardized and optimized your instrument, data from these instruments are reliable and reproducible even over time. And if you have multiple instruments that are standardized to each other, it allows you to use them interchangeably and to increase your throughput. We've shown that we can validate highly complex flow cytometric functional assays and perform them in high throughput, even across laboratories. We validated our 27-color ICS assay for specificity, LOD, LLOQ, precision, linearity, and accuracy. And we gained some very valuable insights from our interactions with the FDA that we can use for future validations. The T-cell data that we produced using this validated assay has been used to support early phase clinical trials and support their authorization in both of these vaccines in both the USA and the European Union. And even if you are working in the research realm and don't want to consider it full validation, you can apply selected aspects of this to qualify your assay. Even in the research realm, it's really important to have high quality and consistent results. And I just want to say thank you to everybody in the HVTN laboratory, in particular Julie and Steve, our collaborators, the statistical support that we receive, and then all of our funders. Thank you.
I have a question. So is any of the data sets that you produced in this study are publicly available?
I believe the ones for the Janssen vaccine are.
Okay. I mean, like the FCS files, not the.
Oh, the FCS files. I don't know. I would have to let you know.
Can I reach out to you later? Thank you. What am I doing?
So just the HDMI.
Right, it's on this side. Nope. Here.
Oh, yeah.
What is this?
That's just power.
Oh, okay, I'll take it.
Yeah,
Thank you.
Need it.
Some juice. All right. Oh, all right, let's say that works. Okay. Oh, cool. All right. Is there a pointer? No, I will make a pointer out of this. Pointer. Laser pointer. Okay. All right. Hello, everybody. My name is Anna Bilkina, and I came here to talk about the gore of bad data. So for those of you who have seen this talk at Cyto last year, I apologize. Most of the slides are same, but the response from the audience can be different. So you're still probably can hear something new. All right. So before I start with this talk, I wanted to give my acknowledgements for everybody who donated samples for this talk. So this talk started, actually was planned by Jennifer Wilshire back like more than 10 years ago. So I inherited the slide deck and I rebuilt it quite dramatically. And things got more complicated since then. So a lot of simple stuff we have to kind of like put on the back burner now that we assume that people know how to do, but still, I'm still using some of that. And then I went to a lot of my friends and asked for examples of what can go wrong with their data. And some of the data in this presentation are also mine. So, but yeah, thank you. And that means that your data can be featured in the next issue of this talk. So I'm absolutely taking submissions. They can be anonymous. We have witness protection system. So absolutely send your stuff. And so that you don't need to take pictures of my slides, the slide deck is available. You can scan the QR code and get a PDF of all slides. And then I'll count how many people downloaded it. So today, I have only 45 minutes and this talk was much longer, so some things will be cut. But we were going to talk about the things that obviously none of you ever commit, right? So you would never do these crimes. It's all education purposes only, right? So we'll start with some more boring stuff and I'll just go quite quickly through that. And then I'll show some more interesting and tricky cases, and we're going to try to figure out what was going on with that. And I will ask the audience, what do you think happened here sometimes? So, or I'll just give you the answer right away if it's simple. Okay, so the reason we're all doing this is because we basically have a reproducibility crisis that has started a long time ago, and it's getting worse and worse and worse. And specifically in cytometry, like we have a lot of, witness data that people are experiencing problems with all steps of the experimental procedures, right? Starting from the planning of the experiment all the way to data analysis, right? And so this was a survey conducted by Beckman Coulter in 2019. And so they basically identified key problems that people have with their reproducibility of the data. So, and it all boils down to like poor choice of settings on their instrumentation, malfunction of the instruments that is not actually like noticed or noticed much later, mistakes in the gating and like simple analysis tools, compromised samples that basically should not be even in the data set, and just generally not following best practices. So majority of problems that we encounter are happening when the experiment is performed. But as I said, sometimes we should start by the sourcing of the samples and reagents and see if things can go wrong there, and obviously the data analysis. Okay, so the boring crimes part of this is all the things that you already know, and I'll just show you some good examples. Again, I put it in the slide deck so that maybe you want to share this with your users. And I know that many of you are actually like running cores or working in the cores or assisting and existing peers in your labs. So a lot of this is generally for educating your colleagues who didn't make it to this talk, right? So if you're here, you probably don't do all this kind of things, right? So the first batch of my cases is like simple cases, I said. So when people do, it's not necessarily like a gating on the histograms, but basically when people start to interpret their data and they think that they checked all the boxes for the proper controls, but they rely on the manufacturer recommended amount of antibody that they added. And I want to say that people who do human immunology are way more guilty with that because it's like 5 microliters per test, right? That's it. So, and then they don't know how to interpret their data. And it gets even worse when you actually sort the samples. So you don't really know what to take with that. Why am I getting so many positive cells? So here in this example, this is a single color CD3 staining. And suddenly they're seeing, oh, there's an intermediate population and everything in the sample is positive, like T cell, looks like T cell, right? So yeah, you need to titrate the antibodies, obviously, right? So you don't want to guess whether it's overstained, it's an awkward sample. And again, like especially with humans, it can be a curveball. It can be an awkward person you're looking at, right? But it also can be the overstaining. So don't guess it, titrate your experiments, titrate your antibodies. So I put an example of how titrations look in a larger panel. Like you can get all different kind of patterns in your titration experiments, and deciding which titer you want to use in your panel, that's a completely different topic, right? But at least like try to titrate, try to make sure that your negative is not accumulating some signal, right? So the antibodies can be bound to the cells non-specifically, so proteins like to stick to each other, so titrate your reagents, right? Also, the non-specific binding of reagents that are titrated properly, right? So this is an example of the sample where we have staining that should be only on the NK cells and some T cells, but it's present on the monocytes. So a lot of this kind of errors, you notice them because you're already trained to notice something in your data. So you kind of like, you know the pattern already. So it's your neural network that already knows how it should look. But imagine that you're doing exploratory panel where you have no expectation, right? So you would never actually find this to be awkward. I know that this is an NK marker, but if I don't know that, that would look exactly like an intermediate population that will actually pass my radar, right? So this is a classical example where we have the antibodies binding on the Fc receptors on the cells and not specifically, it's a specific binding, but not through the variable region of the antibody. So Fc block would actually block Fc receptors and we get our monocytes where they should be, right? So use Fc blocks. It can be a little bit more tricky when you get to the panels that have some other reagents, right? But please, even if you do mouse and human and any other species of the mammals, do Fc blocking. A lot of people whom I know who do human research, they still think that Fc block on human cells, you don't need that. You do, right? So here, this is another example where we have a pattern that looks kind of similar to not having Fc block, but it's actually T cells, right? And the Fc block was used. So again, we're looking at this and it's like, okay, this is an incorrect pattern, right? So we should be able to see the CD4 cells not being intermediate for CD19, right? So what it means is that there's some kind of other unexpected interaction in your master mix, right? So here, in this case, it's the polymer dyes that are actually congregating together. So they're sticky, and that actually happens not on the cell surface, that happens in the master mix before you even add it to the cells. And I mean, this is the polymer dyes, we had them for a while. So kind of at this point, like everybody knows that you need to use this, but we have other new families of dyes arising. So there can be more interesting interactions happening. So investigate these kind of things. Ask the vendor who is promising you amazing results with a new dye family. Is there anything I need to know about the interactions in my master mix with this new reagent, right? So you can be surprised to hear what they say. Okay, so another example, right? So here, this is again about monocytes and macrophages that express Fc receptors, right? But that's not about Fc blocking. So here's an example where we have some intermediate population right here, right, and here. So it happens both with isotype control and with the experiment, but not on every dye in the panel, but only some of them. So this is happening because the myeloid cells can actually non-specifically bind certain fluorochromes, like cyanine dye family and cyanine-like dye family, and you can block it with another concoction that you can get from, it's a proprietary recipe, but you can look up in PubMed what it is. So yeah, but you can block this interaction as well. And so that will basically remove that intermediate population. Here's another example. This is all coming from BioLegend because this is like a monocyte blocker from BioLegend, but BioLegend is not the only vendor that sells you that reagent. So you will see that if you have no blocking or FC block, this is on humans, you will see basically no improvement, but with the monocyte blocker, you will see improvement, right? And that's because this is an example where the on human cells, the FC block may not really do much for your improvement of the signal. You never know which of the antibodies in your 25 color panel is actually badly interacting with the FC receptors in the cell. So don't guess it. But yeah, and I want to say that sometimes this looks, you know, it's not like that drastic. Like for example, here the monocyte blocker was used. This looks like very suspiciously good, right? So it looks like it's not everything is shifted. It's just a little bit. So I would totally buy it for a real signal if I didn't check for that, right? Okay, so make sure you exclude your dead cells when you stain, because if you have dead cells, they like to collect all kind of different signal and debris that could collect the antibody in the whole staining mix. So you can get something, again, like if it's CD4 versus CD8, you know that these are not real, but sometimes people believe that they got something real. So remove dead cells, you will get this awkward double positives away, but sometimes it's not a double positive, it's a single positive, right? So don't skip on viability dyes. Okay, so compensation. So compensation and spectral unmixing, right? So it's exactly the same rules that you need to apply. We've heard about them today in the very first talk. So, you know, like each reference should be as bright or brighter. I mean, I feel like really old when I speak about this. So we've been like hearing the laws of compensation for years and years. But, you know, once you get to, again, like a big panel, you start to like, oh, well, can I just adjust it a little bit if I don't want to redo all these things? Like, yeah, don't do that. So same type of particles, same background for autofluorescence. Use exactly the same fluorochrome for your controls and for your samples, right? Treat them the same way and make sure that statistically get enough events to calculate medians. Because sometimes if your population is, you know, 20 cells, even if they're perfectly okay, like you can't really calculate anything accurately from that. All right, so this is all data. I think this is like says Mario Roder. Okay, so all time. So this is the example where it's not as bright as sample, and actually you can see that once you get over the brightness that was calculated properly, then you start seeing problem with the compensation, right? Another example here. This is where the control was bright enough, but they used a substitute for the tandem dye compensation, and it actually is not exactly the same spectrum, so it actually goes away. So if you use the correct dye, you would have the proper pattern. So you would see that it's symmetrical. So likely this compensation was applied properly, right? I mean, there's other things that I want to say about this slide, but I will not say that because I like Mario. So, and I mean, sometimes people think that it's only the, like if you have a tandem dye, this problem will only arise in the donor, for example, channel. So like PE versus Texas Red PE. So yeah, so that's where it would be. But actually, no. So this is an example from the Cytometry deck, right? So this is the example where I have P-Dazzle versus BUV 661. So if the wrong tandem was used to unmix, you would see the problems far away from the area of the emission where you expect it to be affected. And then, for example, here, these are close, but C-fluor yellow green 584. I think it's B. It's B, right? Is it B? I think it's B. But anyway, so you will see it like badly affecting things. So this is not compensation. This is unmixing, but same rules apply, right? And then this is like, I haven't seen this for a while, right? But don't use FITC controls to control for your GFP, right? Or vice versa. Yeah, so this is not happening. But I mean, yeah, all these dyes are different. Sometimes you get someone coming from the woods and they're like, oh yeah, but it's all the same, right? So no, it's not the same. But I mean, this is hopefully in the past, right? All right, and then this is an example for like sorting problem. I still see people sometimes showing the histograms as they were gating for sorting, and makes me cringe inside, right? And I mean, histograms are only good for reporting data. Just remember that. Like they're perfect if you want to bring attention of the audience to one thing. Histograms are perfect for that. They're only for visualization purposes. They're not good for actually looking into the data at all. It's not okay. All right. So you would mask your signal, right? So this is like the old side is like, yeah, use another channel, don't use it. So the histograms will hide your data. But honestly, it's like anytime you use the low dimensional data when you have high dimensional, low dimensional representation, you may not see the population that's in high dimensions. And I mean, I know that like I write it sometimes in my paper. It's that, oh, we found a cluster that only exists in like 5-dimensional space. It's actually true, right? I cannot put a 2D plot that will gate on that population, but it's certainly real. And I mean, like with spectral sorters, you can actually sort that population. And yeah, you can get it. All right. So yeah, so that was the first part. I went through that like really quick because I wanted to save time for something more interesting. Okay. So, but I want to say that as a, when I wear my like core director hat and I do trainings, I actually like to make my users to go through this kind of cases because it's like, it's this pleasant feeling of superiority that like you think someone else is an idiot. Like it's a very good teaching tool, you know? So I would like, this is like, this is what I use in my training. So this is the case, right? So someone comes to my core and they want to analyze and sort tumor infiltrating mouse cells. Some of them have GFP. And they bring the digested tumor, stain it with CD3 APC, CD4 PSI7, CD8PE, and then they wash, bring it, and then they bring some controls. There's GFP somewhere. They say, well, this is unstained cells, but they actually have GFP, right? And then they stained me the beads with, you know, FITSI, and then they have like leftovers from another person's experiment. They bring it. It's like, well, that's as good as new. It's actually the same cell type, you know, like it's very important to have the same cell type. It's the same cell type. And then like another, like they have like the PE beads, right? And then I'm like, okay, so tear it to shreds. Like, what is wrong with this picture? And they are happy to come up with like all the ideas of like, yeah, you need to have the live dead digested samples, have a lot of dead cells, right? You need to use FC block, especially in the mouse. You know, they don't have the unstained control. They use FITSI beads for GFP, and then they have the RON tandem for their CD8, right? And then like there's one control that looks good. Right? So, and that kind of goes through you through the whole like batch of like things that I want them to think, and without them making these mistakes. So it's a good teaching tool. All right, so now more interesting stuff, okay? So, and then I will ask you what you think, like what's going on here. So here's the case one, right? So this is unmixed spectral data, okay? And obviously what happened here, you can guess, like there's something wrong with this unmixing. Like it looks not, like there's lopsided populations that things look not okay, right? So, and then this is cytic data, but I've seen the same thing, I think, in my core. And there's another like very nice poster here that I saw that I said, like, I want to use it in my next talk, like in this place. So on mixing artifacts. So what I see next is like, someone says like, I fixed it. I spent some time on this table that looks like spillover table, and it now looks so good, right? I mean, it looks good to me, but the problem is that it's actually, this population should be like 4%, and it's huge. Say, and they're like, well, yeah, I'm not sure why I'm having this CD1C population. And then they're stuck, right? Because they're like, okay, what else should I actually tweak to get it there? So, so they applied a lot of correction to their metrics instead of using the proper controls. So especially like, I mean, I don't know if you can do it with the Symphony, like Spectral Symphony, because it's a PMT-based machine, and usually, like you say, that your controls should be run the same day. But on the spectral system, right, you can probably, like, if you recorded poor controls or no controls at all, you can run them next day or the day, like on the APD-based machine where you have more stability over days. You can run it a few days later and still apply that to your unmixing. So if you messed it up first time, don't mess it up second time, okay? And so, yeah, so this is the proper controls used, right? So this is how it should look. And I mean, unless you know how the pattern should look, you would never figure out like which of these two is actually correct, especially like this two, right? They look very, very real. I mean, I wouldn't guess, right? Because it's not my experiment, but the controls, like, don't guess it. Okay, so the controls will help you with that. All right. And then this is another example. This, I've seen that as well. So someone is using something that's not optimal for multiple reasons, and they see that and they're like, well, these numbers are actually pretty low. The corrections, like maybe 6% is too much, but 4%, 3%, it's like, it's a little bit, it's not much, right? So there's actually, I don't think there's a good measure of like how much is too much here, because this is how it looks with the same percentages when your sample is a little bit brighter, right? So, oh, good. So this numbers, you all know, like the compensation matrix numbers mean nothing. How many times you told your user, yes, your compensation says 75%, but that's okay. Your data are correct, right? So, but this is correction on top of unmixing. So this percentages, they should be all zero, right? If you need a small adjustment based on like a very strict set of like what else you could do before you get to this adjustment, don't say, well, it's actually a small number, because you still know that you did it, right? So you have to have a very good explanation why you did that, and you need to report it. If you're publishing it, it has to be reported. Okay. So yeah. Okay. Now this is another case. There's an unexpected staining pattern here. So I have an FMO and I have the CCR antibody, and I see this like awkward high population. And I don't know if it's actually real biologically. It backgates to normal cells, like just nice live cells. Okay. Any idea what's that? Yeah, that's our aggregates, right? So so one thing that people always say is like spin down your antibody before you actually take the antibody out. Now, I had an experiment. I don't have the data, but I have an experiment when someone inherited leftovers from someone's panel. So they ran a staining where they used like last 5 microliters from every tube. So my advice is make your muster mix first and then spin it down. Okay. Don't spin every tube, because the last time the person who pulls out the last drop of that tube is kind of getting all the crap that people spun down over, months on that aliquot. So don't do that. So we did that. We spun it down, and we got this. So it got much better, but I wasn't satisfied because it's still like, I don't like this. So I got a new antibody of the same lot from the vendor, and that was BD, and Ravi, who gave the talk today, actually provided me with that. So thanks to him. So, and it didn't help. So I was like, okay, that's something wrong with the conjugation. So I waited another 2 weeks and they had a new lot, and then I got a new vial. So that actually worked. So that fixed it. So sometimes it's like you spin it down, but it's still there. So that means that this conjugation was botched. So yeah, nothing can be done. All right. So another thing, it's more on mixing artifacts. Okay. So here, I mean, I don't know, like, I don't think it's possible to guess what was going on here, but you can obviously see that something was deeply, deeply wrong, right? So let me give you some clues, right? So this is the antibody, the BV785 that was run on the beads, and that was in this Aurora library from another antibody batch. And then this is BV421 run on there. So you can guess what happened here, right? So we have the tandem degraded, okay? So that's kind of an extreme case. I haven't seen much of those. I don't know what they did to this poor antibody, but I mean, I've seen degradation, but this looks like almost like, there's way more BV421, like 1 log more BV421 than 785 in this mix. So so that's kind of like an absolutely horrendous case. But here I have the tandem degradation example. This is actually from ICCS newsletter. So they highlighted that they have the APC H7, and they usually like the clinical panels are usually small. It's not really like, you don't really do like any unmixing of like high-end panels. So that was maybe like a 8 color, right? So something, and then they're seeing this signal there in the APC channel. And this is my example. So that was a very unfortunate result, because we used the CD34 PECI7 because we wanted to have like a very good separation of different levels of CD34 expression in the hematopoietic stem cells. And then we were looking at super rare GPI80 population that should be double positive in this. And then we realized that no matter whether we add GPI80 antibody or not, we're getting the signal there. So, and we actually went through 3 different lots of this antibody from 2 different vendors, and all of them had enough noise here that we had to just abandon that combination in the panel and just move the 34 elsewhere, because I kind of like, I forgot honestly that this can be that bad, and I didn't realize how small this population will be because it's not like it's not my primary research, so I was just consulting someone. So, and they were using this, like this was their favorite antibody. I wouldn't probably put it there, but anyway, it's not a good excuse because I mean, anyway, we had to abandon that because it was not working. But watch for that, especially if you're like, once you get to like rare event detection, like all these things that you kind of like, oh yeah, that's like very little noise, they start to be like, this is the actual readout that you will get. So it can be really, really hard. All right, so for those of you, like other than Alice, who is doing mass cytometry? Some people, okay. So we have some mass cytometry examples here. I mean, a lot of things that I said actually are applied to mass cytometry. If you don't use FC block, you're as messed up as flow people, right? But so this is a donated data. So that was really cool. That's cool to see. So this is a mass cytometry experiment, right? So we have Ki67, which is a marker for proliferation. And these are cells that have healthy PBMCs, CD45 positive gate, right? And then they have CD8 cells, like a nice population of CD8s, and they all appear proliferating in the sample without any stimulation. Okay, so and in mass cytometry, we really there's no classical spillover problems, right? So you're like, what is going on? And it's like nicely shifted, right? So it's not like any like lopsided, like it looks, but it shouldn't be true, right? So healthy donor. So that's the incorrect staining pattern. Do you know what happens here? Any idea? You know it, like you know what happens. So I'll tell you. So small hint. So this is the 146 newodium, and this is 162 molecular mass. So 16 is the difference, and 16 is the molecular mass for oxygen. So some of the lanthanide family ions, they form really strong oxides that actually survive the plasma, and they appear as the metal plus 16 in the data. So again, like this is the data from fluid. I mean, now it's not fluid, right? So this is the publicly available data, and again, you can see that these numbers are actually very small. It's not like 20% compensation, but that was enough for that population to get shifted. So the take-home message from that was like, don't use this combination for if you're not putting it like on mutually exclusive channels, like what you should do with flow cytometry, right? So this is another example. This is actually from my experiment. So we were looking at, this is IL-17 on different subsets of NK cells and T cells. So this is CD56 versus IL-17. And I was specifically going and checking whether I have any other issues with the data. And I have this diagonal shape that is very familiar to people who do flow cytometry, right? And then in the same data set, I'm looking at gamma delta TCR versus CD57. And again, I see this that would look like uncompensated data for me, right? If it was flow cytometry. But the problem here is that like, okay, it's mass cytometry. So is it actually a real population? Like, can I have like CD57 positive gamma delta low cells? Yes, I can. So again, these two are exactly, they look like spillover because they are spillover. Okay, so this is, and that's the isotype impurity. So you can't really source the pure isotope for, not isotope. My word does the same thing, right? So I start doing it. So isotope impurity happens because you can source the ultra clean isotope. You will still always have impurity in certain metals, and you would have it. It's a fixed value because you kind of like, you know how much impurity you have. So you can actually apply, there was a beautiful paper from Bottomiller Lab, how you can apply compensation to mass cytometry data, and you will fix this, but you would get some spread just because of the nature of it, but you can fix it. But if you don't do it, then you will see something like that. And again, like if you design your panel smartly, you would not have this problem. But this was put together based on like many different decisions, and this was not really the combination we were specifically looking for. So I was looking for like bad examples in the data. But my problem with this is that, as you saw today earlier, the pan gamma delta antibody is problematic in terms of like binding, like if you have CD3 in the cocktail, it doesn't bind that well. And actually in humans, it's the specifically V delta 2 gamma delta cells that are kind of low when you stay with pan gamma delta antibody. So this can actually, like this population can be CD57 positive cells, but they could also be V delta 2 cells that we don't have V delta 2 in this panel, right? So, and the reason I'm putting it is like because the person who analyzed the data actually gated gamma delta T cells like that. So they gated CD3 versus TCR gamma delta, and they put this gate, and it looks totally legit, unless you know that this tip is actually coming from CD57. Okay, so again, like multidimensional data, if you stack it into 2 dimensions, you may have some things hidden, right? So, okay. All right. So 28 color data, and it's problematic. And it's a person who knows how to do stuff. So it's not like you're like, okay, what was going on here? I'm not sure. So, and then I start to look at like all the, like there's technically like all the spectral flow didn't give you any like big error problems, right? But it's definitely problematic. So the key was that they had BB786 versus super bright 780 in the same panel. Okay, so these dyes are nearly identical. Okay, so you can get the spectra that look almost same, right? And I mean, the similarity index is 0.98. So don't do that. I mean, the unmixing algorithm is completely unbiased. It will unmix. It will unmix if it's like I don't know if there's actually like in spectral flow, you cannot unmix more than 0.99 or something, but technically the OLS algorithm will unmix anything that's not 1, right? So it will unmix. You will not like the result. And then sometimes people say, well, but what if I use this like approach where I have a B cell marker on one of this and I would put a C cell marker on another? And that's like, I know that this term is sentinel panels, like BD, I think called it sentinel panels a long time ago. So I use that term. So if you do that, that's totally fine. Use the same color. Instead of introducing all this spread into your panel, I have a panel where I have CD8 and IgD, both like FITSy. So then it's very clean. I have no spread, and my mixing works absolutely nicely, right? I mean, in interpretation, I need to remember that if on my UMAP, if my B cell cloud like lights up for CD8, it's not because they have CD8, right? But other than that, it also absolutely is a legit way. All right, so here's another example. So this is the complexity table, right? So there's a lot of ones and like a lot of identical dyes, although they seem to be fine. So actually, this was how the spectral looked. So this was just someone messing up with the plate. And they just stuck it in there, they read it, and then they're like, oh, something happened there. So probably maybe when they were washing or something happened. So even if someone is like so experienced and is making this large panels, there's still like dumb mistakes happening sometime. And you just, make sure you watch for that instead of like, because I've seen people just like, they don't even look at how they're, you click through the algorithm, spit out the data set, export, done. It's, I mean, we have 15 minutes till the cocktail hour, right? You don't have time to look at your controls, right? So, no. All right. Okay, another example. This is actually mass cytometry data, but it doesn't matter, right? I'll tell you, it doesn't matter. So this is the PBMC sample. This is from Flow Repository. I was looking for this problem and I found the data set with that problem pretty easily. So PBMC sample from the same donor run 2 different days. And that was supposed to be like a, internal control. Same experiment, same instrument. So they're looking at the awkward population that's like RA high, CD4 lower. So any guesses what that is? Mass cytometry. So no compensation issues, no. Okay, I mean, it's kind of hard to guess. Okay, so it's actually someone didn't watch for the time parameter and then they were doing their gating. Okay, so remember that you want to look for stability of the instrument, and I don't know a single instrument where there would be a built-in, marker for like, oh yeah, I flag the sample because something is awkward with the time parameter. I don't think anyone is doing that. If you put your data in FlowJo, you would actually like, there's this round, there's circle next to the name, and it does very simple, no AI, right? Very simple scanning, and actually can point you to the samples that are maybe problematic. But yeah, your time gate should be your first gate in any gating. And you can use, there's several algorithms. You can pick the algorithm that you like to do this cleanup automatically, but do something, right? At least like check out, and especially those people who like HTS systems. So I don't use one for a reason, right? But the BD HTS systems are, sometimes there's some good ones, but there's sometimes the ones that have a lot of problems with the stability. And so this is a very common pattern. So it can look like there, you can see something that in your, like, the acquisition going upward till the end of the sample when you're running out. It can be a clog. It can be something that